microphone and start playing it out. It's really hard to hold the microphone and play it. It's taxing, cognitively taxing. We'll actually talk about that when we get to the topic of attention. And why is it that when we try to combine different tasks together, sometimes we can do it very effectively, other times we can't. Topic that is very important in cognitive science. All right, so last time, this was, I think, the last slide, or the second to last slide that I showed you, and we've been talking about some of the basic assumptions in cognitive science, and these are two really important ones. Right? An assumption is a statement of a position, if you will, in a scientific discipline. That is something you have to accept in order for you to be able to do that particular kind of science. And in cognitive science, we're no different than any other kinds of science. And that there are some assumptions. There are some things that we just basically assume are true before we can even get started talking about our science. And the two in particular that are important for us are reductionism, right? The fact that if we're going to study the mind, if we're going to try to understand how the mind works, we have to break it down into its fundamental parts and see how those parts interact with each other. And that is basically a field of philosophy, right, that says that this is something you can do. You can reduce things into their elemental parts in order to better understand how the system works. Another assumption, perhaps even a more important one, is this one, materialism. I boiled this down to the mind is what the brain does. In other words, the mind isn't something mysterious thing that, is mysterious, right? Because we don't fully understand how it works. But it's not mysterious in the sense that it is mystical, right? That it's something other than biology. So in our view, in the view of cognitive science, we basically make that assumption, right? That the mind is something studyable because it's biological. It is in the material world. It's something tangible that we can point a finger to. Maybe not directly, but certainly indirectly. So the idea in materialism is that if we can understand the elemental processes of the brain, we can understand the mind. All right? So those are the two assumptions. And I start with this today because this wasn't always the case in science. Right? It wasn't always the case in science that we accepted the premises of reductionism and materialism. In fact, there's an older school of thought that predates these two particular views, the views of materialism and reductionism. And materialism and reductionism actually came about in philosophy and in science as a response to these earlier ways of thinking about science. So yes, it was a response to earlier philosophical points of view. And in particular, most important for us, is one referred to as dualism. How many people here have heard of dualism before? You've had a philosophy course right now where dualism is talked about. So dualism, boil it down really to its essence here, summarize what we're talking about, was originally proposed by this guy, René Descartes. And I'm sure most of you have heard of Descartes before, very famous philosopher, also famous mathematician, French mathematician, working in the 1600s. And Descartes played a really fundamental, important role in helping cognitive science get started. In fact, you might even argue that he was one of the first cognitive scientists. And you might know him from this famous quote of his, "I think, therefore I am." Right? Clearly interested in the mind, right? The thought processes. And Descartes was a major figure in thinking about early notions of cognition. So Descartes is what we refer to as a dualist because he imagined that human existence actually exists in two different fundamental ways. There are two fundamental different things that are involved in human thought. The first, of course, is what he referred to as the material world, right? What we were just talking about, materialism, right? That there is this biological, physical world that we exist in. So Descartes obviously acknowledged that. In fact, he made many early observations about human anatomy and physiology, the human brain. Here you see a diagram of his showing the visual system with the two eyes, and he realized from dissecting human brains that the human visual system actually has a cross pathway. We'll talk more about that next week when we start talking about perception. But he made some really keen observations about the body and the brain in particular. And in this diagram here, he's basically showing us that the visual system allows information to come in here, and it allows us to move the muscles and, for example, point to some object that we're looking at. For Descartes, though, that was only half of what was going on, right? What was going on in the material world of our bodies and our brains. Certainly our brains are responsible for telling the muscles to move and for basically registering the input of sight. But Descartes, of course, remember when Descartes is living, he's living in the 1600s. And it's pretty hard, even for a hard-nosed scientist at that point in time, to ignore the really potent set of arguments coming from other philosophers and from the church in particular, that there was this other kind of thing in the world. Maybe it's not really in the world. This thing that Descartes referred to as the soul or the mind. So in Descartes' view, while the brain was responsible for controlling the body and for registering sensory inputs, the actual source of our mind, of our thinking mind, was actually something outside the brain, as illustrated here, right? You see a little angel driving this human's thought process. So for Descartes, and the reason we call this dualism is because he imagined there was this other thing, the soul or mind that existed on a different plane, on a non-physical plane, that was responsible for what we call the mind. And hence the term dualism, right? Two different dual systems interacting with each other. Now, needless to say, dualism, as calculated here, has a problem for modern psychology, right? If Descartes is correct, if the mind is not really a function of the brain, the way we assume it now in cognitive psychology, then we have a problem, right? Because this thing that we're interested in, this organ's ability that we're interested in, the mind, can't really be studied using the physical body. Right? It's on a different plane, it's in a different universe, if you will. And therefore is not amenable to direct observation or direct study the way that would be necessary if we actually were going to scientifically study the mind. So this is the problem of dualism, right? Is that it proposes that the mind is something separate from the body and operates in a different plane, a different universe with a different set of rules, making it virtually impossible for a physical body to study it. Fortunately for us, sometime in the late 1800s, the very first psychologists, right, the very first people that called themselves psychologists, rejected this notion, the second notion of Descartes, right, that the mind was something separate from the body. They in fact argued just the reverse. They argued from a materialistic standpoint that no, although we don't understand how the mind works, we have pretty good evidence that like the rest of our biological functions, the mind is biological as well. And so starting in the late 1800s, you start to see psychologists, largely in philosophy departments, right? The psychology departments didn't exist yet. Largely in philosophy departments, starting to make arguments much more in the scientific realm than in the realm of normal philosophy, right? Where argumentation and logic are usually used. Instead, doing experiments and trying to prove various different aspects of how the mind works. So yes, these early psychologists, these early cognitive psychologists really, these are the first cognitive psychologists. They effectively were arguing that the mind and body are part of the same material world. And that in fact, the brain gives rise to the mind. Yeah, I just pointed out, these were the very first cognitive psychologists. So cognitive psychology has deep roots, right? It's right there at the very beginning of psychology. The very first people that called themselves psychologists, the German psychologist by the name of Wilhelm Wundt, actually started the very first psychological laboratory in Germany. And shortly thereafter, William James, a famous American psychologist, wrote the first textbook in experimental psychology. He was at Harvard in the late 1890s. These two psychologists and many of the students that they trained were really the first, were the pioneers, if you will, in psychology that believed that the mind is something worthy of study. Yes, but how does this work? Again, how do you study mental activity? Right? One of the obvious problems, the problem that Descartes pointed out is that you can't see the mind, right? It's not visible. Even if you open the brain up, open the skull up and you look inside, you can't see a mind. It's not something that has a visible trace of its own. Yeah, so thoughts are taking place in this black box, if you will, that's inside our skull. And certainly this was a huge problem, right, in Descartes' time and even in Wundt and William James' time, that we really couldn't see the brain. The only way you could see the brain was post-mortem, after someone had died. You could look at their brain then, but of course, then the brain is somewhat inert. So this poses a real problem, and it's a continuing problem today for people trying to study cognition, is that the organ that we're trying to study, or the organ that produces what we're interested in studying, doesn't produce any visible evidence of what it's doing, right? In fact, if you look at a brain of someone that has just died, say 15 minutes ago, and you look at someone whose brain is working very, very well, they don't look any different. There's nothing, certainly that the naked eye can detect of a working brain and a non-working brain. They look pretty much the same. Now, over time, of course, that wouldn't be true. A brain of someone that's passed away is eventually going to start to look different as well. But initially, for the first hour or so after that, the brain looks the same as the brain of someone who's alive. You'd actually have to zoom in on microscopic things going on in the brain in order to see real differences. And those differences don't really produce any kind of physical, obvious differences that you can see. So we can't see what's going on in the brain. Not directly, anyway. So what is a psychologist to do? What are the cognitive scientists to do if you can't see the material that you're trying to study? Now, obviously, lots of other sciences you can't see what you're studying, or you can't see it directly, right? Physicists, in general, certainly, one thing that they were first discovered, couldn't see things like atoms, right? They had to infer their existence based upon observing changes in things that they could observe. As it turns out, that is a good analogy for how it is we're able to study cognition. So even though we can't see directly cognition working in the brain, we can infer its existence, as it turns out. And the first psychologists to try to do this used this technique in order to infer what the mind was doing. These were psychologists that referred to themselves as introspectionists, and they used a technique called introspectionism. Nothing really fancy about this term really, because in fact, it's something we all do. Right? Introspection just basically is looking in on your own mind, right? Inspecting your own thoughts to try to figure out, well, why am I thinking the way that I'm thinking? The only difference between you and an introspectionist is introspectionists generally, when they first got started in the late 1800s, Wilhelm Wundt was an early introspectionist, was they tried to come up with a principled set of procedures for introspecting on their own minds. Right? Trying to, if you will, come up with agreed upon ways that introspectionists could look at their thought processes and try to agree on what it is that's going on in the mind. Common sense, right? I mean, I would tell you that even modern cognitive psychologists use their introspective powers when they're thinking about how the mind works. And it's almost human nature to do this, right? To inspect our thought process. Introspectionists imagine that this could be the scientific method, right? Rigorous introspectionism following a set of rules that could be used for studying the mind. So, for example, if you were interested in the topic area of problem solving, right? One of the areas of higher level cognitive psychology that I talked about last time. Problem solving, of course, is this remarkable ability that humans have to take in information and use that information to solve particular problems that they're presented with, right? So solving a math problem would be a really simple example of this, but of course, taking one of my multiple choice questions would be an excellent example of problem solving, right? Figuring out the right answers to obscure questions. Something we're really good at. And the introspectionists said that, well, you can study that because as people do something like, for example, take an exam, they can chronicle, if you will. They can write down all of the steps involved in their thought processes as they come to answers. So yes, you would write down all of the steps involved in solving the problem. Seems reasonable, right? As a way of at least getting started in this new field of cognitive science, cognitive psychology. And of course, critical would be comparing your observations to those of other introspectionists. And of course, we do this in everyday life as well, right? As we view the world around us, we make introspective observations about what we think is going on, and we compare that to what our friends and colleagues' introspections are, and sometimes we agree, and sometimes we don't agree. But this is basically what the early cognitive psychologists were trying to do, was formalize something like that, something that we all do all the time. Well, if you know anything about the history of psychology, you know that this approach failed spectacularly. Right? So while there was a huge amount of enthusiasm in the late 1800s and early 1900s, primarily in Europe, but also in the United States as well, about using this approach to study the mind, after just a few decades of people engaging in this introspective approach to studying the mind, early psychologists started to become very disenchanted. And in particular, young psychologists in the United States, experimental psychologists in the United States, got very disenchanted with this perspective. And the reason is well, these are some of the reasons. First was this one, right? The same one you probably have had if you introspected on particular thoughts and compared them to a friend or colleague, is that frequently you don't agree on what it was that happened in your mind when you did a particular thing. Well, I did it this way, and someone else goes, no, when I solved that problem, I did it this way. And this kind of disagreement was happening very frequently amongst the introspectionist community. They couldn't agree on what it was they were actually quantifying and trying to observe. And perhaps even more important was this, the problem with not having a method, if you will, for deciding who's right. I mean, these are my thoughts after all. How can someone be more right about their introspections than me? And so there was a big struggle early on in experimental psychology with this kind of methodology. And then lastly, this observation. The possibility that there's probably a lot going on in our mental world that we're not able to introspect on. If you were listening last time, I gave you a hint of one example in cognitive psychology where this happens. Face recognition, right? The way we recognize faces, for example. And there are many, many other examples. But the point is that if I ask you all to break down what happens in your mind when you recognize a familiar face, right? You're walking across campus, you see your friend's face, and you immediately recognize who they are. Maybe their name pops into your head. What happened? What were the mental operations that allowed you to recognize that face? I mean, most of you would say, I have no idea. It's just automatic. It just happens, right? You see their face and boom, you recognize them. It seemed pretty obvious, even to these early cognitive psychologists, that a whole lot of what we'll call cognition happens at this level. It happens outside of our ability to introspect into it as to what's happening. This is referred to as the impenetrability problem. You can't penetrate these processes. They're not open for cognitive conscious inspection. And there's probably some really good reasons for that. We'll talk about when we get to the topic of attention. So yes, the possibility that a whole lot of what goes on cognitively is not introspectable was something that these early cognitive psychologists realized was likely to be the case. And probably the most influential person in this area was Sigmund Freud. Sigmund Freud was not a cognitive psychologist. Right? He's the father of modern psychotherapy. Psychoanalysis is his brand of psychotherapy. What Freud did contribute to modern psychology was this notion of the unconscious. Right? The idea that there could be many, many cognitive processes that are not conscious, we're not aware of them. And the fact that that may be the case started to put additional bits, if you will, into the arguments of introspectionism as a way to study the mind. So, what happens? Well, at the dawn of the 1900s, a group of young American psychologists started to rebel against the introspectionists who basically controlled all of the major universities in Europe and some of the ones in the US as well. And what these young psychologists did was they basically said, wait a minute, this brand of psychology that you're practicing isn't really scientific. Right? It's not hard-nosed. It has no real data. You have no way of replicating experiments. And all of the arguments about what makes up a science. What do you have to be truly scientific? And they argued that this was the case for introspectionism. And psychology was going to die as a science, right? It's going to end up back in philosophy departments, being argued back and forth, as opposed to being a real science. Interestingly, along the way, as they rejected introspectionism, many of them eventually also rejected the idea that the mind could be studied. Sounds a little like Descartes, doesn't it? They weren't like Descartes, though. They certainly didn't imagine that there was a mind and that it was on some different plane. They just said, look, it's just too hard to study it. If there is such a thing, it's too hard to study, and therefore it shouldn't be what psychology is looking at. It shouldn't be the source of study for experimental psychology. So yeah, eventually you have many psychologists that call themselves behaviorists actually saying, you know what, the mind is not important, and really, who cares? Right? It probably doesn't, maybe it doesn't even really exist. Maybe it's just a figment of our imagination, so to speak. So yes, what's important then? It's not the mind. What are we going to be studying in psychology? Well, as the name suggests, right, behaviorism suggests, what these behavioral psychologists were interested in and what they felt passionately about studying was human and animal behavior. Now, why is that? Well, because while we can't actually see what the mind is doing, we can see people's and animals' behavior. Right? We can observe what people are doing. Right? I can observe someone stroking their beard. Right? It's a behavior that's observable. I don't know why they're doing it. I don't know what in their mind made them do it. But I can certainly observe that particular piece of behavior. And this behaviorist psychologist said this is what we should be studying. Because you know what? We can quantify that. We can measure it and quantify it. And after all, that's what science is about. Measurement, quantification. And basically coming up with theories and hypotheses based upon these kinds of observable endpoints. All right, so this is the basic behaviorist notion, that you can only study what's observable. Right? What's observable and quantifiable. And so what's the basic idea here? I mean, I just said something about someone stroking a beard. Actually, behavioral psychologists weren't initially studying things like that. What they were interested in what they decided was quantifiable and replicable, right? That's another important feature I forgot to mention earlier. Being able to replicate your experiments, right? Do the experiment again and get the same result. A really important aspect of psychology, of any science, is replication. And what they discovered was that they actually could, by carefully studying behavior, carefully observing the behavior of human and animals, they could do replicable experiments. And what they became interested in, what they referred to as behavior, was actually the linking together of stimuli and responses. So stimulus, right, is any kind of event in the real world that causes a change in behavior. And presenting animals and humans with stimuli, and then observing the response of the animal or human, measuring some aspect, quantifiable aspects of that response, is really what behaviorism is about. These so-called SR pairs, stimulus-response pairs, according to behavioral theory, this is basically what learning and, well, really the whole point of behavior is how these stimuli and responses get hooked together by reinforcements known as rewards, or in the case of negative reinforcements, punishment. So these early behavioral psychologists started observing behavior of humans, but mostly animals. Most of this work early on was done in animal laboratories. Was interested in basically uncovering what it is about the relationship between stimuli and responses that actually is learned by the organism. It's acquired by the organism. And they referred to this, basically this relationship between stimuli and responses and the reinforcements as operant conditioning. And all of you have heard about operant conditioning before, I'm sure, right? This is the kind of classic experiment done first by John Watson, an early behavioral psychologist, but eventually made famous by B.F. Skinner. Many people here have heard of Skinner before. Yeah. Very well-known name in psychology. He was alive up until about 20 years ago. Still doing experimental psychology, behavioristic psychology. What Skinner did, of course, was he put, usually rats, but he sometimes also used pigeons and sometimes humans as well. He would put them into some type of controlled environment, which is now sometimes referred to as a Skinner box. So this would be a small box, maybe twice the size of a shoebox, where you would have a rat, and the rat would have some type of, there would be a wall in the box, where there might be lights, for example, and these lights would be different colors. And there was also usually a lever, and rats can easily be trained to push a lever, particularly if pushing the lever results in some kind of reward, like, for example, a little bit of juice or a small pellet of food. So in a classic Skinner experiment, what you do is you food deprive the rat for 12 hours, right? You don't give them their dinner the night before, and in the morning when they come into the lab, you put the rat in the box, and it's very easy to teach the rat to push the lever to various different kinds of stimuli, and to not press it to other types. You can teach a rat to do this. Maybe some of you have even performed one of these experiments before. I actually did when I was a student here a number of years ago. We trained rats, and really you can do it in about an hour. A rat can be conditioned, can be operantly conditioned to learn how to press a lever for particular stimuli that they're presented with. In other words, you can do this experiment. You can show that their responses change as a function of the stimuli after the rewards that you give them. All right, so this is basically the basic underlying notion of behaviorism and operant conditioning. And this particular approach, well, basically what behaviorists would like for you to believe is that human and animal behavior can best be understood by explaining these links between stimuli and responses. They argue that all human behavior and all animal behavior can be understood in these terms. If you can understand the stimuli, the response, and the reinforcement contingencies that give rise to learning, you basically have explained human and animal behavior. This particular perspective held sway for over 50 years in American psychology and really in most of experimental psychology around the world. Most people that call themselves experimental psychologists, in other words, psychology professors at university doing research, were doing this kind of research. They were trying to understand the relationship of stimulating responses and reward contingencies. Pretty amazing, really. I mean, and it really only is somewhat more complicated than what I just described here. So really, for decades, we were involved in, refining and better understanding how this works. And you can still see people doing this. I mean, obviously, most of the people that do animal training use the principles of operant conditioning to train animals in really elaborate, complicated kinds of behavior. I mean, there's tons of YouTube videos where you can see rats being trained to do amazingly complex tasks. And they basically were trained using these operant training techniques. They could build one little behavior, and then they add another one onto it. They adjust the reward contingencies, and you can do amazing things with this. You really can. The question, though, of course, is this all there is to psychology? Is this all there is to understanding human behavior? Well, starting in the 1950s, again, a few young, renegade kinds of psychologists. Kind of like those original behaviors, right? It's always young people in the field that they're getting trained, they're in a PhD program somewhere, and they're learning about their field, and they're probably being trained in behaviorism, and some of it starts to not make complete sense to them. Particularly because also in the 1950s, behaviorists started to make more advanced claims. So, for example, in a really famous book written by B.F. Skinner called Verbal Behavior, Skinner argued that you can explain the entire faculty of human language, in other words, how babies acquire language, strictly by appealing to operant conditioning techniques. He believed that language was completely the result of behavioristic operant conditioning. Babies are reinforced by their mothers to say certain sounds, and then to make words, and then to produce sentences, and this is how language is learned, strictly in the matter of operant conditioning. Well, a young linguist at this time, right around the time Skinner's book came out, a guy by the name of Noam Chomsky. Anyone ever heard of Chomsky before? Probably a few of you. He wrote a book, actually he wrote a series of essays that became a book, challenging Skinner's notions. And then they had a famous set of debates at Harvard where Skinner and Chomsky argued about whether or not human language can really be explained strictly by operant techniques. And Chomsky is widely recognized as having won those debates because there were many, many questions about language that just did not seem to fit into the notions of operant conditioning. There's way too many observations about what babies can do, what babies can learn, and how complicated language is. I'll just give you one little piece of this, right? So babies, by the time they're 1 year old, usually are saying their first word, and they usually can comprehend many more. By their second birthday, they're making sentences, and they have vocabularies in the thousands of words. Right? Pretty amazing transformation in 1 year. In fact, by the time children are about 3 years old, their competence in the language is nearly complete in terms of their ability to make complicated sentences and understand complicated sentences. And yet, how many of you here can say you've learned another language as an adult in 3 years? Because you could call yourself competent users of that language. Anyone here can say that you've learned a second language in a period of 3 years? I mean, there may be a few people, right, that have been able to do it. Well, I venture to say that all of you are cognitively more capable than a 1 to 3 year old, right? I mean, most of you would like to think that you're able to do much more in the cognitive domain than a 1 to 3 year old. This is what Chomsky pointed out. He said, look, babies are not cognitively competent, right? In fact, they're incompetent. They can barely do anything. And yet, they learn language. In fact, the metaphor you frequently hear is that they learn language like they're a sponge, right? Like a dry sponge put in water. They just soak it up. And they don't even seem to be trying, right? Those of you who tried to learn a second language in 3 years were probably trying really hard, right? Putting lots of your cognitive effort into learning that language. And yet, you still had a difficult time doing that. So how could that be if learning a language is strictly stimulus and response linking? Right? How could it be that a cognitively incompetent individual can learn a language passively by just being around it, whereas a cognitively competent adult can't do it nearly as efficiently? Well, Chomsky had lots of reasons for that, and we'll get to those when we start talking about language. But one of them is the notion that there is probably a critical period for learning a language, right? A period during development when it's much easier to learn a language than it is later on in life. And of course, what that implies is that there's some kind of mechanism, right, in the mind, in the brain, that is set up for dealing with language. And it's not just a matter of, if you're exposed to this stimulus and this response and this reinforcement that you'll learn something. It's way more complicated than that. All right, so there are many, many other examples of these early young cognitive psychologists starting to challenge the notions of behaviorism, or at least the notion of behaviorism as having all the answers for psychology. They started to note that there are many, many domains of human existence, of human mind, if you will, that don't seem to fit into the basic operant conditioning paradigm that the behaviorists were pushing. The problem is that using SR patterns to explain, probably can see it on my monitor here, to explain why people did certain things, right? Which was very difficult, right, to explain how it is that babies learn language using just stimulus and response kinds of experiments. So, needless to say, behaviorism went down, right? In the same way that cognitive psychology at the end of the 1800s was pushed out by behaviorism, behaviorism was largely pushed out by cognitive psychology, starting roughly in the 1970s and running up to roughly the year 2000. So by the time you get to the early 2000s, you actually have a hard time finding behavioristic psychologists in psychology departments. Right? They've almost all been replaced by people that embrace the principles of cognitive psychology. Some people think we might experience a paradigm shift again. This is what we refer to as a paradigm shift, where all of a sudden, by all of a sudden we mean maybe 10 years, that's all of a sudden in the grand scheme of things, you get a big change in what the questions are in the field. So we had that big one back in the late 1800s, and then we had another one in the 1970s and 80s. And some people think that maybe cognitive psychology is running its course now, and then maybe we'll have another paradigm shift. Although no one seems to know what that shift will be to. I'll tell you though that while behaviorism has largely been replaced in modern academic institutions by cognitive psychology, there is a legacy. The behaviorists did do something really important, right? Remember back to what I said. One of the big arguments that they made early on was that you've got to have observable, reproducible data in your experiments. Right? If you don't have that, you're not a science, you're a philosophy. So the observation of behavior as the primary methodology is something that the behaviorists introduced and that the cognitive psychologists actually embraced. Early cognitive psychology embraced the notion that yes, behavior is all we got, right? It's the only thing we can really observe and quantify as behavior. Also, the notion of using rigorous methodological testing of various hypotheses, using small changes in experiments, and that's a mouthful. Basically, what this means is you're careful in the way that you do experiments. You do them step by step. You form hypotheses based upon the results of one experiment to do your next experiment. Behaviorists did this for psychology. They made it, in some cases, painstakingly. They did small little changes from one experiment to the next. This is why they lasted for 50 years, right? Because they did literally tens of thousands of experiments manipulating the stimulus response characteristics. But in doing that, they made it I think, pretty obvious and pretty clear that this is the way you've got to do science. You've got the way you've got to do psychological science. You've got to have rigorous methods and things that you can observe and quantify. All right, so we're now going to switch gears a bit. We'll continue to talk about the history as we go here, but we're going to now jump into cognitive psychology. And as I told you last time, we're going to start by talking about the methods, right? These rigorous methods that the behaviorist gave us. The observation of behavior and rigorous methods. So of course, as we've already talked about, this is the organ that gives rise to cognition. This is the human brain. Easily in the palm of your hand, and although you really couldn't, because the brain in its normal state, you've probably seen pictures of people holding brains. Those are brains, those mortar brains obviously, that have been fixed. They've been injected with formaldehyde or some other substance to make them more firm. The actual human brain is about the consistency of jello. So you probably really couldn't hold it like this. It would be a bit runny. Here's what the brain looks like on the inside, and we're not going to spend a lot of time talking about this. We're going to have to take a course in cognitive neuroscience to really get into all the nitty-gritty details. But what you're looking at here is that same brain. If that person were to turn and look at you, and if we were then to just basically slice through their brain so that we could look inside. So that's what you're looking at here. And you can see that it's probably appears pretty complicated to you to look at it here. Although a view like this really, really, really underrates the complexity of the brain. Because what you can't see are the tiny, tiny little cells that make up the brain. And there are actually 100 billion of them. 100 billion nerve cells, neurons, are in actually a very small piece of the brain. See this outer gray area here? This is the so-called cerebral cortex. The cell bodies of those 100 billion neurons live in that thin outer mantle that surrounds the brain. And we're pretty confident now. We have plenty of data that suggests that this is where cognition happens. Most of cognition is happening in those 100 billion neurons that surround this larger, much larger area of the brain that is more white. So what's the white stuff? The cells are up here. The white is actually not really parts of those nerve cells. These are a different kind of cell called glial cells. They're actually made mostly of a fatty substance called myelin. And that fatty substance wraps itself around a really long projection from each of those nerve cells, something called the axon. You guys have probably all had some basic coursework where you talked about neurons and their parts. Well, the axon is the part of the neuron that basically attaches to the cell body of the neuron. So the cell bodies are up here in the cortex. And the axons are these really long, thinner than a human hair projections that go from one brain area to another, or across here all the way over to the other side. So these are the wires or wiring diagrams, if you will, that connect all of these neurons together. And each of those axons is wrapped very tightly in some of that fatty substance, that myelin. It's like insulation. It's like a rubber coating on a wire. Right? It insulates the axon so that one axon that's sitting next to another axon doesn't accidentally communicate with it. And you can't see that here. There's so many of them, right? And imagine if there's 100 billion neurons up here, and let's say 20 billion of them are sending axons to other brain areas. That's a whole lot of wires, right? 20 billion wires. That's like as many wires as there are people on the planet. Each human brain has that level of connection from one brain neuron to others. In fact, it's way more complicated than that. Because each neuron can actually have as many as 10,000 other neurons interconnected with it. And any given neuron can communicate with thousands of other neurons. So start to do the math, right? 100 billion neurons, probably 25,000 connections on average per neuron. You've got lots and lots of possibilities. That's why the brain is so complicated. And it's probably why our minds exist, right? You need something that complicated to account for what it is that our minds are able to do. So let's just blow up one of these little regions here a bit. And this is just an artist's rendition of what that might look like, right? So now we're actually looking at the individual neurons and the axons, those fibers that connect one neuron to another. And you start to get the idea of just how complicated it is. Another view of the same kind of thing. All right, so how does that organ work? What does that 100 billion neurons organ actually do? And how can we study it? How can we study, right? Remember, we still have the same problem that the early cognitive psychologists had. We can't see the brain working, right? That was the post-mortem brain I was showing you before. We can't actually directly observe what it's doing. So how can we study it? Well, these early cognitive psychologists I was talking about in the 1950s and 60s and 70s figured out some ways to do it. They developed a technique called the information processing technique. The basic assumption here is that the brain is a processor of information. And this theory, this information processing theory, proposes how it is that the brain exchanges information between various regions and between our bodies and the world around us. So this technique, this information processing technique, has been fundamental in understanding how the mind works. In fact, the information processing approach is the basis of modern experimental psychology. This will be far from the last course you have, where professors will talk to you about how information is being processed by people and animals. And this approach, devised by early cognitive psychologists in the 60s and 70s, is the basis of that. And it's the dominant paradigm now, right? People in social psychology, personality psychology, developmental psychology, many, many different branches of psychology now embrace this basic paradigm, the information processing paradigm, as the way to study human and animal behavior and cognition. All right, so that's the information processing paradigm. What I'll tell you, though, is that the information processing paradigm has been augmented or has been added to by many, many other approaches to study cognition. And an early one that kind of parallels that very early cognitive psychology group that I talked about in the late 1800s. While those cognitive psychologists were basically formulating the beginning of cognitive psychology, there was a parallel group of people in medical science, particularly in neurology departments, that were studying human cognition using a very different approach. They were looking at patients with brain damage, patients with what we call brain lesions. A lesion is basically just some source of damage or some kind of insult that's been done to the brain. And these people were practicing what now is referred to as neuropsychology, or I'll call it classic neuropsychology because it's really harkens all the way back to the late 1800s. So what I'm going to do here is I'm just going to list a whole bunch of different techniques that have been used and devised for studying cognition over the last, 150 years. These two being particularly old ones. The basic information processing paradigm and lesion studies started in the late 1800s. In more modern times, the study of lesions in the brain, and I guess important I didn't mention, is we're not just interested in lesions in the brain and brain damage for brain damage sake. We're interested in it because when you damage a part of the brain, you damage a part of the cognitive system. And as a result, the individual is usually impaired in some way. Right? Their cognitive abilities are changed or altered. And studying those impairments are what neuropsychology is about. Now, in ancient times, 1800s, it was the case that to do neuropsychology, You imagine that someone had had brain damage, right? They were in some kind of accident, and it appears that maybe their brain was damaged. But you didn't know for sure, because again, the brain is still inside the skull. Maybe they were bleeding somewhere. There may be other signs, but you couldn't know for sure where the brain damage was. You had to wait for your patient to die, right? And then hope that the family would allow you to inspect the brain. This was a very, very cumbersome and lengthy procedure that was not very good, right, for making discoveries. So it wasn't really until modern times when we came up with new methods for actually taking pictures of the brain, of the intact brain, right? In other words, our own brains, without actually having to go into the brain itself to look. And these are what we refer to as structural imaging. And we'll talk more about that later. All right, a third kind of technique. As it turns out, starting in the 1950s, it became possible to actually stimulate the human brain. And certainly way before the 1950s, physiological psychologists were stimulating the brains of other animals, probably all the way back to the 1930s. What do we mean by stimulation? I mean, obviously, any experiment, right, involves a stimulus, right? See a stimulus and it's processed by your visual system. What I'm talking about here is directly stimulating the brain, actually sticking a wire, what we call an electrode, into the brain tissue itself and electrically stimulating the brain. So this technique, as you can imagine, has been very valuable in understanding what neurons and what groups of neurons might be doing in the brain. Of course, how many people here would volunteer to be in one of those experiments? Probably not, right? Don't want anyone sticking wires into our brain. So in order to do this, you either have to do this kind of research in animals, or you have to study people who are already going to have their brain stimulated for some other reason. And as it turns out, there's a number of clinical syndromes that brain stimulation is useful for trying to understand their pathology. We'll talk more about that later. Yes, so you can actually do that intracranially with a wire, right? So you actually drill a small hole in the skull and lower a wire into a target region of the brain and turn on a little electric current. But as it turns out now, in the last 10 years or so, we've come up with techniques that allow us to stimulate the brain without making any holes in the head. In fact, I bet someone here has had this. I won't ask you. But this is now a frequently used technique in certain kinds of anxiety and depressive disorders. In fact, it's now recognized as one of the best and most effective treatments for people who don't respond to medication. You can actually stimulate the brain with a device called a TMS machine, and you can achieve something like what happens when you put a wire inside the brain. All right, another group of techniques that have become available in the last 20 years or so are techniques called functional imaging techniques. These techniques are basically what allowed birth of cognitive neuroscience. Right? That part of cognitive science is really interested in how the brain actually does the computations that are involved in cognition. Functional imaging has allowed us to go beyond the limitations of cognitive psychology. Right? Cognitive psychology, we can only quantify observable behavior because we can't actually see the brain working. Well, functional imaging actually gets around that problem. It actually allows us to look at the brain as it's working. A really, really huge breakthrough in modern cognitive science was the advent of structural imaging techniques that allow us to do this. And some of you have probably heard of these before. How many people here have heard of PET scanning before? And it's a pretty commonly used procedure these days. There's other ones. There's something called fMRI. And there's now more modern techniques that use basically light to give us a signal of what the brain is doing. And lastly, there are ways of recording the electrical output of the brain. And like functional imaging, it allows us to see exactly what the brain is doing in real time as the brain is functioning. These techniques actually are a bit older. In fact, the first successful recording of human electrical activity from the brain was done in 1935. A German physiologist by the name of Hans Berger recorded the first electroencephalogram, or EEG. But we wait about another 50 years before people actually start to use that experimentally in psychology to try to understand what the brain is doing. So, recording the electrical properties of the brain can be done on a number of different spatial scales. What do I mean by that? Well, you can record from single neurons, right? Single cells in the brain. To do that, you've got to do something like what we're talking about up here in the stimulation technique. You've got to put a wire down into the brain and get that wire really, really close to one of those 100 billion neurons, so that you can listen to that neuron functioning. It's a really powerful technique. We've learned a tremendous amount about how the brain works, particularly about how early sensory and perceptual systems work through this technique of listening to the activity of single neurons. That's probably occurred to one of you already, or many of you already, that listening to a single neuron, while it's really interesting, since we have 100 billion neurons, and all 100 billion of those neurons are working almost all of the time, listening to one isn't going to tell you much about lots of aspects of cognition, right? It's not going to tell you about something really complicated like problem solving. You're going to have to listen to hundreds, thousands, maybe even billions of neurons simultaneously if you're going to use this strategy to understand what it is that the brain is doing. We can't get anywhere near that. Right now, we can record from maybe as many as 500 neurons at once. Nowhere near the number we need, though, if we're going to understand cognition. So you can also record electrical potentials from electrodes placed right on the surface of the brain. Now, these electrodes don't record from single neurons, but record from probably a few hundred to a few thousand neurons. As you can imagine, we're starting to get a little bit closer now to higher level cognitive numbers of neurons here, right? Thousands, maybe tens of thousands of neurons might be involved in certain aspects of cognition. So listening at this level turns out to be probably more fruitful for certain aspects of cognition than listening to single cells, single units. The problem, though, is the same that we have with single units, is that nobody's going to volunteer for these studies, right? Because they involve actually being inside the head. And so you only see this kind of research going on in very, very restricted environments where a person is already undergoing surgery for some other reason. However, there are techniques that allow us to look at the electrical signals in the brain from outside. And as I mentioned, Hans Berger, that German guy, was the first to put electrodes on the scalp and record EEG. It's also possible to record something called MEG, which also tells us about the electrical activity going on inside the brain. And you can do it from outside without actually penetrating the skull. These are what we refer to as non-invasive techniques. Right? So there's a number of non-invasive techniques up here. MEG is one. fMRI and optical imaging are non-invasive. And structural imaging and information processing techniques are non-invasive. In other words, you don't have to compromise the nervous system to get this kind of data. These techniques, though, like stimulating intracranially, and even PET scanning, right? PET scanning is invasive as well because you have to inject people with a radioactive dye. So that's considered obviously invasive. And a balance of invasive and non-invasive techniques is obviously necessary if we're going to understand all of what is involved in cognition. All right, so this is where we're going to be going over the next lecture and a half. We're going to be talking about all of these techniques in detail. But what I'd like to do now is just get us started talking about the first one, information processing, because it is so key. This is really the fundamental paradigm, if you will. By paradigm, I mean the approach that people studying cognition use, the way they do their science. All of those other techniques I told you about, almost all of them involve using the information processing paradigm in order to do experiments. So what is this? Well, the first thing to note is that the basic underlying assumption here is that the mind is like a computer and that its primary purpose is to compute. So the assumption here is that what our brains are doing, what those 100 billion neurons are doing, is they're doing computations. They're solving problems. And if you know anything about what neurons are doing, you know that one of the things they're doing is they're firing something called an action potential. Right? They fire basically a signal. A signal starts in the cell body of a neuron and it travels all the way to the end of the axon, and that signal is then picked up by the next neuron, which in turn fires a signal. That's a kind of computation. Right? One neuron sending information to the next neuron. Fire a signal, cause the next neuron to fire, cause the next neuron to fire, and so on. That's the kind of computation we're talking about. This is processing of information. It's, in other words, one neuron decides to send a signal to another one. It either decides to send a signal or decides not to send one. Right? At any point in time, one neuron will either fire and cause the next neuron to fire, or it won't. Another basic insight from information processing is that when information is processed in the brain or in any information processing device, it's not actually all done at once. It's not the case that they're presented with a stimulus and then boom, all the neurons fire. No, what actually happens is that information is processed in steps or stages, as they're called. So in other words, there's an initial set of neurons that are active that are doing some kind of computation. And those neurons send information to the next group of neurons, which do some other computation, and so on. If we think back to something like face recognition, the first neurons register that there's just an object there, there's something there. The next set of neurons perhaps recognize the outline or the shape of the face. The next set of neurons perhaps start recognizing the individual elements of the face, like the nose and the eyes and the ears, and so on. And all of this, right, each of these steps is happening basically one after another. There are a sequence of steps. And yes, each step or stage is a separate set of computations. Right? One set of computations is figuring out the shape of the face. Another set is recognizing noses. Another set is recognizing the hairline, and so on. Another observation is that processing goes from lower levels of computation to higher levels. And again, I think I talked about this a little bit last time. Lower levels, for example, might be, just finding all of the little line segments that make up the face. And the next level might be recognizing the shape of the face. And the next one might be recognizing the features of the face. Right? This is what we mean by lower level to higher level. The highest level in face recognition would be the point when you go, aha, I recognize that face. That's the highest, or if you will, the output of the face recognition system. This is why we say that information is processed hierarchically. There's a hierarchy of processes, starting with simple processes and going through more advanced and perhaps more complicated processes the further you get. And perhaps the most important of all insights is that all of this takes time. Like I said, it's not the case that the face strikes your retina and then all of a sudden, boom, you recognize the face all at once. That's not how it happens. It happens in a series of steps, and each of those steps takes a finite amount of time. So that in fact, it takes your brain something like a third of a second to recognize a face. When a face first pops on the screen of your computer, it takes about a third of a second before you actually recognize that face. And the sequence of steps or stages that basically intercede between when the face first came on and when you recognize it, that's what we're talking about here. Each of these steps or stages takes some amount of time to occur. That turns out to be really key to understand information processing. that each stage takes a finite amount of time. And here's the basis of this approach, or at least historically the basis, is that measuring processing time can give us an estimate of processing duration. If we could somehow get a handle on how long it takes each of these steps to occur, we would start to be able to understand the individual steps themselves. So here's the basis of information processing theory, is that you manipulate variables in an experiment, right? So you change something about the stimulus that you present to subjects. And these changes influence the number of stages or perhaps how long each stage takes. And from that, we can infer what those stages are and what they're doing. What's key here? Well, obviously, the inference process is important, but also manipulating variables and measuring, very carefully measuring time, turns out to be really important. And the first guy to actually do this was someone working at the same time as Munt, a guy by the name of F.C. Donders. He was actually a Dutch ophthalmologist. And he kind of toiled away in obscurity. Nobody knew he was doing this work. This is basically his hobby. The discoverer of information processing, the information processing approach, was actually doing cognitive psychology in his basement, pretty much. And he wrote an obscure group of papers, scientific papers that got published in some Dutch journals. And people didn't rediscover this guy until modern cognitive psychology started to take off in the 1960s. Someone found his paper and said, oh my God, this guy figured this out, almost 100 years ago. So yes, this is the father, if you will, of modern information processing theory. And what he devised was what we call in modern days reaction time, and what he referred to as the method of subtraction. So how does that work? We've got a few minutes left. I'll give you just kind of a brief overview of Donders' approach. So what Donders did was he did all of these experiments, right, in the late 1800s, way before computers, way before accurate methods of measuring the time it takes to do something. Right? There were only mechanical watches back then. And he came up with some really elaborate homemade devices for measuring how fast people could make responses. Usually how fast people could press a button, a telegraph key. I remember this is in the day of the first days of the telegraph, where there was that little key where you type Morse code out. But he would take those telegraph keys, hook them up to some elaborate mechanical timers that he had come up with, and he basically was able to measure how fast people could respond to a stimulus at the level close to millisecond accuracy. What does that mean? Basically, he was accurate within 1,000th of a second in measuring people's response times, pressing a button. So what did he do? Well, he did really simple experiments like this. He would turn a red light on in front of someone, and he would tell the person, as soon as you see that red light come on, press the button as fast as you can. Now, how long do you think that takes to do? You want to guess? How long does it take to make this kind of really simple response? No? Well, if you practice a lot, and he had people practice for thousands of trials, right? Just one after another, turning that red light on and measuring people's reaction time. What you find is it takes roughly 270 milliseconds for the average person. So basically 0.27 seconds from the time the light comes on to the time people press a button. Okay? So what does that mean? Well, Donders imagined, right, so here's the stimulus, and then here's the person's response 170 milliseconds later. He imagined that this is kind of the average amount of time it takes for the brain to register the stimulus and to execute what he called the motor response. Motor is meaning the motor system is the system that controls your muscles. So he imagined that what that must involve is, first of all, something he called stimulus detection. You obviously have to detect when the stimulus comes on the screen. Otherwise, you're going to press too soon or too late or just randomly. So there has to be some kind of stimulus detection process involved. And you imagine you have some kind of motor response organization process. So at least two processes you imagine. One that detects the stimulus and one that results in the pressing of the button. And he got a really accurate measure of how long that took on average across lots and lots of different participants in experiments. Now what he did was, he did one of those changes to his experiment, right? A very small change to his experiment. He's manipulating a variable. And he's going to look for a change in reaction time as the result of this manipulation of a variable. So what did he do? Well, one of the experiments he did was, again, involved presenting the red square and having a person press a button to that red square. But in this particular version of the task, equally likely on each trial of the experiment was the fact that the square could be red or it could be green. And if it was green, what you were told to do was to press a different button. So you're sitting there with two telegraph keys, and randomly, or at least randomly from the subject's perspective, there's a green light or a red light coming on. You're having to press one button if it's green and another button if it's red. Now this is a more complicated task, right? Donders reasoned that at the very least, this task involves another step or stage, what he called stimulus identification. In other words, in order to know which button to press, you have to identify whether you're seeing the red square or the green square. So before you can pick the right button, before you can response organize, you've got to do this particular process here. And what he observed is that in this task, it took people on average, now these again, well-practiced subjects, thousands of trials before he actually started collecting data, is that it took people about 30 or 40 milliseconds longer to make choice responses like this, one button versus another, compared to the simple reaction time experiment when they only had to press one button. And now you can see probably what Donders' insight is, right? That additional 30 to 40 milliseconds must be how long it takes for this process to execute. Right? In order to engage and execute this process required an additional 30 to 40 milliseconds. So Donders was able to come up with, if you will, an estimate of the amount of time of this additional process in his experiment. And I'll tell you that this basic approach is what underlies all of the experimentation going on in cognitive psychology today, and a good amount of other experimental disciplines as well. People in social psychology, as I said, personality, clinical psychology, anyone that's measuring people's cognitive abilities, or their social cognitive abilities, their emotional cognitive abilities, is using Donders' original insights about information processing when they do their experiments. So this is a really fundamental, this guy deserves a Nobel Prize, right? But very posthumously, 150 years ago, he made an insight that allows most of modern neuroscience and cognitive neuroscience and cognitive psychology to actually exist. All right, this is probably a good place to stop. And we'll pick up here. All right. So last time, I was giving you some historical introduction to cognitive psychology. And we ended up here talking about the information processing paradigm. And I told you that this, basically the discovery of this approach to doing experimental psychology was made originally by this guy. That's the Donders. Ophthalmologist working kind of in obscurity all by himself in Holland. It was only really in the 1960s and 70s that cognitive psychology, early cognitive psychologists in America kind of rediscovered this guy. And basically, what he had done was pave the way, if you will, for modern cognitive psychology, in particular, for implementing this approach to studying cognition, using information processing approach. Which basically means, basically implies a number of different assumptions here. The first is the idea that information is processed in a sequence of steps and stages. So in other words, when we process information, we don't just process it into it all at once. The brain isn't although the brain is, we'll talk about here, a massively parallel processor of information. It does a lot of things at once. It nevertheless still takes time will happen in the time range of, let's say, less than a second up to a few seconds. And so many of the very important things that our cognitive abilities allow us to do, for example, comprehend language. Right? As I say the words that I'm saying, your brain is decoding each of the sounds that make up the words and the words that make up sentences. And much of that is happening on a time scale that is in the sub-second range. In fact, each word that I'm saying probably takes less than a second for me to utter it, and certainly less than a second for your mind to figure out what that word is that I'm saying. So that is the basic idea here of information processing theory, is that to do something like recognize a word or recognize a face, as I used this analogy last time, an example last time, is actually composed of a number of different substeps or stages. Right? So when you recognize the word that I'm saying, again, you don't do that just all of a sudden. It happens gradually over a period of a few tenths of a second. And so that after about roughly half a second or so, you'll have recognized each of the words that I'm saying. The point is that it takes time. Right? It's a small amount of time, it's half a second, but nevertheless it takes time. Time with the sound waves which you hear, next week, to the time that the signal is transformed into an electrical signal that the brain uses, and then makes its way to the language centers of the brain where comprehension occurs. All of that takes about half a second. And each of those things that I just mentioned is a step or stage, if you will, along the way to recognizing words. That's going to be true for virtually everything we talk about this semester. Almost all of the basic cognitive abilities we have take time to be executed, if you will, to actually work. Even though our impression is right, that it kind of happens all at once. When you hear me say a word, it doesn't seem to you probably like it's taking time for that to occur. And that's because the time we're talking about is still very, very short. Very, very short compared to, for example, the time scale of many other things we work on around us. So it gives us this kind of false impression that there really is this kind of all or nothing kind of phenomenon going on. You go from a state of not knowing what the word is to all of a sudden knowing where it is. Well, trust me for now, but over the course of the semester, we'll be showing that in fact, that's not what happens. What happens is more like what's proposed here. There's a series of smaller steps or stages involved in breaking down the sounds that combine together to make the words that you're understanding, and then eventually to access the meaning for those words. Right? So that's obviously the bottom line in language, is accessing the meaning of the words that I'm saying to you, and putting those meanings together into larger coherent messages. All of that is happening in a series of steps. And Donners had figured that out, right? He figured out this must be the case, right? That it can't be the case that cognition happens all of a sudden. It has to be something that's happening over time. And he demonstrated that in his reaction time experiment. The fact that people take a finite amount of time, a short amount of time, but still a finite amount of time to press buttons, even in really simple tasks, like, for example, detecting a red square. So he figured out that, yes, processing of the mind is taking time. And that you might be able to use that particular element, if you will, of cognition to study what's going on in the mind as people process information. So that was Donners' insight, and it's an insight that turned out to be extremely valuable, and one that we continue to use today in studying how the mind actually gives rise to cognition. All right, so let's just quickly go back over the Donners' notions here. I know this is probably a bit complicated first time you hear it. So remember, Donners did this simple reaction time experiment. By simple, we mean this is as easy of a reaction experiment as he could come up with, where you present a participant with a stimulus of some type, simple stimulus like a red square, and you tell the participant to press a button as quickly as they can as soon as they see the red square illuminate on the screen. Okay, obviously, in Donners' case, it wasn't a screen, they were just talking as a red light bulb. turns on, press the button. And what he found was that people could make that kind of response on average about 270 milliseconds, or about 0.27 seconds. And these are subjects, again, that are well practiced. So these are subjects that have done this for hundreds or even thousands of trials. The trial being the presentation of a stimulus and a response to the participant makes. We hear that terminology used a lot this semester. The measure of a trial is usually some kind of single stimulus followed by some kind of response from the participant. And usually in experiments like this, you don't have a person just do a single trial to figure out how long it takes for them to respond. You instead have them redo that experiment, or redo that kind of trial multiple times. So in fact, in Donner's experiment, people did this pressing to a red light thousands of times. And what he does then is he averages together all of those thousands of responses and average or mean reaction time, response time. So that's a pretty standard procedure in modern cognitive psychology as well. So we have exposed subjects to many, many trials, and we then take an average of the people's response, of the person's response, in order to get an estimate of how much time it takes to do a particular task. So why do we do that? Why do we use lots of trials? Take a guess. Why isn't a single trial good enough? Yes?
Make it accurate, so it's not just like a random occurrence.
Exactly, yeah. So any given one that gives us a single response from someone, maybe they weren't paying as close attention. Or if it's the very first time they do it, maybe they didn't fully understand what it was they were supposed to do. So by getting many, many trials and averaging them together, right, calculating the mean, you get a more stable measure of a person's response time. And by doing that with many, many participants, averaging together many, many participants in the experiment, you can get an idea of what the population in general looks like in a particular kind of behavior. So these are the standard psychological, experimental psychology approaches to studying any kind of behavior. Donners is just now applying it in this information processing paradigm to get an idea of how long the various mental processes involved in doing this task take. So this was the first experiment he did. He did this on a number of people. He had them respond to many, many trials. And what he found was this is what the average reaction time looked like for lots and lots of observations. About 270 milliseconds is what he found. So what he imagined was, of course, this is kind of the introspective part of cognitive psychology. He tried to figure out what's going on in a task like this. If I present a person with a stimulus and I have them respond to it, what must be going on mentally in order to accomplish this task? In fact, this would have been the same question that an introspective psychologist, like Bush, would have asked. Of course, what Bush would have done was he would have asked 3 or 4 other introspective psychologists what they think is going on, and then they would compare their thoughts and their ideas and come up with perhaps something similar to what Donners did. But they wouldn't have the kind of data, right? The observable, quantifiable behavior that Donners did. So Donners imagines that, well, at the very least, in order to respond to a simple stimulus like this, what must be going on in the mind is there must be some kind of process somewhere in the mind that allows you to detect the stimulus. Otherwise, if you don't detect it, you're not going to press the button in response to that particular stimulus. So you have to have some kind of stimulus detection process going on. What else must you have? Well, because people are pressing the button, he realized, he hypothesized that there must also be some kind of response system, right, that allows you to actually execute that button pressing behavior. So if you have to have at least these 2 processes according to Donners in order to do a simple reaction time experiment. Now there may be more processes involved, right? We are saying here, and Donners certainly would say that there could be more things involved, but this is the very least you have to have. And what he imagined was that you're looking at, you look at the reaction times, you're looking at basically the stimulus feeding into some kind of stimulus detection process, which then feeds into a response organization process, which then allows the parts of the brain to control the finger pressing to be executed. Right? And that, we can estimate, takes about 270 milliseconds. So the beauty of Dogger's experiment, of course, is that, and like all modern cognitive psychologists now, you don't just do a single observation like this, or lots of observations on many, many trials and many people. What you have to do is you have to contrast that kind of response to some other experimental condition that differs, hopefully, only in a single dimension. Only a single thing differs from one condition to the other. That's a really important concept in science, right? So when you're comparing two things to see if they're different or not, is that you hopefully control things enough so that the actual differences between those two conditions can be boiled down to a single factor, to a single thing that we could causing the difference. In Donner's case, what he said, what he imagined was, well, what we're going to do is we're going to compare pressing a button to a red light to pressing a button to a green light. When the participants are told, you don't know from trial to trial which kind of stimulus is going to be presented. And you have to wait until you see which kind before you press. So in other words, if a red stimulus comes on, you press the right button. If the green stimulus comes on, you push the left button. And again, you do this for hundreds of thousands of trials and many, many subjects, and you measure the people's reaction times. And what Donders found was that in fact, people were slower in the choice reaction time test. It took them longer to do this test. I'm not terribly surprised it would take longer. But in fact, it wasn't a whole lot longer. It was only about 40 milliseconds, or about 0.04 seconds, which is not very long, but it is a quantifiable, measurable amount of time. You can do statistical analyses on this and show that these are really, really different kinds of responses based on this very small difference in reaction. So the important thing, of course, is what Donders infers from this difference. What he infers is that what you're having in the choice reaction time task is the addition of an additional process. Right? In order to do choice reaction, you need more than stimulus detection and response organization, right? Because you have to be able to discriminate between two different kinds of stimulus. And he imagines that process must sit in between the process that's involved in detecting a stimulus and the process that's involved in organizing a response. In other words, choosing which button to push. And what he's going to propose here is that the duration of this additional process right here must be about 40 milliseconds. And so this is the logic of information processing, is that you use small changes in the experiments, in the experimental conditions, and observe some type of measurable behavior in those different conditions and compare them to see if they differ. And if you find a difference, and you can find a quantifiable difference, you can use that difference as an estimate, if you will, of how long this additional process is actually taking. So this is the logic of Donders. He referred to this mechanism here as something called pure insertion. In other words, he imagined that our cognitive apparatus is flexible enough that we can actually, depending on the demands of the task we have to do, so we can actually insert additional processes into our streams of information processing. And that if that's the case, you can then imagine methods like this that can allow you to estimate how long those inserted processes actually take. And this general approach to studying the duration of mental processes is now sometimes referred to as mental chronometry, an explanation of chronometry here, having something to do with measurements of time. Well, this approach has been now used successfully for over 50 years in cognitive psychology, and thousands of experiments, thousands of small differences between conditions allow us to try to figure out what the durations and differences are of processes involved in a whole host of different kinds of experiments, everything from word recognition, face recognition, to things like problem solving and high-level decision making. This general mental chronometric approach has been very, very successful. So yes, modern cognitive psychologists have used this information processing paradigm, originally used by doctors, originally discovered by doctors, and they continue to use measures like reaction time to quantify the duration of the mental process. However, they don't only use reaction time. Certainly, while reaction time might be the dominant measure used by cognitive psychologists, what do we call a measure like reaction time in statistics? What kind of variable is the variable you're measuring? What do you call it? And that's that. Yes, this is the dependent variable. So in mental chronometry experiments, reaction time is the so-called dependent variable. That's the one you're measuring. Just remember that the actual variable that you're measuring in an experiment is the dependent variable. And that's the one you expect to change as a result of some experimental manipulation. For example, the nature of the task, the dominance manipulation. A simple reaction time or choice reaction time. That's the independent variable, the type of task that people have to do. An independent variable is the variable that the experimenter manipulates in order to see if there's a difference in the dependent variable. And just a quick reminder for those of you who have stats and for those of you who haven't, this is a common terminology that we'll be referring to frequently throughout the semester. Practice is the basis of the scientific method. Right? It's looking for differences in dependent variables as a function of manipulating independent variables. All right. So yes, a variety of other dependent variables that are used. Reaction time is one example. It's probably the best example for measuring things like mental chronometry. But we can be interested in many other phenomena that don't necessarily require us to measure the amount of time taken to do something. So, for example, probably used equally often, and in some areas even more often, are the patterns of errors people make in experiments like this. So think back to the Donders experiment. Probably, you're not going to make too many errors in a simple reaction time task, right? There's not much you can do that's different from trial to trial. I suppose you could miss a stimulus, right? You could blink or something when one of those red lights come on and you might not press the button. And that would be an error, right? You missed it. But in choice reaction time, what kind of errors can you make? Right? If there's a red and a green light and two different buttons, what could you do wrong in that experiment? Press the wrong button, right? You see the red light and accidentally press the button for the green one, or vice versa. And of course, in addition, you could also miss a trial, right? You just fail to respond. Or you could press one button real quickly and then the other, which happens very frequently in these kinds of experiments, because people tend to correct their errors in many kinds of different kinds of experiments. And so the patterns of errors people make actually turn out to be very revealing in many experiments that cognitive psychologists do. So for example, if you have people do the choice reaction time experiment, press the left button for the green light, the right button for the red light, and then halfway through the experiment you switch it. So that now you have to press the button on the other side for the red light and on the other side for the green light. You can imagine at the point when the task switches, that the error rates go up, right? People are going to, because they develop this pattern of responding, and now you're asking them to do something different, their errors go up. And you can use that change in the pattern of errors that subjects make to make some hypotheses, perhaps, and to test some hypotheses about, what different factors perhaps are impacting competition for responses. So errors like this can be very revealing, as can patterns of correct responses, right? So the opposite of an error response is a correct response. And when people tend to be correct versus when they tend to make errors can be very revealing about the underlying cognitive mechanisms involved. So yes, it's also possible to combine accuracy measures like errors and correct responses along with reaction time. You can put those together to ask even more complex questions. So these are the basic tool bag that many experiments we'll talk about will use this, we'll talk about this method that we use in order to try to figure out what's going on in the mind. There are other techniques as well, and I'm going to tell you about one now because this is a pretty good segue into the next lectures we'll have next week when we start talking about perception. So there's actually a sub-branch, if you will, of cognitive psychology that's called psychophysics. What is psychophysics? Well, a definition of it might be the quantification of the relationship between some kind of stimulus and the sensations and perceptions that they generate. Sounds a lot like cognitive psychology, right? Stimuli resulting in some type of sensory and perceptual and cognitive change. The interesting thing about psychophysics, though, is it really is focused in on perception and is not generally as interested in later and higher level kinds of tasks. And moreover, the term is also sometimes used actually to refer to the methods that can be applied in studying sensory and perceptual processes in the brain. Now, what do I mean by sensory and perceptual processes? For those of you who have not had a course to talk, differentiated sensation and perception from cognition. When we talk about sensation, what we're talking about is basically the initial signals that arise in the various sense organs of the body when a stimulus is presented. So in the case of vision, right, when the square in the Donner's experiment was illuminated, the eyes, of course, take the photons of light coming from a light stimulus, bounce off the retina, and those signals are changed into electrical signal that then moves into the visual parts of the brain. That's what we mean by sensation. Basically, it's the activation of the sensory apparatus and the early parts of the information processing system that basically registers the stimulus. Okay, that's sensation. Perception, of course, then is what you do with that sensory information. Right? The fact that at some point you recognize that it's a square you're looking at. And of course that happens after the sensory experience. Alright, so what do psychophysicists actually do? Well, the one thing that they're generally interested in, I would say the one thing that kind of differentiates psychophysics from the rest of cognitive psychology is that psychophysicists are interested in so-called thresholds. Now, what's a threshold? An idea of what this term refers to here? The most convenient. So actually there are two possibilities, right? It could be a threshold could be at the very high end, so like the loudest kind of figure. But I think you were implying the quietest kind of. So, a threshold is basically the weakest stimulus that you can perceive. Okay, and psychophysicists generally are interested in that. They ask questions about, they're interested in sensory and perceptual processing, and they're particularly interested in the kind of limits, the boundaries, if you will. The threshold is an assessment of the intensity of the lowest or weakest stimulus that can be detected by an observer. Why is that interesting? Why is it interesting to probe the boundaries of perception, the low end boundaries? Yes. So a human performance standpoint. So human performance is kind of a branch of cognitive psychology that's very interested in knowing like what can humans tolerate, right? So at the high end in particular, right? How many people can a person tolerate? Also perhaps at the low end, what are the weakest signals that a person can detect? Like someone that's operating a radar or a sonar, trying to detect an enemy submarine. What's the weakest signal on a sonar scope that a person can actually detect? So yeah, there are lots of practical applications in the military and aerospace industry for this. Yeah, so how bright are these street lights, right? So how much, we want to limit the amount of electricity we use with street lights. So how dim can we make a street light and still have it give off enough light that people can, for example, walk without tripping, see the things that they need to see. So that's what we'll do. Sleep? Yeah, sure. Yeah, and we'll have a lot to talk about sleep later when we talk about numbers. But yes, certainly things like how much sleep do you need would be an example of the question here.
Like a pain threshold, like this is
Yeah.
how much pain you can feel, so your body goes like shock or pain.
Yeah, so you probably all heard this before, your pain threshold. Different people, you know, in common everyday life talk about, you know, low or high pain thresholds. When does something get intolerable? But there's another case about that. All of you have been involved in a threshold experiment at some point in your life. It's used almost universally in our society. What am I talking about? Vision for sure, right? We've all had a vision test. And in a vision test, right, they have you look at those E's that are in different directions, and they also use different letters, and they make them as small as possible, right? Adjusting the lowest threshold for being able to recognize the letter. What else is it? Hearing test, right? The hearing test you probably had done in school when you were first or second grade, probably. You were to take your hearing test tested. Right? Probably around the same time as the school also tested your vision. And of course, the reason is that children frequently don't report that they have problems with their two most important modalities, vision and audition. And you need to test them to make sure. I mean, I know my son, for example, we had no clue, but he needed, you know, almost Coke bottle lens glasses to see it. He was sitting in the back of the class and not sure he needed material. It was only once he had a vision test in school that they said, hey, this kid needs glasses. So this is something that is really important. And audition perhaps even is important, right? Because if you can't hear what your teacher is saying, obviously you're going to have problems with it as well. Yes. Yeah, so this is before coming off. People with diabetes, people with background or something with diabetes. And yet, one of the first really bad symptoms that can happen is the lack of sensation, particularly in the extremities, the feet and the hands. So yes, this has many practical everyday applications. And I think audiology is probably the one that you have heard about the most, and perhaps ophthalmology as well for vision. But in audiology, this is done. In fact, this is where I give a plug for our sister department in the speech language and hearing science department, where audiology is taught actually here. And this is a test being done on a young child here, and he's wearing headphones, and he's being presented with what? Very weak tones, pure tones, or beats and boots, if you will. And they're testing to see just how quiet of a tone can the child hear in each ear. And they graph that. Left ear is the blue X's here, and right ear is the red circles. And what you're looking at here is the threshold of this particular child for different frequencies that they've been listening to. Now, what is this about? What's the frequency part of this chart telling us? What does frequency refer to when we talk about sound? Yes, so how high are those pitches? And why would that be important if you're testing someone's hearing? And after all, shouldn't just any old pitch work? So you need to know the range, and there's a particular reason for the range. Particularly if you look at this range here, basically they're mostly testing between about 125 cycles per second and up to 8,000. And I'll tell you that actually the human ear is capable of all going all the way up to 20,000 cycles per second, 20,000 hertz, right? Those really high pitch sounds of music, some instruments make go all the way up to 20,000 hertz. So why would we stop at 8,000? And we can also go all the way down to about 20 hertz, really low pitch sounds. We can hear all the way down to about 20 hertz. So why this range? It is. So this is the range frequency is higher or lower. But usually certainly what we're worried about in school is, can you hear what your teacher is saying? And if you have a deficit, right, in other words, a problem with certain frequencies in this range, it's going to be really problematic. So this is where most audiology testing is being done. And here's a person with a pretty severe deficit in both ears. Right? So here's kind of the normal range of hearing here. So again, someone with intact hearing, these lines would go straight across here, across all the frequencies. So what you're looking at is each of these little marks here is a different frequency tone that was tested. So the audiologist will present a whole bunch of tones, different frequencies varying back and forth, and different levels of quietness. So different what we call amplitude. Really low amplitude would be a quiet sound, a high amplitude would be a loud sound. So they're testing lots of different amplitudes, lots of different frequencies, and when they're all done, they look at the child's performance. Oh, by the way, I didn't even tell you the task. Of course, the task here is that whenever you hear a sound, you raise your hand. And you raise your left hand, it's the left ear, the right hand, it's the right ear, and you test both ears simultaneously. This test takes someone 20 minutes, 30 minutes. And at the end, when you plot all of the kids' responses, you get a graph that looks like this. And this one is suggesting that this particular child has a pretty severe hearing loss in the range of roughly from 2,000 to 8,000 hertz, which could be a pretty severe impairment for being able to recognize particularly female voices, right, which tend to be slightly higher pitched than male voices. Something like this.
Are thresholds in relation to these being constant questions asking whether or not takes information to see the fire response to the measure?
Yeah, that's roughly what we're talking about here. So there's a sequence of neurons in the brain, right? Early neurons in the auditory parts of the brain, in the case of a test like this, they're the first ones to receive information coming from the ears, like the signal from the ears. And there'll be neurons that will respond to it. And some neurons respond to low-pitched sound, and some respond to high-pitched sound. And so that's basically what we're talking about here. And of course, those neurons send the information on to higher level centers, right? The ones that allow you to raise your hand. Oh, I heard that. All right, so everyone kind of clear on how this works? All right. So yeah, stimuli below the threshold. Right, so obviously the notion of a threshold suggests that there's some kind of barrier, right? That when stimuli are below that barrier, you don't hear them. And by the way, those are called subliminal stimuli. So they would be below the lemma. Lemma just means the threshold. Stimuli that are stronger than that are higher amplitude, auditory stimuli, are going to be detected. Interestingly, and I'm not going to get into this in any detail, thresholds are actually kind of a fictitious concept. They don't actually exist in the human brain, or probably any brain. More likely, we have something like a threshold, but it's constantly varying. Right? So when you get down to really weak stimuli, sometimes a really weak stimulus you might detect it, but that very same stimulus presented a few minutes later, you might miss it. And that's because we're not machines in the sense of being perfect measures of what's coming at us. Everything is kind of superimposed on other things going on in the brain, what we refer to as noise. The brain is kind of a noisy place. And at some point in time, the audiologist presents you with very, very faint auditory stimulus, and your brain is generating a little more noise. At that point in time, you might not hear that stimulus, and you might not raise your hand. But if at other points in time, as that noise kind of waxes and wanes, goes up and down, we have to hit a really low point of noise in the nervous system. That same stimulus might be heard, and you might report this. That was a stimulus. And so you can see what I mean, this notion of a fixed absolute threshold, that you always hear something if the stimulus is right above it, and you never hear it if it's right below it. That actually isn't really what happens in the brain. In fact, if it was, you could do one of these audiometric exams in just a few minutes, right? You only have to present one time with each frequency in both ears at two different amplitudes. An amplitude above threshold and one below, and you have it. And that turns out not to really work. You got to present lots and lots of tones of above and below the approximate range of the threshold in order to figure this out. All right. So that finishes cycle physics. You can actually teach an entire course on cycle physics. It's full view here, but we need to move on now. So really where we are now is we're now going to start moving into the lecture on cognitive neuroscience. So the way I'm going to talk about, we're going to talk about cognitive neuroscience all semester, part of our discussion of cognition. But the chapter that I assigned you on cognitive neuroscience and what I'm going to talk to you about now, I'm mostly going to focus on the methods that cognitive neuroscientists use in order to understand the neural underpinnings, the neurological underpinnings of cognition. I'm going to do that because, in fact, as I pointed out last time, was that the methods turn out to be really important. In fact, before we had many of these modern cognitive neuroscience methods, the only kind of cognitive psychology you could do was the kind that involved reaction time errors. It's really only in the last 20 years or so that cognitive neuroscience has allowed us to come online with many more techniques that allow us to look at what the brain is doing while we're performing cognition. It's allowed us to get insights into actually how does this work? How do these things that Donders and other cognitive psychologists have discovered through behavior, through reaction time, how do they actually work in the brain? And understanding the methods that are used to do that tells you a lot about how that understanding has unfolded in the last 20 years. With the exception of this first technique. The first technique here, the technique of brain lesions, what I'll call classic neuropsychology. That is actually almost as old as cognitive psychology itself. Neurologists back in the late 1800s, at the same time that Wundt and James and all these guys were first coming up with the ideas of cognitive psychology. So neurologists observed that people who had had some kind of head injury, whether it be some type of overt injury, an accident falling off a horse, I guess, probably in the late 1800s, one of the motorcycles yet, or perhaps someone who had been in war and had been shot with a projectile of some type through the head. Or more common, actually, is something called cerebrovascular accident. That's another word for that. Yeah, stroke. Right? A stroke is basically what happens when a blood vessel in the brain is either blocked by a clot, or in some cases, the blood vessel can actually rupture, like blood flow stop in that part of the brain. And I will tell you that the brain, more than any other organ in the body, needs a rich and continuous flow of blood to work. If you interrupt the flow of blood to the brain for even a few seconds, you can start to see changes in what the brain is doing. Right? In fact, if you choke someone and they pass out, why do they pass out? It's because you cut off the blood supply to the brain, the blood that's necessary to maintain consciousness. Right? You cut off that, basically that component that makes the brain able to function. So if you cut off that flow internally, right, by one of these cerebrovascular accidents, like stroke, it causes damage to the brain, and some of that damage can actually be permanent if the stroke was large enough. The brain tissue will actually die. And as a result of that, I think the logic is pretty simple here, right? If you kill a part of the brain, then that part of the brain is no longer going to function in cognition. Right? So, one strategy for trying to understand how cognition works in the brain is to study people who have had strokes or have had some kind of damage to their brain. And in general, the smaller the damage, the more, as we say, focal the damage. In other words, the more restricted it is to a small region, the more helpful it is to study someone like that to try to figure out what it is that's gone wrong. In other words, what aspect of cognition, sensation, perception has been interrupted. If you have a massive stroke, right, you hear about massive strokes, this is where large parts of the brain have lost their blood supply, and that part of the brain actually dies very quickly. Then you get also massive changes in cognition. Many systems are impaired in this happens. Now, underlying assumption here, something we haven't talked about. If the size of the stroke matters, that implies that the sensory and perceptual and cognitive abilities that we have must be localized in particular areas of the brain. Okay? Otherwise, it wouldn't matter what size of the stroke was. I mean, you might have a small stroke might cause just a little bit of impairment, and a big stroke would cause a lot, and that's certainly true. But it's different than that. A small stroke usually only impacts one particular perceptual or cognitive ability. So if you have a stroke over the back of your head, on one side, let's say on the left side, it turns out that only vision is affected by that, and only vision for a certain part of the visual field that you're looking at. And that's because the visual parts of the brain are actually laid out, kind of like the environment. Right? So different parts of the visual system in the brain process different locations in the environment that you're looking at. So this is what we call localization of functions. Functions in the brain, like, for example, vision, are localized to particular places. That turns out to be really good news for those of us trying to figure out how cognition works in the brain. Right? If in fact the brain was kind of like what we refer to as mush, and everything that we do was processed in every part of the brain. Right? In other words, there was no differentiation between vision in the back, or vision over the sides of the brain, and so on. Then it would be really hard to figure out how the brain works. But as it turns out, many of the functions that we're going to be talking about seem to be localized to particular parts of the brain. And this is what's allowed the insight here. Cognitive neuroscience, the cognitive neurosciences to actually have an impact on our understanding of cognition. Is that by studying these different parts of the brain, we can actually study the different aspects of sensory, perceptual, and cognitive processing. So that's the kind of underlying assumption, again, another assumption here, but it's largely borne out, even in these old studies done by these neurologists back in the late 1800s. They were some of the first ones to figure out that if a person had a stroke in a particular part of their brain, what you would find is that only a particular behavior would be impacted. In fact, two early neurologists working in Europe were the first to make the observation with regard to language. So a famous French neurologist by the name of Paul Broca discovered a patient who, after having had a stroke, or apparently having had a stroke, had serious problems with producing language. This person could no longer really speak. He could utter some sounds, but he largely was mute. Interestingly, he was still able to comprehend language. He could demonstrate to you that he understood what you were talking about. Right? He said, put the red block on top of the green block, and then put both of them in a box. He could easily do that. Lots and lots of comprehension language tasks. But he wasn't able to produce language. When he tried to produce language, he was stammered and could really only say very few words. It was very hard for him to do it. Now, that's an interesting case. I mean, in and of itself, it doesn't tell you a whole lot. But around the same time, a German neurologist discovered a different patient who had also had a stroke and had a completely different language impairment. This patient could actually produce language. In fact, he spoke very readily and a lot. His problem, however, wasn't comprehending language. He couldn't understand what he was saying. So he told him to put the red block on top of the green block, put them in the box, and he might knock the box onto the floor. And he understood you were asking him to do something, but he didn't understand what it was you were asking him to do. These two patients, two of the first neuropsychological patients, started basically the beginning of neuropsychology. This is when scientists and neurologists and early psychologists realized they could study people with brain damage to try to understand where and what these different parts of the brain were doing. Now, of course, the problem that Broca and his colleague in Germany, Wernicke, had was that they didn't know for sure where the damage was. They didn't have any way of localizing in the people's brains that they were studying where the damage was. And of course, that's because the brain is sealed inside the skull and you can't see it. So they had to wait actually for their patients to pass away, to die. And then, fortunately, the patients' families gave their brains to science, and they were able to dissect the brains and figure out where the damage was. But this could be, right, in theory, years or decades later in these kinds of studies. So it makes the science of neuropsychology pretty slow in most cases. So it really wasn't until the 1960s and 70s that changed overnight. Overnight, neurologists were able to image the brain without taking it out of the skull and dissecting it. And this became possible because of a new set of techniques that became available called structural imaging techniques. The term structural here just means to indicate that these techniques allow you to take an image of what the brain looks like, the brain structure. And here's a really good example of some of these first studies that did this. So here's an intact brain. This is a postmortem brain that's been spliced. And here is a patient with a postmortem brain from a patient that's had a stroke. And you can see, probably even with an untrained eye, you can see that there's a massive hole here in this part of the brain here, in the parietal and extending into the temporal lobes. So of course the brain is occurring, and you don't need to know that part. But needless to say, this is a massive amount of damage. Probably would have had important linguistic impacts on this particular patient. Now this is, of course, exactly what Brooke and Bernie were doing, right? They were looking at postmortem brains to figure out where the damage was. However, in neuropsychology, modern neuropsychology, you can do this. You can try to infer brain function from brain damage using modern imaging techniques that allow us to take a picture of the intact brain. So yes, neuropsychologists do this. And usually the way they go about doing their science is that, first of all, they find someone that they think has had a stroke or some type of brain damage, and then they do a systematic series of experiments on the person, right, testing all kinds of abilities, language abilities, perceptual abilities, memory abilities, decision-making abilities, abilities to make judgments. All of these kinds of things will be tested systematically. So someone going in for neuropsychological testing like this probably will get 3 or 4 hours of many, many kinds of tests. Some of them will be reaction time tests, some will be error tests, some will be paper and pencil tests. And the neuropsychologist will get a profile, if you will, of which of a person's behaviors are intact, in other words, have not been affected, and which behaviors are impaired. And they can do that because they can look at a person's scores on a particular test and compare them to what are called the normative data for that test. So in most of these neuropsychological tests, the tests have been given to hundreds of thousands of quote, normal individuals. So you have a pretty good idea of what the range of responses in normality are. And when a person falls outside of that range on a test, for example, say a word recognition test. You have a person, you say 30 words to them, and you have them define each of those words. And the average response is that most people can define 20 of the 30 words, and you run into someone who can only define 5, then that might indicate that they have a language problem. Now, you can only say that if, in fact, when you tested their other abilities, like their visual, perceptual abilities, and their memory abilities, and all of these other things, if those were closer to normal. Right? If someone could have some type of general mental defect that causes them to be low on a whole bunch of tests. It's only when there's basically a pattern of deficits and a few things where everything else is within the normal range, you can then start to use this neuropsychological approach. So this is what they do behaviorally for subjects. So you get a profile of scores on this test. That's something about what the various brain areas normally do. How do we know about the brain areas? Well, we have to use these structural imaging techniques to figure that out. So in other words, a person's been tested with the neuropsychological test. Now they're going to go to see a neuroradiologist to have pictures of the brain. two kinds of pictures you can get. You can get the structural ones, which we're going to talk about first, and you can also, which we'll talk about later, ways of looking at brain function. There are brain tests that allow you to look at brain function as well. All right, so how does a neuroradiologist use these kinds of so-called medical imaging to get a picture of a brain? Well, there's a couple ways to do it. The oldest way, right, the way that started becoming popular in the 1960s and 70s is something called a CAT scan. How many people here have had a CAT scan? This is a really common technique, right? It's not just for heads and brains, it's for lots of parts of the body, right? If you've done something to your knee, if you have abdominal pain or whatever, a CAT scan is one of the first things that a physician will test. And that's because it's a fairly inexpensive procedure to use. It does carry some risk, though, because in order to get the image, you have to use X-rays. X-rays, of course, are high-energy rays that are shot through the body, right, to get an image of whatever organ you're looking at. And those X-rays carry some risk of causing damage to the chromosomes and the nuclei cells in the body, which can be cancer. There's some small incremental risk each time you get an X-ray of that happening. This is why you only get some of the X-rays in certain periods of time. So in a CAT scan, these are some examples of images from CAT scans. This is one brain. And what you can see here is there's a whole bunch of images of that brain. What you're looking at, basically, is one of the beauties of medical imaging like this. And that is that you can take images of the brain from lots of different angles and perspectives. So here what we're doing is you're looking down from the top of the person's head. You're looking literally down on top of their head. And this is the very top of the brain here. And as you go this direction, you're going deeper and deeper through the brain. So these are sections, if you will, through the brain as we go deeper and deeper and deeper. And eventually, you see, we come out through the bottom here, and you can start to see the eyes and the eye sockets and the nose and stuff. And of course, what you can also see is some anatomy of the brain here, right? You can see that there's white matter in here and gray matter, and then these openings here in the middle. These are the ventricles of the brain. These contain cerebrospinal fluid. They're like a cushioning and air conditioning system for the brain, if you will. This is a fairly normal brain. I don't see any evidence here. I'm not a neuroradiologist, but I don't see any evidence here of a stroke. So this would be someone with probably a relatively normal type of brain. And of course, you need normal brains when you're doing neuropsychology, because you need to know what things look like when there hasn't been a stroke. So this is a CAT scan. And this is just a really nice image showing you what you get with a CAT scan, right? Each of these is one of those sections we were looking at, and they're just going to pile up here in a big, nice even stack. And a neuroradiologist can go in and say, well, I'm going to look at this particular one right here. And basically, they can pull this out of their computer and look at that image. And then if they want, they can go to the next one, and so on. So you get a three-dimensional view, if you will, of the brain with this type of technique. You get a three-dimensional set of images that can be turned and twisted and looked at any way that you want. And here's an example of a great blow-up image, but you can still get the idea here. This is a CAT scan of someone who's had a stroke. And you can see, again, this large opening here. This is basically an area where there's a massive stroke, and over time, the cells, the neurons, and the other cells, the support cells of the brain, that would be in that region have basically become necrotic and withered away. And what happens is that those areas get filled in with cerebrospinal fluid. So it's like you just have a massively expanded ventricle in the brain when someone's had a stroke like this. And of course, someone with a stroke of this size would have lots and lots of deficits. This is what a CAT scan looks like. Those who've not been one before. So you lay on this bed here, and the bed is motorized. It can be moved in and out of this donut hole here. These are the actual X-ray scanners that are in this kind of brass-looking outer ring. So as you move the head or body through the scanner, you can take multiple images as you move the person through the scanner. So to get all of those three-dimensional image look, you first would move their head to the very top of the head. You take some CAT scan images, You move them in slightly more, you take more images, and so on. Until you come all the way down to the bottom of the brain. Right, so that's basically how the technique works in collecting the actual X-ray images. Here's kind of the physics of what's going on. In that brass outer ring that I showed you, what's actually in that ring are a whole bunch of what are called X-ray tubes. So there's actually a whole bunch of them that go all the way around that ring. And on the opposite side of each X-ray tube, there's an X-ray detector. Right? So again, there will be detectors all the way around. So what happens in a CAT scan is that you turn it on, and X-rays come from every which direction, and as they pass through the head and through the brain, they're picked up by detectors on the other side. And that detector sends a signal, a digital signal, to a computer that basically measures the density of tissue. Right? Basically what happens is the amount of X-ray that passes through and is picked up by the detector tells you something about the density of the tissue as the X-ray passes through. And by comparing that from all of the detectors around the brain, putting that into a computer algorithm, you can figure out exactly changes in the density of tissue down to really, really small differences. Right? So that's why white matter of the brain and gray matter of the brain have different densities. And that's what allows the computerized axial tomography, which is what CAT scan stands for, allows the image to be assembled on this type of information. All right, so a CAT scan is great, right? I mean, as I said, it's not very expensive, and it's easy to do. You can get a full CAT scan from someone's brain in probably 10 15 minutes maximum. And you get a pretty good image, pretty good first pass image of what the brain looks like if there's been fairly large scale damage to the brain. The problem with CAT scan is that its resolution, here, I mean its ability to see small details in the brain, is somewhat limited. X-rays tend to go straight through the brain, and their signal is only slightly changed by the densities of the tissues. And therefore, the image you see is kind of blurry, right? And those images we were looking at back here are a little blurry, they're grainy. Look like they were taken looking at a digital camera from the 1980s. High resolution, modern cell phone cameras, which can take much higher resolution images. So, CAT scanning, while it's still used quite often, is usually only the first line of investigation when you're trying to image what the brain looks like. What's used more often these days to get fine-grained images is a technique called MRI, or nuclear magnetic resonance imaging. We got rid of the nuclear when they started using this technique because people were afraid of this word, right? Nuclear magnetic. They're basically exposing you to high levels of radiation, and while it's true that you are exposing people to a kind of radiation, this is a safe kind of radiation. Certainly much safer than X-rays. You can get an MRI scan done every day for a year and not have any increased risk of cancer or anything like that. So MRI is magnetic resonance imaging. fMRI, we'll see a bit later, is functional magnetic resonance imaging. And the difference between these two here is that difference that I highlighted a few slides back when I told you about structural versus functional imaging. MRI is structural. It takes a picture of what the brain looks like, just like CAT scan. It gives you an image like a photograph, like what a camera does. fMRI tells you what the brain is actually doing. In other words, it gives you an image of its function. So, kind of the analogy would be, you've all seen those kind of night vision goggles, those infrared cameras, right? They take a picture that shows you what things are hotter, which things are cooler. That's more of a functional image, right? Because it tells you where is there a source of heat and where is there a source of coolness. Functional imaging tells you which parts of the brain are working harder and which parts are not working so hard at all. So you see the difference between functional and structural. Structural is just what the brain looks like. And in fact, the point that I made last time, your brain would not look much different than if you were to pass away and then take another MRI scan of your brain, let's say a half hour after you passed away. They would structurally look very, very similar. A functional scan of your brain would look completely different. Okay? Because in fact, after you're dead, your brain slowly stops functioning. And you would not see all of the big differences between the different parts of the brain. So basically, as a function of how hard they're working. All right. Yes? Yeah. It does. As we'll see, I'm going to explain how fMRI works, but it basically is a measure of blood flow in the brain. And it turns out blood flow is tightly correlated with cognition. Surprising. All right, so here's a magnetic resonance image, a structural MRI image of the brain. I think you can probably see how much more detail there is here. In fact, you might have been hard pressed to know that this was an image that was more sliced through the brain. In fact, they look really similar. To the untrained eye, it's hard to tell the difference. In fact, modern MRI scanners, the very, very most powerful ones, take images that really are almost indistinguishable from real tissue. So how does MRI work? It's pretty complicated. I'm not going to go into all the details, but this is what a machine looks like. Everyone before you know. It looks a little bit like a CAT scanner, and there's a big donut that goes around you, right? And there's a hole in the middle that you insert the body that they're trying to image into. One difference, though, is that usually in fMRI, there's also a secondary apparatus. In this case, it's this called a squirrel cage here. So this is basically a plastic device that's got, looks like bars on it. And that's part of the scanner as well. So it works the same way, though. You use a motorized bed here to basically move the person into the scanner, and then the image is taken. The only difference is you don't have to keep moving them the way you do with CAT scans. So there's not a single circle of detectors. In fact, the detector is actually this entire cage that surrounds the head. So once you're in the scanner, you're going to probably be stationary. So this is kind of the schematic of what's going on. This big outer area of the MRI scanner is actually a gigantic electromagnet. A magnet is so strong that it's like those magnets in the junkyard, right? You pick up a whole car. This magnet's that strong. In fact, one of the big dangers in MRI research, MRI technicians, one of the things they have to do, they have a safety course they have to go through before they can actually work in an MRI scanner, is because you never ever want to bring anything into the scanner room that is basically magnetically susceptible. Right? So things like ballpoint pens that have steel on them or nickel in them, paper clips can be lethal. You walk into a scanner room with a piece of paper and a paper clip on it, and the scanner is all the way across the room, it'll rip it off the sheet of paper and it'll fly at the scanner at a high rate of speed, and anything in between the scanner and the paper clip is going to have a lot of, the person is going to experience a lot of pain. In fact, there's some great training videos they show where they take like a big metal bucket and they put it by the MRI machine, and someone has a big monkey wrench that they let go, and the wrench flies across the room and smashes the bucket flat. So that's how powerful this magnetic field is, and why it's so dangerous to not, why it's important not to let anyone into the room that's got any kind of metal, loose metal on their body. Now that said, things like dental work and things like this are fine, because anything that's fixed will not be susceptible to this. But anything that can potentially move is certainly dangerous. So anyway, that's this very large magnet that's put around you. There's another set of magnets in here as well. Those are electromagnets that can be turned on and off. And then there's that cage that goes around the head. Those are the three component parts of an MRI machine. Here's that cage I was talking about. So how does it actually work? Well, it's as I said, pretty complicated. Here's basically a schematic of all of the equipment involved in getting an MRI image. There are these big magnets that I showed you here, and the so-called radiant coils here, and then the RF coils are the ones that are in that cage around the head. And then there's computers and several pieces of equipment that control the magnets. And basically what happens is that once you're put into this very strong magnetic field, all of the hydrogen nuclei in your brain line up. Normally they're kind of just randomly moving around, and once you're in the scanner, they all line up in one direction. And you're not aware of this at all. This is something that happens right at the atomic level. So you're not aware of this. But this is what happens when you put any object, any substance inside an MRI scanner. All of the nuclei line up, and then a very strong radio signal is beamed into your brain. It's called an RF signal, radio frequency signal. And all of the hydrogen nuclei in your brain, right, which is the most prominent substance in your brain, right? All of the water molecules in your brain are made up of 2 hydrogen and 1 oxygen molecule. And so all of those are lining up, all the hydrogen molecules are lining up. They get hit by these radio frequency waves, and that causes them to be disturbed. And then slowly, after the radio frequency is turned off, they all return back to their state. And as they do, those hydrogen nuclei give off their own radio signals. And that's what the MRI machine picks up. It's basically, you zap them with radio frequencies, and give back off the radio frequency that's picked up by the scanning apparatus. And that makes the signal. Different amounts of energy get given off by different parts of the brain, different really small parts of the brain, can actually be used, just like in a CAT scan, to assemble an image. Basically a pattern of those radio frequencies that are being emitted by your brain after they've been charged by one of these signals. Pretty amazing when you think about it. Basically, an MRI machine is one of these giant junkyard magnets and a radio station. They beam the equivalent of a radio station into your brain, and your brain then returns the signal, and the signal that returns differs as a function of the density of the tissue. And it's a pretty amazing relationship here. We get these incredibly high resolution images as a result of this technique. Right, and so here is a similar set of images from an MRI scan that I showed you earlier for a CAT scan. I don't know, it's probably all the way to the back, it probably doesn't look all that different, but if you were up here and looking really close, you can look at these all places on Blackboard, you can see there's many more details of the brain. And in fact, you can blow these images up, and you can look at literally a few square millimeters of brain tissue with this technique. You can differentiate different things in a few square millimeters of brain tissue. So it really is a high resolution image. And it's been very helpful out of this CAT scan. It gives you a much better image of the brain. Now, why isn't this completely replace CAT scans? Well, it's more expensive. It takes much longer to get an image. And this technique that does the CAT scan, so if they're in a hurry in the hospital, this is probably not the technique that you want to use. But if you need really fine details of what's happening in a particular tissue of the body, this is the technique to use. Here's a really good example of how much you can blow it up. So we're in this region here in the brain, we can blow it up, and you can see literally tiny little collections of neurons using this technique. Right there in the middle of the brain. So yes, enough so that you can label really small different areas. You can see how this solves so many problems for neuropsychologists, right? And neuropsychologists are waiting basically for their patients. It's a very morbid thing, right? You're waiting for your subjects in your experiments to pass away so you can look at their brain. Here now, we can image anybody's brain whenever we want. And it's a little bit expensive. It's about $500 to image someone's brain with this technique. But it's painless. There's little to no risk associated with it. And you get these incredible images that tell you about even tiny little areas of damage in the brain. Allowing neuropsychologists, right, to then correlate bring in that pattern of responses on battery of tests to get an idea about what that brain area might have been involved in. No, I'm not going to ask you about it. Yeah, I'm going to ask you about that. That's an illustration to show you how to do that. This was kind of a neuroscience that we asked you. All right. I'm almost out of time. This is probably a good place to stop. So we're going to pick up the pace a bit. We'll see if there are questions we can ask using these more traditional behavioral methods. The methods most cognitive scientists obtain from behavioral research, right? We've been asking questions about behavior. So really, in your lifetime, the neuroscience techniques now have matured enough that even mainstream cognitive psychologists are now starting to say, yes, we do have the kinds of techniques now that allow us to start asking those questions about how cognition actually works in the brain. And these are those techniques, the techniques that we're going to talk about in more detail today, build upon these so-called structural imaging techniques that we talked about last time, right, called medical imaging techniques. Structural, remember, being techniques that allow you to take a picture of the intact human brain, the alive, behaving human brain, but take pictures that are like a picture in that sense, or like a photograph. So a photograph of the brain. Now, obviously, they're not really a photograph because cameras can't penetrate through the skull. There are special cameras, if you will, that allow us to basically turn the skull transparent so that you actually can see inside what's going on. So these are structural imaging techniques. Structure meaning the actual physical structure, what it looks like. And that turns out to be an important set of methods and questions, what the brain looks like. If for no other reason than, as I mentioned last time, taking structural pictures of the brain has allowed neuropsychologists to correlate what you can see in these structural pictures and the deficits that some individuals who've had brain damage exhibit. So in other words, you can carefully test people's behavior, their cognition, their memory, their language, whatever, and correlate any problems a person might have with images from these structural imaging techniques. And of course, this works very well in patients who've had some type of head injury, some type of either a stroke, right? So we've had these cerebrovascular accidents that cause damage to, usually to focal, in some cases small, but in other cases large areas of the brain. But also people who've had injuries, right, through some type of automobile or motorcycle accident or some type of head injury. And these individuals end up, usually, if you damage your brain, not surprisingly, you end up with cognitive deficits. And the logic, you remember, of neuropsychology is correlating those deficits, in fact, the particular pattern of deficits you show, with what we can see in these structural imaging techniques. And so that's what neuropsychology is. Neuropsychology is an attempt to try to figure out what different brain areas are doing based upon correlating changes in people's cognition or differences in people's cognition and relating those to the areas of the brain that have been damaged. And as I said last time, this is probably the oldest kind of cognitive neuroscience, right? Because in fact, there were people doing this back in the late 1800s. Back when Donners was doing this reaction time experiments, the kind of first cognitive psychologists. There were also neuropsychologists, mainly neurologists, right? A neurologist is a medical doctor who specializes in diseases of the nervous system. And so early neurologists in the late 1800s were the first to kind of use this neuropsychological approach. But as I mentioned, before modern imaging techniques, it was such a painful endeavor because you had to basically wait until your patients died in order to see what part of the brain actually was damaged. So modern structural imaging techniques revolutionized neuropsychology. And interestingly, these techniques started to become available right around the time that cognitive psychology was taking off. So you get these two parallel fields, right? Neuropsychology, using structural imaging and careful testing of patients, and then cognitive psychology, mostly doing behavioral measures like reaction time initially. And these two fields take off, and then they start to intersect. And in fact, this is where cognitive neuroscience comes from. It's the intersection, if you will, of neuropsychology and cognitive psychology. All right, so we've talked now about structural imaging techniques and how they're helpful in neuropsychology. What we're going to talk about next are functional imaging techniques like fMRI. So this is what a structural picture of the brain looks like from a normal magnetic resonance imaging image, so an MRI, a structural MRI. And many of you probably had these done, right? If you've ever had a concussion or been in any kind of accident, your doctor might have said, well, we want to do an MRI just to make sure that everything was okay. And this is the kind of resolution you can get. This is what the brain actually looks like inside the skull. It's pretty amazing. And of course, we talked about the actual machine that we use to do it. And I'll tell you that we now have one of these machines on our campus. It's a research MRI machine. It's a recent addition, it's just about a year old now. And I talked about how this works, right? The person's inside a really strong magnetic field. And a combination of that strong magnetic field and basically the equivalent of a radio station being beamed at your brain are what allow us to take these really fine-grained, high-resolution images of the brain. And this is just an example of one person's brain with lots of different what are called slices through the brain. So you can see this is what a scientist that uses this technique, or usually if it was clinically used, it would be a neuroradiologist who would be looking at images like this and trying to find evidence of, if you've got some kind of accident, some kind of evidence of brain damage. And this is just to compare it to what comes out of a CAT scan. So these are the structural imaging techniques. And here's just, I think I showed you this last time, this is a super blow-up, fairly small region of the brain here. And it shows you just how good these images are. We've got details here that normally would only be visible in very, very high-resolution photographs of the post-mortem brain. All right, so before we actually turn to fMRI, before we turn to talking about functional imaging, let's take a brief tour to another technique that's used in modern cognitive neuroscience. To ask questions about brain function. So far, the only way we've been able to talk about brain function is in neuropsychology, right? We look at people who have some kind of brain damage and we correlate the brain area that's damaged to their change in their behavior. There actually are techniques, though, that allow you to directly probe what the brain is doing without necessarily having to study a part of the brain that's been damaged. And those techniques, some of those techniques, involve using rather than recording from the brain or taking pictures of the brain, these actually involve stimulating the brain in some way. Now, of course, all of cognitive science, all of cognitive neuroscience involves stimulation. Right? We present our participants, our subjects, our patients with stimuli to see what happens in the brain when a person gets a stimulus. That's not the kind of stimulation I'm talking about here. This kind of stimulation is actually directly stimulating the brain itself. So rather than using the sense organs like your eyes or your ears to stimulate the brain, we're going to actually stimulate directly the neural tissue, right, the brain tissue inside the skull. And that can be done two ways. One way is the way it's done usually in patients. Some type of neurological patient is using intracranial stimulation. As the name suggests, intracranial means inside the cranium, inside the skull. Okay, so this means basically putting some type of stimulation device into the brain. Now, much of what we know about different parts of the brain comes from animal studies where scientists have stimulated different parts of the animal brain. What you you might be surprised to hear that actually there are human experiments using this technique as well. You've never heard of this kind of experiment before, where you actually put an electrode inside someone's brain and stimulate it? Anyone want to volunteer to be in one of these experiments? Probably not. I don't think I would. There's certainly risk associated with having this done, and no scientific review panel would ever approve this type of study in a population of people without brain damage. So the people that are in these experiments are people who are already undergoing brain surgery for some type of problem. And usually the problem is epilepsy, it's seizure disorder. A problem where the electrical patterns in the brain become uncontrolled. And when this happens, particularly when it happens in a severe way, the seizures can cause a person to have significant problems. In fact, in a grand mal seizure, you've heard of this, a person loses consciousness and loses control of their body. So this type of epilepsy that causes grand mal seizures, particularly if it's causing them frequently. Some people can have multiple grand mal seizures in a week, and sometimes even in a day. And when the disorder gets that severe, which is a very severe epilepsy, basically can't lead anything like a normal life. So there are drugs that can be used to treat epilepsy, and they are very effective in most people. But in a small percentage of epileptic patients with these severe forms of seizures, the drugs aren't effective. And for these people, right, there's no relief from their seizures. And these are the people, the small, relatively small percentage-wise group of people with epilepsy, sometimes are referred to a neurosurgeon for surgery. And of course, what they're going to be doing is removing part of these people's brains. Usually they try to remove as small a piece as possible, right? The area of the brain that's causing the seizure activity. And this surgery can be very, very effective, right? It can result in complete remission, particularly if drugs are added along with the surgery, and these people lead normal lives after this kind of surgery. Very long story here to describe how stimulation occurs. Why would you use this kind of intracranial stimulation in these patients? And the simple answer is that you want to find out exactly where the seizure activity is coming from, and you want to make sure that when you're about to resect, in layman's terms, to remove the part of the neural tissue that might be causing the seizure, you want to make sure that when you remove that brain, those small areas of the brain, that they don't perform some important function. Right? You don't want to leave a person worse off than before. So the stimulation comes in and figuring that out. What is the brain area where we think the seizure activity is coming from? What does that brain area normally do? And you can get some pretty good ideas about that by actually taking a very small wire and actually touching different parts of the brain and passing a very weak electric current through that part of the brain. And when you do that, the person that you're doing this on will report different sensations in some cases. You might be saying, well, wait a minute, these people are awake? Yes, they are. They're awake. This kind of surgery requires that the patient be awake during this part of the procedure. So what happens is the doctor brings them into the operating room under anesthesia, the skull is open, the brain is exposed, and then the person is brought back to consciousness. And while they're conscious, the neurosurgeon will use this probing technique, right? This wire that basically is used to stimulate different parts of the cortex. And you might be wondering, this must be excruciatingly painful. How can anyone undergo this? And the answer is no, it's not actually painful. And why is that? Why isn't it painful? Yeah. So what's that? It's past the skull, right? So they're directly stimulating the brain, the cortex of the brain itself, right? The part of your body that registers pain, right? When you feel pain anywhere in your body, it's not the location on your body that's feeling the pain. Pain perception is happening in the brain, like all perception. Right? I mean, when we see the world, you're not actually sensing the exact thing in the world. It's your brain that's doing it. And pain perception is the same. So why is it you can stimulate, why can you remove the skull like this? And by the way, they do use a local anesthesia on the skull once you've been woken up. So you're not feeling the sensation that you obviously would feel if someone cut into your skull. But the brain itself, why aren't you like excruciating pain when they're stimulated? Yeah, the brain has no nerve receptors, no pain receptors. Pretty ironic, right? It's literally the only organ in the body that doesn't have pain receptors. So the organ that's responsible for the sensation of pain itself does not have any pain receptors. Now the exception to that is that the blood vessels in the brain do. In fact, anyone that suffers from migraines knows that migraine headaches are vascular headaches inside the head. Regular headaches are not the brain, right? It's the muscles mostly around the brain. And migraines, vascular headaches, are because of the blood vessels in the brain. But the brain itself does not feel pain, it's not registered pain. And you can actually go poking around in there, and people are kind of completely unaware that you're actually touching it. There's no sensation of touch in the brain. So they're completely unaware of it. So this is why you can do this, right? Why people can do this way. It's weird. On top of it, it sounds, so you could actually do this to someone, and it actually works, and it's a fairly simple procedure. I mean, there is risk of infections, there's within surgery. So that's intracranial stimulation, and this is just showing you some images from that. So here's actually an exposed cerebral cortex in the person. You can see some of the doctor's instruments here. You can see these little sticky notes. These literally, the doctor has put little sticky tabs all over the brain here to remind her that basically this is an area I've already stimulated, and somewhere someone's writing down. There's probably an assistant in the operating room writing down what happened when they stimulated area A or area 26. So what does happen? What happens when you pass a very weak electric current? I think I should back up a second. What's the logic here? Why would passing electricity do anything? Why would what would electricity do? Yeah, remember the brain works on electricity, right? So when neurons communicate with each other, they fire these action potentials, one neuron to another. So it's clear that we're talking about using the medium, if you will, electricity, that the brain normally works on. And so the logic here is that if you stimulate with a weak current, kind of like the currents that maybe the brain itself is using in communicating between neurons, then you might see some similar kind of activity. Now the interesting thing is that you don't, right? If I stimulate some particular part in your brain, you don't all of a sudden have some memory of, the dog that you owned when you were 5 years old. That doesn't happen with this kind of stimulation. Well, let me rephrase that. It almost never happens. More often than not, when you stimulate a brain area like this, is that you tend to cause certain sensations or certain perceptions or certain current behaviors to be basically arrested, to stop. So this technique seems to be much better at kind of inhibiting what's going on than it does in actually stimulating some kind of thought process. And why is that? Well, we don't know for sure, but probably it has something to do with the fact that the actual firing of neurons and communication between neurons is an incredibly complex thing that's going on. Right? I mean, remember, just in a small piece of cortex like this, you literally have hundreds of millions of neurons, right? Each communicating with thousands of other neurons in a very intricate, precise way. And coming in here with a wire and putting it down on some part of the brain and turning on some electricity and then turning it off is highly unlikely to stimulate the kind of normal things that are going on in those brain tissues. That makes sense? And so it's going to be pretty tough to stimulate something like the normal experiences that these parts of the brain support. The kinds of thoughts and perceptions that these parts of the brain normally produce. So that's probably why. That's why you can't just go in here and figure out exactly what all the neural circuits are doing. It's also why when you do it, the reason it probably causes behaviors or sensations to be blocked is because probably what you do is by kind of massively stimulating some of these circuits, is that you probably interfere with what they're normally doing, right? And that causes behaviors to be usually prevented. So how do they use this technique? Well, what a neurosurgeon will do is they'll have you engage in some kind of behavior usually. So if they're worried about, for example, removing a language area, and this is usually what they are worried about. They're most concerned with impairing a person's ability to use language, right? Because you think about all of the things that we're capable of doing, making someone mute or making them unable to comprehend language would be one of the worst things you could do to someone. It's also the case that the type of epilepsy that usually is treated happens to be in areas that also support language function. So what they're normally looking for when they do this stimulation is they're looking for some type of interruption in your ability to either process language or produce it. So for example, if they're in an area that they think might be important in speaking, they would have you start to speak. They might show you a bunch of pictures and have you name each picture, one very quickly after another. And they'll be stimulating different areas, and all of a sudden they'll stimulate an area and you'll stop speaking. And that's pretty common that they can get that type of interruption of behavior. They might stimulate another area, and maybe they were having you listen to a bunch of words being spoken over a speaker. And your task is to maybe press a button every time you hear the name of an animal. And they might stimulate an area that, in fact, is involved in language comprehension, and basically that area will, for a brief period of time, stop functioning. And what people will report is that, yeah, I heard speech sounds, but all of a sudden I couldn't make any sense of them. It was like the person was speaking a foreign language or something, even though the words coming from the speaker were in fact in the person's native language. So that's how this technique is used, and it's used pretty, as I said, pretty successfully to isolate where the important areas are, and to give the neurosurgeon, some additional information before they actually try and remove one of these small regions that they're typically in. All right. There's other types of brain stimulation, not always done in epileptic patients. This particular set of electrodes here, as you can see, this is a CAT scan of someone who's had electrodes put very deep into the brain here, into parts of the brain that we know are involved in emotional processing. This surgery has been used successfully in people with chronic and very, very severe depression. These types of electrodes can be used to stimulate parts of the brain that we know are involved in mood regulation. And some of these patients, this works very well to help regulate their mood. Obviously, a pretty severe form of depression would have to be occurring for someone to undergo this type of procedure. Another type of stimulation is what's called single-cell stimulation. Here you bring a really tiny wire. So this wire here is actually been blown up many times under a microscope. This is a wire that's many times finer than a human hair. It's being brought in a very close position here to the cell body of a neuron in the brain. And with this type of electrode, you can actually, by driving a very small electric current, you can actually cause this neuron to fire an action potential. This technique is used extensively in animal research. I don't believe it's ever been tried in humans, and the reason is that there's no known therapeutic reason to do it in humans. You couldn't use this technique to help you figure out questions about what part of the brain a neurosurgeon needs to remove to correct epilepsy. So the technique is not used in humans, but I'll tell you that a good amount of what we understand about certain parts of the brain has come through studies in animals using this single-cell stimulation technique. Usually in conjunction with recording. the electrical activity as well. So these same electrodes that can be used to stimulate a neuron can also be used to record the electrical properties of this neuron here. And we'll get to that in a few minutes. All right, the last type of stimulation technique I want to talk about is what we might refer to as a non-invasive stimulation technique. Actually, there are going to be two of them. So clearly, the previous techniques, right, the ones that involve intracranial EEG or the deep electrodes, deep stimulation techniques, obviously involve compromising the person's body. Right? You have to go into the skull to do that. This type of technique, transcranial magnetic stimulation, does not require that. Right? As you can see here, this person is sitting normally in a chair, and this technician here is applying a device right over their head. And when they turn this device on, it's actually going to induce current flow in the brain underneath it. And by doing that, you can get some of the same kinds of responses that we were just talking about with intracranial stimulation. You can actually cause certain behaviors to cease that were happening just before, depending on where you stimulate it first. You can, in fact, even if you do it over the parts of the brain that we know control the muscles of the body, you can even get some involuntary movement of those muscles. So this is kind of an exception to what I told you earlier, right? If you stimulate the brain, you don't usually get any kind of overt behavior. You actually can with this, although it's not any kind of coordinated behavior. Usually it's something like, maybe moving your fingers like this. Turn this current on and off. This technique, however, is becoming used more and more. In cognitive neuroscience, it's used because it's a really great technique for literally testing whether an area of the brain is actually involved in a function you think it's involved in. Right? And you can test that because by stimulating that part of the brain, you can stop that part of the brain momentarily from doing what it normally does. This technique is very good at this. It's almost like an on-off switch. You can turn off a part of the brain for as long as the stimulation is on, and as soon as you stop it, that part of the brain comes back on. So it really is a very handy technique. Now, the disadvantage of the technique is that because we're outside the brain with this device, is that we can't get the kind of fine control that a neurosurgeon with a very small electrode can get by stimulating directly the cortical tissue right under the electrode. This is a much blunter instrument, as we would say. So you can get, probably as little as a few square centimeters of brain area that gets stimulated from this, but you can't get any finer than that. And so a few square centimeters, you're talking about probably an area like the size of, twice the size of a quarter or something like that. Clearly, in that much tissue, we're talking about, again, millions of neurons. Right? So it's not a very fine-grained, precise technique. But it can be used, right? I mean, this type of procedure is approved for use in studies like the kind that I do. I don't use this technique myself, but I have colleagues who do. And it does not cause any kind of lasting change in the brain. It's been tested very, very carefully. And if used appropriately, all of the effects are really in a matter of a few minutes at most. I will tell you, this is used clinically now. Some of you might know this. This is kind of one of the last lines of treatment for people, again, with depression. And obviously, much better than having electrodes put into your brain. But this type of technique is actually now used pretty regularly to treat depression. It's largely replacing ECT now. So ECT, which we might talk about when we get to memory, electroconvulsive therapy, right? Shock therapy is not all of that. Many people here have seen one flew over the cuckoo's nest, that old movie with Jack Nicholson. Yeah, they don't paint a very nice picture of ECT in that movie, or most of Hollywood. ECT actually is, though, very safe and very effective in people with really chronic and difficult to treat depression. And they don't use it as a method of punishment by any means. You're sedated when they do it. This technique, though, you can be wide awake with when you undergo this kind of treatment, you come in daily for 4 or 5 weeks, and they treat you for like 30 minutes with this TMS, transcranial magnetic stimulation. And you're kind of not even aware anything's going on, but over a period of 4 or 5 weeks, it definitely seems to help with certain kinds of depression. Just an image showing you what it's doing. So basically what's happening is they basically shoot a really strong electric current through a coil of wires. They place that very close to the head and actually induces an electrical impulse straight through the skull. Anyone here have one of those inductive chargers for their cell phone? You just kind of lay it on top and it charges it. And you guys have all seen those. Kind of the same idea, but in reverse. Right? So if your cell phone is like your brain, and that charger is like the TMS device. They've gotten much more sophisticated. I need to update this picture. And now they have helmets. You basically, it's like a giant hair dryer in a salon, and you just put this on top of your head. And you just sit in there and get to read a book for 15 minutes while they do the treatment.
Would this have to be like continuous treatment because of the lasting effects of the stimulation? Like momentarily, or does it sort of like dull the brain in certain areas where it just like ceases flat?
Yeah, we don't really know exactly what's doing it. This is pretty theory-driven treatment. So they stimulate a part of the brain called the dorsolateral prefrontal cortex. And we know that part of the brain is involved in mood regulation. And so the thought is that basically this treatment over a period of weeks kind of helps rewire that circuitry. Some people think that circuitry might be overactive in people with depression. And but through this continual kind of inhibition of that brain area, you eventually kind of reset those circuits. It's the same theory as electroconvulsive therapy that we mentioned a minute ago. That involves a much stronger electric current delivered straight to the temporal lobes through electrodes on the outside of the head. It actually causes seizure in that case. This does not. All right, here's the last technique I'll tell you about with stimulation. This is a more recent approach to doing brain stimulation. It's called transcranial direct current stimulation, TDCS. This is a much weaker electrical current that's just passed through electrodes directly onto the scalp, and some of it penetrates into the brain. And this technique may at some point be used to treat certain disorders. Right now it's not. But there are a lot of experimentation going on that are stimulating different parts of the brain, so you can do different electrodes at different regions of the brain. And this technique has been shown somewhat convincingly to alter certain things like learning. In fact, I tell you this, you guys can go out and Google this, and you can actually build your own TDCS machine, or I don't recommend it. Most people that do this, it only requires a 9-volt battery, but people that do this frequently end up singeing their hair or burning their scalp. So probably not something you want to just try on your own. But yeah, there's a huge interest in this right now, because it might, I say might, very carefully say might, that it could help in some cases with learning problems and other psychological issues. All right, so let's move on now to talking about the functional imaging techniques. And so we talked about structural imaging already with CAT scanning and MRI scanning. Functional scanning, just to remind you, takes a picture of what the brain is doing rather than what the brain looks like. Structural scanning is taking something equivalent to a photograph of what the brain looks like. Functional techniques take the equivalent of a photograph of the parts of the brain that are actually performing some perceptual or cognitive behavior. And so it's a way of looking at what the brain is doing. And of course, as psychologists, as cognitive scientists, that's really what we're interested in. Knowing what the brain looks like can be helpful, particularly if we're talking about damaged brains. But if you're talking about the typical brain, the undamaged brain, we could look at 30 or 40 of you in here, we could look at your MRI scans, and we would have a very difficult time from the structural images to know anything about you. Right? Anything about your intellectual abilities or perceptual abilities, anything. In fact, because all of your brain brains would look somewhat different. There's nothing telltale in the structure of a normal brain that helps you figure out what cognitive behaviors are going on in the brain. You need functional techniques for that. So functional imaging came onto the scene in the 1980s for the first time. A few experimental procedures started to become available. The first of them was a technique called positron emission tomography, PET for short. And here are some early PET images showing the brain from the side here, right? So this is like if you were looking at my brain like this. The front of the brain here, the frontal lobes, the back of the brain, the temporal lobe here. And the way these images work is the warmer the colors get, the more activity is going on in that area. Right, so really a lot of activity happening here, less activity here. In the black areas, there's no particular activity associated with what was going on in this experiment. So what do I mean by that? Well, because we're studying what the brain is doing, in functional imaging, people whose brains are being scanned actually have to be engaged in some perceptual or cognitive task. And you've got to have them doing something, in other words. And for example, maybe you're interested in what areas of the brain are active when you're reading. Right? Reading a slide like this, or you're reading in a book. What areas are important for reading and language comprehension? To do an experiment like that, you'd obviously have people engaged in some kind of reading task. But looking at a brain scan of someone while they're reading really doesn't tell you too much, because in fact, even though this image doesn't show here, pretty much the entire brain is active all the time. All of your neurons are working. The difference is that if you're engaged in some particular task, some parts of your brain are going to be working a little bit harder. This is the logic of functional imaging. Is that the parts of your brain that are engaged in a particular task should be working somewhat harder than the areas that are not engaged. Now the question is, how do you show that? Right? How can you demonstrate that? Well, you have to do an experiment with more than one condition. So in one condition, we have people reading out of a book when we scan their brain. We've got to have something to compare that to, right? And so, for example, in the early experiments like this one, where they did this, they had people reading words in one condition, and in another condition, they had people just staring at a computer screen. Maybe like a little crosshair right in the middle of the screen. So both of these tasks require you to use your visual system to process the screen. But in one case, presumably the language parts of the brain would be working, and in the other, probably not. Right? Staring at a crosshair, probably you're not engaging nearly as many of the linguistic parts of the brain. By then subtracting the images from these two conditions, much like Donders did, right? When Donders subtracted the simple reaction time from the choice reaction time. By looking at the difference between these two tasks, we can see the parts of the brain that are working harder during reading. That's the logic here. The parts of the brain that are involved in reading should produce greater activity in the reading task than the fixation task. All the other parts of the brain, right, that might still be whirring along, generating nerve activity, those should be the same in the two tasks, right? So fixating across and listening to words should activate all the other brain areas that are not linguistic, pretty much the same. At least that's the logic here. So by doing that kind of subtraction then, subtracting one brain image from another, you can see the parts of the brain that are specifically engaged in the task you're interested in. And that's what we're looking at here. Because this is a task for people who are reading compared to the fixation task. So these areas of the brain we know were more active. Here's the device used to collect that data. Looks a little like a CAT scanner. And that's because it works very similarly to a CAT scanner in some ways. So you lay on this bed here, and your head is basically inserted in this donut here. The difference between a CAT scan and a PET scan is you need this machine as well. This machine is called a cyclotron. course in advanced physics, you think you've heard of one of these before. A cyclotron is used to add a radioactive label to certain substances. What that means is, you take a substance like oxygen, for example, and if you put it in a cyclotron and you manipulate it correctly, you can make the oxygen radioactive. So why would you do that? Well, because in a PET experiment, what happens is that when you're laying in the scanner, right, so this is just an image you're looking down on the top of someone's head, there's the scanner here, where the person's head's been inserted into it. In a PET experiment, what happens is just before the experiment begins, right, just before you start reading words, the technician will actually inject you, right, into a vein in your arm. They'll inject you with radioactively labeled oxygen. And so this radioactively labeled oxygen then starts flowing through your circulatory system. Obviously, a good amount of it makes its way to your brain, because the brain uses more oxygen than any other organ in the body, for its size anyway. And when that radioactive oxygen arrives in the brain, one of the things that happens to radioactively labeled substances from a cyclotron is the radioactivity starts to decay. You've all heard of this process before, right? Radioactive decay is basically the process whereby the radioactively labeled oxygen converts itself back into regular oxygen. And when that happens, little subatomic particles from different places in your brain are actually ejected out of the brain. They fly at the speed of light in every which direction. And the PET scanner can read those. Right? Basically, it's like the CAT scanner. Right? Remember, the CAT scanner can pick up an X-ray that's been shot the other day. The PET scanner does kind of the reverse. Rather than shooting a particle through you, the particles that are actually in your brain already, right? Because someone injected you with this radioactive material, some of that material made its way to your brain, and some of it then decayed and can be picked up by this device. Now, how is this going to work? Right? I mean, stuff is decaying randomly in your brain and shooting off like this. Well, how is oxygen actually moving through the brain? When you breathe in oxygen from your lungs, it's transferred from your lungs into your blood supply, right? And then where does it go? Some of it goes to your brain, right? And so this radioactively labeled oxygen is flowing basically through the vasculature of your brain, through the arteries, veins, and through the capillaries, right? The tiny little blood vessels in your brain. And there are literally millions of these little capillaries. Your brain is bathed in blood constantly because it needs a continuous rich supply of oxygen and nutrients for it to work. And we talked about this last time, that if you cut someone's oxygen supply off, basically by blocking the blood vessels in the brain, you basically faint within a matter of seconds. So the brain needs a continuous supply of oxygen. It really is an on-demand system. As parts of your brain start working harder, they basically tell the vascular system, bring more oxygen. I need more oxygen here. And that actually varies by how much different parts of the brain are working. If you think back to our PET experiment, right? So if part of your brain that's involved in reading starts to work harder, more blood immediately starts flowing to those parts of the brain. And if there's radioactive oxygen in that part of the brain, you're going to get more of these basically annihilations, right? The radioactive particle decay. So the PET scanning works on the assumption that parts of your brain that are working harder, that require more blood flow then to support that harder work, that more cognition that's going on there, means that there's going to be more oxygen in that part of the brain. And if some of that oxygen is radioactively labeled, more of that oxygen is going to decay in that part of the brain than in other parts. That makes sense? Fairly straightforward assumption here, right, is that the radioactivity is kind of evenly distributed throughout your blood. If more blood goes to one part of your body than another part, there are going to be more of these decaying particles, which then are going to be picked up by the PET scan. So that's how that PET image is formed. And this is just a diagram showing it here, right? So the radioactivity, this is actually radioactive water, not oxygen, but it's the same idea. In a normal blood vessel, right, if your brain area isn't working very hard, there'll be some of that radioactivity making its way here. And of course, the neurons are needing the things that are coming out of the blood here, so that's what you're looking at. Now, if all of a sudden an area starts to work harder, what happens is the blood vessels in that area dilate. Same thing that happens when you're working out, right? Anyone that works out knows that if you feel the muscles right in your legs, if you've been riding an exercise bike, they get hot. Why do they get hot? Well, because there's more blood flowing to that region is part of the reason why it's getting hotter. Same thing is happening in the brain. Is the part of the brain that is working harder needs more nutrients. The blood vessels dilate and they arrive in that area. So you get more of these molecules, some of them being radioactive and labeled ones, in this area. And if you were able to take a picture of this radioactivity, you would see more of it here than you would here. If this is the fixation task, right, where the language parts of the brain are working very hard, and this is the brain area important in language, this is kind of what you can see. Here in the language task, you see a lot more work going on in these neurons, more of this radioactivity going to this area, more decay, and therefore a brighter signal. So yeah, here we are. Actually, I showed you earlier the hearing works on us. This was some of the very first PET work ever done, where they basically were demonstrating that, in fact, you can get these kinds of images that I'm talking about. This is a proof of concept, if you will. And they showed that in areas where people were listening to words, you got a different pattern of activity than in experiments where people were seeing words. And we know that that would be true because these areas here we know are involved in visual processing, whereas these we know are involved in auditory, sound use processing. So this was a really a huge finding for people in cognitive neuroscience. For the first time, we were actually able to look at changes in brain function as people did different kinds of tasks, and we were able to relate those task effects, those different kinds of cognitive and perceptual tasks, to specific brain areas in intact normal human brains. Prior to this, we had to use neuropsychology, right, to answer these questions. So let's move on to our last functional imaging technique, fMRI. So fMRI really is basically just a less expensive and, as it turns out, probably higher resolution approach to doing the same thing that PET scanning does. It also has the advantage of you don't have to inject people with radioactivity. Again, most people are a little reluctant to volunteer for studies, right, where you're the experimenter tells you, well, we're going to use a needle and we're going to introduce radioactivity into your bloodstream. Even though it's a tiny amount of radioactivity, there is still some risk associated with it. And in fact, in modern times, in the last, let's say, 15 years, PET scanning can't even be done in women between the ages of probably 14 and 50. What do you suppose that is? What's that? It has to do with reproduction, but what do you suppose the risk would be? If you were pregnant for sure, right, you would want to know. So, but they have a way around that. They can give you a pregnancy test before you actually do the experiment. So what other risk factors would there be? Yeah, so being exposed to radiation, right, if they inject you systemically, right, with a radioactive substance, right, it's going throughout your entire bloodstream. Some of it, the interesting stuff is going to your brain, but of course it's going to all the other organs too, back to reproductive organs. And so why wouldn't that be an issue for males? We have reproductive organs too. Ours aren't as complicated, but we have them. So why is it not a problem for men, but a problem for women? What's the difference between the reproductive mechanism? So males in these experiments are just told, obviously this is not a good time in the next couple of months for you to be thinking about, planning a family. But for women, you don't have that luxury, right? You have all of your eggs, so any radioactivity risk that you're exposed to is a lifelong risk. It's a much bigger issue. And for that reason, human review boards that review human research just say, no, it's not worth it, right? To put someone at risk of, a small risk, but still nevertheless a significant risk of genetic damage to their eggs. So yeah, I mean, PET scanning is not useful for, half the population pretty much. And if you're talking about, research volunteers, in fact, in our experiments anyway, we get more women volunteering for our experiments than men. So you really are dealing with a much smaller potential pool, and obviously the conclusions you can draw are much more limited. So PET scanning for basic cognitive science research is not, it's fallen out of favor in the last 10 or 15 years. And it's largely been replaced by fMRI, because in fMRI you don't have to be objective. So fMRI works pretty much the same way as PET. It picks up on blood flow in the brain. So it's using the same assumption that we basically derived from PET. The idea that the brain is modular. In other words, there are different parts, different modules, different components of the brain that are involved in all of these different perceptual and cognitive skills we're going to be talking about. So there's parts of the brain that are involved in memory, there's parts that are involved in perception, there's parts that are involved in language and decision making and so on. And these aren't exactly the same areas, right? They're different. Different parts of the brain perform different components of perception and cognition. And therefore, when these different parts of the brain become active or more active in a particular task, one of the things that we know happens is that blood flow to those areas increases. It's a pretty remarkable thing if you think about it, right? That the brain really, it doesn't make a lot of sense evolutionarily, right? If you were designing a new system, you probably would want to say, this is a really important part of the system, the brain. We want to make sure that it's well protected and that it's immune to lots of kinds of insults and damage that can be done. And it is true that evolution has done that to a large degree, but it didn't do it with respect to nutrients, right? With respect to the things that the brain needs to work. There really is no storage of the important ingredients that the neurons in the brain need to function. The brain basically gets them on the fly as it's doing what it's doing. So when a brain area starts to work harder in some particular task, literally within seconds, the blood flow to that region increases. In fact, it's about 2 seconds. 2 seconds after you start doing a task like reading or listening or whatever, your brain summons additional blood to that region, the regions that underlie that task, that support that particular cognitive function. And because of that, I guess in some sense, fortunately for us in cognitive neuroscience, we're able to pick up on that change. That's what fMRI does, is it picks up on the change in blood flow in a brain area when you compare what the brain is doing in one task to another task. So just like the experiment we talked about earlier, you're listening to words and reading words, and you compare that to maybe a fixation condition. You can do the same thing with fMRI. You can basically image changes in blood flow. So how does that work? Well, there's a hint on this slide here, right? There's a molecule in your blood called hemoglobin. What do you guys know about hemoglobin? Do you know anything interesting about this particular molecule? There's a particular substance that's in hemoglobin. It's in your diet. In fact, it's easy to get deficient in this particular substance. Yeah. Iron, that's right. So iron is an important part of the hemoglobin molecule. In fact, it's a part of the molecule that's really involved in helping the hemoglobin in your blood carry oxygen. And as it turns out, because it's iron, right, it has a magnetic signature. Remember, the fMRI machine is based on picking up magnetic aspects of what's going on in your brain. The structural MRI uses the magnetic spin of the hydrogen nuclei in the brain to form a structural picture of the brain. fMRI picks up on changes in the hemoglobin when it's carrying oxygen, as it is here, right? So here's the hemoglobin and here's a molecule of oxygen. There's a change in the magnetic properties of the hemoglobin molecule when it switches from this state to this state. And as the brain region starts to call on the blood system to send more oxygen, what happens is you all of a sudden get a glut of this oxygenated hemoglobin flowing to that region. You get a big boost in that. And that's what the fMRI signal picks up on. It picks up on the increase in oxygenated hemoglobin compared to deoxygenated hemoglobin in a particular brain region. And that particular response is called the BOLD response, B-O-L-D, all capital letters. And that's because it stands for blood oxygen level dependent. Blood oxygen level dependent is the signal, if you will. It's what the fMRI is reading. BOLD response, you read scientific papers about fMRI, you'll hear them talking about the BOLD response. That's what they're talking about, is that basically this change in the amount of oxygenated hemoglobin in regions that are working hard versus regions that aren't working hard. And because of that particular property of hemoglobin, you can use the MRI machine, right? So the fMRI signal is actually something that any MRI machine in the world can pick up on. In fact, it was back in the 1970s, someone was trying to figure out how to get rid of the blood response, because it actually was causing problems with the nice, pretty structural images you could get. It was considered what we call an artifact, right? Something that made the structural images not as nice. And someone at that point realized, hey, wait a minute, that particular problem that you're having, we might be able to use that. Right? We might be able to actually use that signal to figure out what parts of the brain are working harder. And so they were thinking about what they knew about PET scanning and how, obviously, if you could come up with something like PET scanning with an MRI machine, you would be really in much better shape. You wouldn't have to inject radioactivity. You can, in fact, use a standard MRI machine, right, which every hospital has. Now, many psychology departments around the world have them as well for research purposes. So how does it work? Well, you're laying in this tube here, right? In the MRI machine, here's that the RF coils around your head. You can imagine it's a little hard to do experiments with people laying in a tube like this, right? Looking up at a big donut going around. So how do you do experiments like this? Well, it is possible. And so here's someone actually that did an experiment with visual stimuli. And the person's looking up at two little displays here. Actually, they're looking up at a prism. So you look through the prism, and an image is being broadcast down to two little screens that are right here. And so basically you can show people a visual screen, just like if you were sitting in front of a computer. They're laying on their backs in this machine here. You can also put earphones in their ears if you want them to hear auditory stimuli. You can do pretty much any kind of experiment. One limitation being that obviously if you lay down in a scanner like this and you're sleep deprived, like probably most of you are, people tend to fall asleep in your experiments. So that's one of the issues with having people laying down in experiments. But otherwise, you can pretty much do any kind of experiment, right? You can do a Donders experiment, for example. You show people green and red squares and have them press little buttons. Usually put a little button in your hands where you can press it. So it works quite well. So here is another real big advantage of fMRI. In an fMRI experiment where they're measuring changes in your BOLD response, they can also simultaneously get a structural image of your brain. Remember, this is an MRI machine after all, so we can take a picture of what your brain looks like. Why would you want to do that? Well, if you've got an idea about where different areas are that are active and inactive in a task, you need to be able to superimpose those onto a picture of what the brain looks like, right, to know where it is you're looking. So that's a really big advantage of fMRI, is it allows you to basically capture a structural image of what a person's brain looks like and then superimpose on top of that the different parts of the brain that are functionally active. You couldn't do that with PET. PET scanning does not show you what the brain looks like. It only shows you in 3D space kind of which areas were active and which weren't. You have to take a separate MRI and then put them together. Here we can do that all in one scanning session. So here is a structural image of this person's brain, and then superimposed on top of it the functional activity. So this is a person in a visual task, or in some type of visual task. And these are all the visual parts of the brain here that we know. Lots of work in neuropsychology support vision. And these are the areas that are working harder when a person's doing a visual task. And again, just like with structural MRI, you can look at this kind of bold response throughout all of the different sections in a person's brain. Right? So you can look throughout the entire brain for changes in the bold response in a particular task. So it really does seem like a great technique, right? It allows you with a fairly inexpensive machine, I say fairly inexpensive, to run a participant in an experiment using fMRI costs about $500. So if you factor in how much these machines cost and how much the upkeep is on them, it's about $500 per participant. So I will tell you, it is substantially more money than what it costs to have someone in a reaction time experiment. We can run probably 100 people in a reaction time experiment for that kind of thing. So it's still very expensive compared to basic behavioral cognitive research, but it's way, way cheaper than many, many other techniques. This is just another display of what you can do with fMRI. So before I was showing you what the brain areas inside the brain look like, this is actually fMRI activity on the surface of the brain. So you can do that as well. You can also, because after all, these are computer images, right? All of this is stored in the computer. You can manipulate the image in lots of ways. So for example, you can inflate the brain. What do I mean by that? Well, if you go back to this image here, this is a regular brain here, and you can see the brain has got all of these crinkles, right? All these convolutions. In fact, about 2/3 of the surface area of the brain is inside these crinkles, inside these indentations of the brain. And so when you look at an image like this, you're actually missing about 2/3 of the brain's surface. So by inflating the brain, right, since this is in a computer, you can basically transform the image and you can pop all those crinkles out. This is why the brains look a little bigger and they're smoother. And with this then, you can look at the indentations as well as the protrusions. You can even kind of, if you will, because the brain is three-dimensional, you can like take scissors to this image, right? And cut it in ways that you can then flatten it out. And if you took a beach ball and inflated it, just cut it up so you can lay the beach ball flat. Same idea is here. So it really is a great way to show the different parts of the brain, what's active in a task and what's not. All right, so that's fMRI very quickly. We'll come back to talking about fMRI throughout the semester. We'll talk about various experimental findings. Let me just tell you one other quickly imaging technique that is like fMRI, but involves something called optical imaging. It's not as popular as fMRI because its resolution, its ability to see the fine details in the brain aren't nearly as good. But it can be used very effectively in young children. fMRI experiments, as you can imagine, are difficult in young children. To get them to do on the scanner is not easy. In this technique, you can actually put an array of sensors on the scalp, and the way it works is it shines an infrared light that actually penetrates the skull and bounces off the brain. And as the light is reflected off the brain, you can get some information about blood flow in the tissue underneath these sensors. So you get something like the fMRI signal, although, as I said, not nearly as detailed in terms of how small an area you can look at. All right, so let's turn to our last techniques here. In the last few minutes I've got. We're going to talk about electromagnetic recording techniques now. And this is in some way my favorite area. This is the methods we use in my laboratory. So in electromagnetic recording, what we're talking about is basically looking at the electrical signal being generated by neurons in the brain. After all, it's those electrical potentials that neurons generate when they generate action potentials that accounts for how neurons communicate with each other and give rise to sensory, perceptual, and cognitive processing. So, in some sense, this is a direct measure, if you will, of what the brain is doing. fMRI and PET were indirect, right? Because we were looking at changes in blood flow that are correlated with changes in sensory perceptual processing, but don't actually directly measure those processes. Here with electrical recording, we can, in theory anyway, look directly at what we're interested in, which is these changes in the electrical patterns in the brain. So we already told you about this. You can do this with single cells by putting an electrode close to a neuron. This research is limited to use in animals only. And a kind of classic kind of experiment that you might do this in, right? Here's a monkey with an electrode going into probably 50 or 60 different electrodes going to different parts of this monkey's brain. The monkey might be looking at some kind of computer screen here, and they've been trained to do some particular task. And we can actually look at, these are the firing patterns of neurons, different neurons, depending on, that are firing at different rates as a function of where the stimulus is and where the stimulus is. So you can get really fine grained information about what particular neurons are doing in particular perceptual and cognitive tasks. But again, really you can only do this in animal studies. So what can you do in humans? Well, as we talked about earlier, in patients who are candidates for brain surgery, you can stimulate different brain areas, but you can also record from them. And so actually much more common as a technique is recording intracranial EEG. EEG stands for electroencephalogram. Electroencephalogram is basically the electrical activity that's being generated by the brain. It can be recorded from electrodes placed on the surface of the cortex. And this is what an electrical seizure looks like. So what you're looking at here this is a grid of lots of different electrodes here. And each of these traces is one of those electrodes. And this is time going this direction. And you can see the activity in this one here was pretty excited, but all of a sudden it got very excited. So this is actually the onset of the seizure. So you can get good information about a patient who might be thinking about removing a particular brain region, right? You would want to know that these are the tissue underneath these electrodes are the ones generating this kind of activity. You can also use that technique, though, to ask questions. So many of the patients who are candidates for this kind of surgery, it takes several days or even a week to figure out exactly where the seizures are coming from. So they put those electrodes in, they put the skull back in place, and they wrap the person's head with a bandage, and they actually stay in the hospital, moving around, having normal as you can have, behavior over a period of a week or so, while the activity is being recorded. And during that time, cognitive scientists can actually go in and do experiments. Imagine these people are pretty bored, they're just hanging out in the hospital, they're happy to be involved in something useful. So you can have them do language experiments, perception experiments. You can actually record directly from these people's cortex in a variety of experimental paradigms, just like you would do in fMRI or PET experiments. Fortunately, it's not the only way you can record activity, electrical activity. You can also do that from electrodes placed on the scalp. Usually an elastic cap like this, where each of these little white spots here, underneath that is a little metal disc, and there's a gel that's injected into this disc, and we can then record the electrical activity from the brain as seen from the scalp, right? So as seen from the skin on top of your head. Now, of course, you can probably imagine already that the signal that you see, right, the electrical activity you see, isn't nearly as fine-grained, isn't nearly as detailed as the electrical signal you get by putting an electrode directly on the cortex. I mean, there's a thick skull, there's the scalp, and there's also some other layers of tissue between the electrode and the actual brain tissue we're interested in. And as it turns out, all of that extra tissue gets in the way of seeing a nice, clear electrical signal. So those nice electrical signals you see from the cortex are smeared out and made noisier, is the term that people use, when you record that same activity from electrodes on the scalp like this. So the technique isn't as nice. It certainly doesn't give you the same precision as if you were inside the brain. But obviously the big advantage is that anyone here could be in one of these experiments, and the worst thing that can happen to you is to get some of this gel in your hair that we use to conduct the electricity. So how does this work? Well, basically, the electricity coming through your scalp is measured in microvolts. 1 microvolt is a millionth of a volt. And so a little AAA battery is 1.5 volts. So we're talking about a millionth the size of the output of AAA batteries, which the brain is producing that makes its way all the way out to the scalp. So it's a really tiny signal. So you have to boost it up. We have to use an amplifier to amplify the signal. That's what this is. So we send the electricity from those caps that you're wearing into this amplifier, which boosts the signal, and then you then feed it into the computer. And just like an fMRI PET, we use a computer to kind of assemble, if you will, an image of the electrical activity. And that's what we're looking at here. This is the electroencephalogram. It's recorded from the surface of the scalp. And some of you may have seen images like this before. These are actually the EEG patterns from someone in a sleep study. EEG is used extensively to measure various normal and abnormal sleep issues. Anyone that's ever had problems with insomnia or various kinds of sleep disorders, go to a hospital and have your EEG recorded all night long. And this is kind of what it looks like, right? You get a different kind of pattern of EEG as a function of what state of consciousness you're in. If you're deep asleep, you get these really big, slow-moving waves that go oscillate very slowly and are very large from their peak to their peak. Surprisingly, when you're wide awake and thinking really hard, those same brainwaves are much smaller and much faster. In other words, they go up and down much more quickly. And then in various in-between states here, you can see that there's big changes. The problem is, in looking at EEG like this, right, straight off of the machine as it comes out, you can't really tell much about what a person is thinking about, right? You can get an idea about whether they're really alert or whether they're relaxed, but that's about all. To really see anything, in fact, these are the two prominent kinds of waves you see in people who are relaxed versus wide awake. And they're being generated in the cortex, we know that. We know that the neurons in the cortex, illustrated here, what are generating these signals. In fact, it's electrical activity at the very top of those trees we're looking at, right? So here's the neurons, here's the big trees that kind of sprout from the top of them. These are the dendrites of these neurons. This is where the EEG signal is coming from. Literally, those 100 billion neurons, as they're firing constantly, that's the electrical activity we can see from the sky. And probably this is a good place to stop. So I've got a couple of slides left.
Were you expecting me? I'm subbing for Dr. Holcomb, just this one class for now, in the semester. Somebody here knows me, so I teach another class here. Today we're going to talk about perception, which I just talked about in my other class, and we review for the students who have to overlap. Perception. How the world gets into the mind. This process by which we can assess our environments. We know where we are. We know what we are. An input, in a sense, but not only. you find the face? Raise your hand when you see the face. Okay, it's not easy. I'm going to show you where it is. Time, but there's a face. And it looks like the thing around it. It's hard to find it, to perceive it, get it outside of the whole image. Have a look at this. Is it moving? It looks like it's moving. It's actually a fixed image. Okay? Optical illusion. Here's another optical illusion. Stare at the center cross and tell me what happens. So what's happening? The spots are disappearing. If you go and you look at the spots, you can see that they're still there. If you stare at the middle, they've disappeared. There's only one missing. It's the 1 o'clock spot. Okay? So what I'm trying to point out here is the disconnect between what's actually there and what your brain is perceiving. This is easy to do with optical illusion. Here's something that we might not consider an optical illusion. Is this moving?
Yes.
No, it's not.
What?
What you're seeing, and you know this, is images shown one at a time in sequential order that gives us the perception of movement. These are fixed images. We're so familiar with this in our worlds today, moving images, videos, but these things are not moving. We're being shown image after image after image, and it is creating the illusion of perceived movement. How about this? Is the middle of this lady gone? No, it's an illusion. And this one we can figure out. She's holding a mirror. So I'm giving these extreme examples to point out that the input is the bottom-up sensations that we're given is only part of perception. It's what you're exposed to, what your retina is exposed to, what goes back to your primary vision areas in your occipital lobe. That's one part of perception. The other part is what your brain or mind does with that information. And optical illusions, I hope, have pointed out to you that these are two different things. The input, the bottom up, what's coming in, and what the mind knows about the world. So your mind is going beyond the stimulus, beyond, in this case, what you're seeing, and interpreting it beyond what's present. In those examples, your mind is tricked. But your mind does this all the time. You guys remember this one, right? How many think it's white and gold? How many think it's blue and black?
Because it is.
Flip this. Yeah, I can't for the life of me see it blue and black. I only see it white and gold.
How? I don't understand.
Okay? Here, the same dress. It's obviously blue and black. This dress in this picture is blue and black. This also is an optical illusion. And it has to do with the expectations of your mind on what you're seeing in the case of this image. And we people that are duped into thinking that it's white and gold, we're duped because of the lighting, because there's no person in this dress to show us what a skin tone would look like in this light. And we don't really even know what's going on in the background. So we don't have expectations for this color based on the lighting scheme that we know. Here, we can see skin tones. We assume this is a wedding dress. The lighting is familiar. We can obviously see that it's blue and black. So our minds are tricked into seeing white and gold by top-down processing, by expectations, for those of us that see it white and gold, of this, what our brain is assuming are the lighting conditions in this picture, which are really hard to decipher. In general, our perception perception of color depends on interpreting the amount of light in the room, and when these cues about ambient light are missing, people may perceive the same color in different ways. And you know you have something that's a certain color when you see it in a different light. You take it outside, it's slightly different. You know it didn't change colors. Okay, and your brain is adapting the ambient light conditions to perceive the color. Staircase is going up or going down? It's going down. You could also see it going up, and you can probably very easily flip between the two on this one. If I give you more context, it's easy to see that the staircase is going down. So context helps, and that context is part of your top-down processing, your ability to understand and identify this image. So you have this bottom-up incoming stimuli, which is a visual message that's arriving into your brain, and it is combined with what you know about the world. Top-down, what you know about the world, bottom-up, incoming stimuli. So I just showed you some optical illusions and talked a little bit about expectations and bottom-up and top-down processing. And when the system is duped, I'm going to show you now an auditory illusion. And, yeah. Bottom up is what's entering your mind. And I'm going to drive that home on the next slide. But first I wanted to show you an auditory illusion. And I just want to make sure loud enough.
But there is one illusion that reveals this isn't always the case. Have a look at this. What do you hear? But look what happens when we change the picture. And yet, the sound hasn't changed. In every clip, you are only ever hearing ba with a B. It's an illusion. Take another look.
What?
Concentrate first on the right of the screen. Now to the left of the screen. The illusion occurs because what you are seeing clashes with what you are hearing.
In the illusion, what we see overrides what we hear. So the mouth movements we see as we look at a face can actually influence what we believe we're hearing. If we close our eyes, we actually hear the sound as it is. If we open our eyes, we actually see how the mouth movements can influence what we're hearing.
It's a bizarre effect. Remember, the only sound you're hearing is ba with a B.
Okay. Mind's blown. You're only ever hearing the ba. So what's going on? The input, the bottom-up stuff, is the sound of a ba. actually dupes us into hearing another phoneme, another sound, which is false. Okay? So I hope this is good evidence too that there's more than just stimulus. There's more than just bottom-up, incoming and an interpretation. There's also what you know about the world, prior experience that's having an influence on this incoming information. The incoming information is bottom-up processing. That's what your brain is doing from the input to higher levels. And your experience, your knowledge about the world is having its influence, and that's the top-down processing part. Okay? And in this illusion, interestingly, vision wins. And that should just tell you how important visual speech is, okay, to all of us, not just lip readers, all of us. Have you ever noticed that a phone conversation can be way more difficult than a face-to-face conversation? Okay, you're missing a lot of information of this type, and also environmental. And you're missing some of the frequencies on this one too. So, perception begins with sensation. It begins with these incoming signals, okay, whether it's vision or sound or touch or smell or taste. These senses that I've just cited, these are our physical capacities for input. Computers have keyboards for input. My iPhone has a camera for input, face reading, or taking pictures, right? We have vision, hearing, touch, taste, and smell. Those are how things get into the brain as sensations. We have more. Oh, they have fancy names. Okay, and we have more that we only know by their fancy names. Thermoception, nociception, equilibrioception, and proprioception. They can probably guess if they're detecting temperature, thermoception, pain, nociception, balance, equilibrioception, and something that's not the same thing as balance. Proprioception is the sense of self, where your body is in space, where the parts of your body are in relation to other parts of your body, and its movement in space. We also have sensors for this in our body. So, sensation versus perception. Sensation is passive in the sense that it's just the translation of external energy into a neural signal. Okay? The energy could be touch on my fingertip. There's a neural signal that is treated eventually by my brain. It's a neural signal. Light on my retina, treated in the primary vision area as a signal. Transferred from light to neurons firing. Okay? Same thing, sound wave hitting your ear. Translated into a neural signal. That is sensation. Perception happens after that. It happens in the brain or in the mind, and it's active. It's the interpretation of sensation. So perception goes beyond the information presented and received by the brain. It has to be presented, and the brain actually has to receive it, i.e. pay attention to it. This perception, I like you to think of it as constructive, okay? It is constructive from the bottom up, the incoming stimuli, and what your mind is, what's reverberating in your mind between what you know and what you're seeing. And this applies to simple object identification. This is a normal thing here, perhaps, to have on my desk. You might not know what this is. It would be harder to perceive. Actually, a shopping bag is normal. We know what this is. It's easy to perceive. It's normal to see these things here. And by saying that, I'm saying you have experience with these items in your everyday come to class life. Okay? So perception is not only seeing this thing, the light that reflects off this thing, making it to your primary visual cortex, but knowing what that very well be a water bottle, and not maybe in the back you could think this is a bowling pin, right? You can't see it that well. Perception, what you're actually seeing and what the brain knows about the world. So here's where I'm setting that bottom-up, top-down message home. Bottom-up processing, top-down processing. Perception uses the senses. The senses start this bottom-up processing. We have this incoming raw data that results from firing neurons, okay? And perception calls at the same time on the brain, on a person's knowledge, experience, and expectations. Bottom-up processing is stimulus-driven. Top-down processing is knowledge-driven, experience-driven. So this is a reference to where this processing is coming from, where it's happening. From higher level things like memory and your knowledge system, your knowledge network. These act on the incoming information. Obviously, the incoming information is this bottom-up information, this lower level processing of the incoming information influences the top-down processing. Without the bottom-up, you can't really have the top-down. You have to have the bottom-up to get the top-down started, and together they help you perceive, recognize an object in the world, understand the spoken word. Look at these black shapes. A lot of bottom-up processing going on, right? Or little, it's not that hard to look at. But if I tell you, read the word, I hear oh, okay? So reading relies on a lot of higher level top-down processes. Just looking at those black squares don't. So you engage both systems. And I wanted to separate it like this. First, this bottom-up processing, black blobs on the white background, and when I tell you that there's a word there, boom, very, very quickly, something you're very, very good at, and it's very, very high level, kicks in, and your experience with the world of words makes you able to read that in a flash. This slide underlines how you could think about bottom-up component of processing, and this might look like just vision, okay? And it is just vision until it gets back here, and it's no longer light, okay? It's being treated by your neurons, bottom-up processing, and handed off to higher level processes with which this information interacts. So this is a pretty good example. This is right from your chapter. This is a pretty good example of top-down processing because this arrangement of pixels, this thing, this blob, can be perceived as many different things when you stick it into different scenes. So this is obviously evidence of a brain or a mind, higher level processes telling me that that is a, what is that? That's a shoe. These are, we've changed the orientation of this blob, but it is this blob. Here, it's not a shoe. It's a water bottle, probably, right? And here, a car, vehicle, and here, a person. So that also, I hope, is very good evidence of top-down processing. It's the exact same bottom-up thing. The exact same light is hitting your retina in all of these images, yet you're doing object identification and coming up with very different results based on the context, and that context gives us knowledge of a scene, and that's the top-down processing. I'm going to choose more images. Anybody know what that is? a lot of bottom up, right? You can see it, but you're not having ease with object identification. This is a series. Could be abstract art. And these things are related. So, anybody see anything here? A what? Okay, Earth. Okay. So, okay. Okay. How about this? You still see Earth? What else do you see? A little house? Okay. So we're looking from above. It is the Earth. That was pink. This is white. I have another one that's pink. So I'm telling you these things are related. What we're looking at are aerial photos, which we gathered in the series. And these are salt flats. It's just a really cool aerial photo of salt flats. They turn pink when the shrimp are blooming. Maybe shrimp are hatching, they turn pink like that. But now that I told you what it is, it's much easier to guess what we're looking at. So I've given you knowledge, I've given you experience, and you can look at these photos and interpret them. Looking at those photos, we had bottom-up processing, but it took a lot of top-down processing and knowledge to help us decipher what we were looking at. That's pretty easy to identify, okay? Recognizing this as an arrow uses less top-down processing. And my point here is you use both. You use both bottom-up and top-down processing all the time, but perhaps in different measures, okay? And I thought there was a slide here for that, but it's okay. This is just kind of a thought question. I don't know if you're the talking crowd, but could you have purely bottom-up processing? Something that you see and you recognize with no top-down processing? That's where we have to go, right? Is a system that knows nothing, right? And that would be the only example that I could find, is a system, an infant, a baby that knows nothing and just relies on bottom-up processing. But very, very quickly, actually, establishes some kind of neural network of understanding of its environment. So that would be the only example that I could think of. Purely top-down processing. Think about what I'm saying here. I'm saying no input, no vision, no hearing, no taste, no touch, purely top-down. Perhaps hallucinations. That would be top-down processing within the system of a computer, but probably computers just don't respond. They have inputs that they respond to. So they have bottom-up and top-down. Anybody else here? So where are you from? Yeah, so they are not experiencing, like locked-in syndrome. You can still see. Over here? Dreaming. Dreaming. Yeah. That's one that comes up. But this slide is not to show you that you can have one without the other. Even though you can think of these really marginal circumstances where we might consider it bottom-up only or top-down only in the case of hallucinations hallucinations or dreaming, but in general, when we're interacting in the world, both things are happening in different measure for every object that we recognize, every word that we decipher, everything that we touch and we know what it is. We're perceiving it with both bottom-up, stimulus-driven, and top-down, knowledge-driven processing. You're always using both, but in different measures. Okay? And this one too, if you guys still want to talk. So how are the bottom-up and top-down things related? What's going on in the McGurk effect? Kind of unpack it a little, but what's the bottom-up in the McGurk effect? Oh, ba, ba, ba. Sorry. What's the bottom up? Go for it. Hearing and seeing. And the top down. There's actually a lot of top down going on, even when you hear a ba. And I'm going to tell you that my ba doesn't sound like your ba. And my ba in ball doesn't sound like my ba in, I didn't prepare this, but what? But no, ba, yeah, but that is kind of a different ba. I'm looking for a real ba, like balance. Well, even balance, ba, and balance, and ball, all those bas are different. I was actually looking for a ba that came in a different syllable in the word. Amoeba, I like it. Okay, so the ba in amoeba, even if I'm saying it, isn't like the ba in ball for me. If you analyze them, they're different sounds. And her ba is not like your ba, and a child's ba isn't like an old person's ba. Males and females have different frequencies, average frequencies in their voices, and they're all different. So even to interpret the ba, you have top-down processing that has to do with your experience of the world and how people sound, and what your native language is. Okay? So you have this incoming ba, you have this bottom-up sound, which is ba. You have a bottom-up image, which is a face either making a ba movement or a fa movement. And you have top-down interpretation about what the brain expects to see associated with something that sounds like a ba or a fa. Okay? This wouldn't work if it were ba and ki. You couldn't convince me that it was ki. But these things are so close together that you can convince me that it isn't ba. You can dupe the brain, and it's happening with the top-down processing, with the visual stimulus carrying the race or the game, the visual stimulus that is here. Which brings me a little bit off of visual object recognition, talking about hearing words in a sentence. This would be the energy in a sentence. And what I'm trying to point out here, you might think that you see spaces. Words in spoken sentences are not separated by spaces. Okay? We don't put in spaces when we speak. Yet, we separate the words out when a flood of words is coming at us. Okay? This is actually, what is this? Mice eat oats, and does eat oats, and little lambs eat ivy. And you can see that there are no spaces here. Okay? You might think this is a space. It's pause in the energy that's coming out. But it is not a space between words. So how do we do that? We segment. In order to perceive and understand speech, we have to segment the flow that's coming, the flow of sound that's coming at us into individual words when listening to speech. And you might not believe me that there are no spaces between words. This is called segmentation. Here's an example. Context helps, it always helps perception. But here's the the example that I want to show you. Have you guys know about these misheard lyrics, right? Lyrics they're interesting because we don't have a lot of context. Context helps you understand and segment my words when I'm speaking. If you've ever tried to learn a foreign language as an adult, you realize how important it is to segment the words. And that's when you realize there aren't any spaces to help you, right? It's just coming at you, and you have to find the beginning and the end. In your native tongue, English may or may not be your native tongue, but something like kiss the sky or kiss the sky, it sounds very, very similar, okay? And it's segmentation. K and G are essentially the same sound. There's very little difference between a K and a G. We'll come back to that. So this is, where does this S go? Kiss the sky or kiss this guy? Okay, that's segmentation. So this is just an example. Devoid of context, which songs sometimes are, often are, and we mess up the lyrics. Okay, segmentation takes cognitive effort, and it's an important part of speech perception. I'm going to give you, it's usually too loud, some examples, and I'd like you to try to identify anything, any word, any concept in these streams. Okay.
Bonsoir à tous. Les Pays-Bas et le Mexique font match nul pour le moment, un partout, et les deux équipes se dirigent vers la prolongation. On va débriefer quand même cette rencontre dans une poignée de secondes, alors que nos invités sont en train d'arriver sur ce plateau. Mais pour le moment, c'est Agnès Darfeuille pour un rappel des titres. Bon. Les Pays-Bas mènent donc 2-1 et sont en train d'éliminer le Mexique. Ils sont extrêmement intéressés par ce qui se passe bien évidemment sur le terrain. Et puis on parle pas du tout de la taxe.
Maybe you guessed it was French. Any French speakers? I'm not asking you this question because you can segment. You people who do not speak French, could you segment out any words? Do you have any idea what they were talking about? Yes? No? What? Okay, I'll give you another example. This one's going to be easier. It's Arabic. Any Arabic speakers? Sorry. I want to read back.
Soccer.
Okay, so was the first one, and there were just a few words like goal and Mexico. It was a report after the horrible Netherlands-Mexico match by the French. And there were just a few words, but it's really hard, right? It's really hard to pull these words out. I gave you an example where it was hard, the French, and an example where it was easy, the Arabic. There were other contextual cues in the Arabic one, right? You can hear a crowd, you can hear the music. So it wasn't that hard to start segmenting out people's names and goals, perhaps. So that ability to pull out some words in a foreign language that you might not understand, it's context that's helping you that, and prior knowledge. Knowing that you're listening to a football game or soccer game might enable you to hear different players' names or certain football-related words. we're going to talk a little bit more about the detail of perceiving the phonemes that we speak. And perceiving these phonemes helps us establish the contour, the segmentation of the word. And in order to do that, and because my ba doesn't always sound the same and doesn't sound like your ba, we need a system, right? And the system that our minds have developed is that of categorical perception for phonemes. And first I'm going to tell you what I mean by categorical as opposed to continual perception. Okay? Here are 7 different stimuli, and I could ask you to rate them for lightness of skin tone. Okay? We might look at this and we might say, not light. Okay? Over here I might say very light. And I might say not very light, a little bit lighter, a little bit lighter. That's continual, right? I'm perceiving these things on a continual scale of skin tone lightness. All right? I could also ask you to look at these faces. I don't know if you notice this, but they not only do they go from light to darker, they also go from female to male, or male to female. So if I were to ask you to rate these faces for gender, what would you say? Male? What would you say? What would you say?
No.
What would you say?
Female.
And
Female.
okay, so there's a moment where we flip, and for most of you it seems to be this one flip to female, number 4, right? That is categorical perception. I'm asking you to put these things in category. You have to choose one or the other, and there's a moment where it flips from being one thing to being the other thing. Okay, and they don't give us the same kind of experimental results. Here, this is percentage of people in this class who identified these, in continual perception, the stimulus as, oh, I did it the wrong way. Stimulus 1 is not light, so flip these two things, sorry. Stimulus 1 is not light, and stimulus 7 over here is very light. So, God, it's upside down, I'm really sorry about that. But basically, 100% of the class identified 1 as dark. Oh no, I did it the right way. I did it the right way. As not light. And we get down here to number 7, and almost 0% of the class would call that one. I'm a little confused by I just changed the things on the side. But on the one hand, let's do the gender, and that will be a lot easier. Number 1, right? 100% of the class said that was a dude, right? And then number 2, 100% still said that was a guy, and number 3, 100% still said that was a guy. Here, on number 4, some of you were maybe saying guy, some of you were maybe saying girl, but that's the drop-off point, okay? So this is the graph of something that is perceived categorically. All right, and this is what it would look like if you were perceiving it for lightness of skin. 100% said number 1 is darkish, and 100% said, okay, 100% said it's dark, and over here, 0% said it's dark. So that's what this should be. This should be dark and light. So 100% said dark, and over here, 0% said light, and it fell off in a linear fashion because it is continual. All right, and gender is categorical. And my point here is that we perceive phonemes in a categorical fashion to make our lives easier. Do you know the difference between a B and a P? If you're doing the McGurk effect, my face is exactly the same. Yes, but I don't think that the letting air out of my mouth with a little explosion. In one case it makes a B sound, in one case it makes a P sound. And you guys can follow, those of you who are native speakers of English, and probably those of you who are competent English speakers have no problem distinguishing B, 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 P, B, P, P, B, P, P, what I'm saying. If you just had my face, you would be hard-pressed. And what's happening between the two is tiny, and it has to do with voicing. The difference is very small, 30 milliseconds of voicing. Voicing is turning your voice box on and vibrating. The E part of that is a vowel. Vowels are always voiced, okay? So when I say B, the B phoneme is the first explosion, and it's followed by the E vowel, B, P. And the difference between the B sound and the P sound is I have turned my voice box on 30 milliseconds later in the case of a P and the case of a B. And your brains can tell the difference easily. Very, very easily. Okay? And we have other consonants with the same kinds of contrast, D and T, G and Ki, Ta and Da. Okay, they're very similar and they differ by very little. And yet we don't have any problem distinguishing these when I'm showing you very directly or in the flow of conversation. We're really good at this. And the way that we do this is categorical perception. Okay? I don't spend a lot of time analyzing the sound because it differs wildly depending on where it is in the word, depending on who's saying it, but I classify these things coming at me as being B's or P's. I just said all this. We use categorical perception to identify phonemes, and we do this because there's a lot of variability between speakers and even within one speaker. Okay? And here's that same thing that I showed you with faces. Where here we're going to rate for ba-ness. Now I'm going from B's to ba. Same thing though. Here, manufactured by a computer, because you can imagine that a speaker cannot control how quickly he turns his or her voicing on, right? So by a computer we have a nice ba sound and a nice pa sound, and we manipulate the onset of the voicing synthetically by a computer in steps from ba to pa. And we give these to subjects to listen to, and much like the faces, we say, did you hear a ba or a pa? And over here, 100% of the people responding over here in number one vowel say, yeah, it's a ba. And over here, number two ba, it's slightly less of a ba and a little bit more towards a pa. But they're like, oh no, that's a ba. And number three, yeah, that's a ba. Number four is where it would fall off. And this fall off could be anywhere depending on how poorly or well you made your steps between these two things. But you get to a point where everybody is hearing a pa. What I want to point out is the difference between 3 and 5 is 2 steps. Yet, we know one is a ba. One will be classified as a ba 100% of the time. That's number 3. Number 5 will be classified as a pa 100% of the time, and they only differ by 2 steps. Number 1 and 3 also differ by 2 steps. They're just as different as 3 and 5. Yet, we're going to categorize them as bas. Okay, so it's this steep falling off of identifying a phoneme being one thing or the other thing. And that is learned, and that's very important that it's learned, because without this, understanding speech would be very difficult. And how do babies do this? Right? How do we get this ability? Let's think of it that way. How do we develop this ability? Babies are actually born able to discriminate, and by that I mean tell the difference between a ba and a pa, or between the and da. Okay? They can discriminate the sounds of all languages. They lose that ability in their first 6 to 8 months of life They can no longer discriminate all speech sounds. It's not very useful to discriminate all speech sounds. What they do is they hone in on the speech sounds of their language. I'm going to give an example. I hope sends this message home. It's not. It wouldn't be useful for me to differentiate between someone's two different das when they're really saying da, right? Because I need to know they're da and not. It's not helpful to know they're two different da or the, because that would make that said two different times, two different things to identify when I want to identify it as one word. So what we want is for newborns and people learning a second language is to know when you hear a ba, okay? And not different kinds of ba. So there is no the in the French language. The, okay? Yet French babies under the age of 6 months will discriminate between the and da. They can hear the difference. And once they've developed their phonemic system of French, when they hear the, they classify it as da. Does that make sense? It's not useful to them. It's not a phoneme in their language. And if a French speaker produces a da that somehow sounds like a da, they'll classify it as a da. Okay? How do we know that? How do you test a newborn? Newborns suck more when they're interested in things. Okay? So we can use a non-nutritive sucking method. Okay? So when you change a sound, you give an infant steady da, 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 da. He might suck it first, but he gets bored. Da, 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 da. But if you throw in da, 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 he's going to notice the change. And all babies can hear all the changes of all the world's languages. And they lose that ability as they hone in on the phonemes of their mother tongue and the sounds that they're hearing around them. Like I said, between 6 to 8 months, they can hear everything, but they're working on it. Okay? And if you test those same babies between 8 and 10 months, I think I said this here, after finalizing their phonetic categories, 8 to 10 months, French babies will not discriminate. They will think that the and da are the same sound. Any questions on this categorical perception in general and the use of categorical perception for listening to the sounds of your language? So I gave you an example of some perception that is categorized. But we know that we're good categorizers as humans, and it's very helpful. But sometimes we are faced with ambiguity. Where one stimulus could belong to two categories at once. This could be an A or an H. Right? You don't have any trouble reading that. Okay? This stimulus is ambiguous. So we categorize it as, we read it as an A or an H, depending on the context. That's why you can read that so easily. Okay? I know this word. I don't know a CHT word. Nobody read the CHT today. Everybody read the cat. Okay? So depending on the surrounding context, I can resolve ambiguity. Once again, context. Once again, prior knowledge. So we do this a lot, and for all different types of stimuli, I'm going to give you another example from language. And this one's fun. I think it's fun. the brother of the actor who had met at the party. That's ambiguous. The police arrested the brother of the actor who met at the party. Who was at the party? The brother or the actor? Don't analyze it, just write it down. Who was at the party? The police arrested the brother of the actor who met at the party. Write it down. Who was at the party?
Brother.
Okay, it could be either one. Do we agree? Okay, so there's no correct answer. Your brain has to decide, and your brain's deciding based on what do you think. You don't have context, you don't know the cops, you weren't at this party, it's a random sentence, yet your brain's going to decide. All right, how many of you thought that the brother was at the party? A lot of you. How many thought that the actor was at the party? Okay. I'm going to ask this a different way. I'm going to separate you into groups. Spanish speakers, all Spanish speakers, whether you're native or just proficient, raise your hands. Hi. Okay. Now, it is my mind. Keep them up. Keep them up. Keep them up. What I want to know is how many of you Spanish speakers thought the brother was at the party? Keep it up or take it down. Okay. Now, Spanish speakers who thought it was the actor. It's hard. I wish I had a polling mechanism. I'd say more Spanish speakers thought it was the brother. Agreed?
Okay, so I thought at first, I said, oh, I wrote down the actor. And then I was like, well, wait, I thought about it for a second after I wrote it down. I was like, it's the brother of the actor. And I was like, well, maybe the brother was us, right? So I changed my answer. But originally, I thought it was the actor.
The actor. Okay, so now I'm going to ask that question. And I want your first response. I don't want you overthinking it. So your first response was actor. So now, all you native English speakers, raise your hands. All you native English speakers, raise your hands. And now I want you to put down your hand if you think it's the brother. All you English speakers think it's the brother. English speakers, hands up. Okay? If you thought it was the actor, put your hand down. Okay, so I'm getting mixed signals here. Okay. English speakers only. Who thought it was the brother? English speakers only. Who thought it was the actor? Oh, more people in general thought it was the brother. That's interesting. Okay, why am I going on about this? It's ambiguous. We can't resolve it. If we don't know the cops, we don't know the brother, we don't know the actor who didn't go to the party, right? We can't resolve it. But interestingly, the two languages that I've been contrasting, Spanish and English, have different, and these are not rules. These are preferred patterns in the language. Okay? Because in either construction, the brother or the actor could be at the party. But in Spanish, speaking Spanish, this construction is preferred, high attachment, okay? Where this one goes with this thing, and the first thing you hear goes with the first clause, okay? And that is preferred in Spanish. In English, low attachment is preferred, where you attach it to the closest thing, which would be the actor. So the results, the non-conclusive results that I got here, I was expecting native English speakers to prefer the actor, which you did not. It seems, in my not very scientific counting method. It was true, however, that the Spanish speakers preferred brother. Okay? If you think about your cognitive machine, low attachment is an easier cognitive feat. And everybody here seems to want to attach high attachment. I want to use high attachment attachment. Not everybody, but a majority of people seem to want to attach to brother, which is really interesting. And if we think about that, we're not speaking Spanish here. We are only kind of honing in on the linguistic experience of people in this room who, there are a lot of Spanish speakers, and the English speakers have heard a lot of Spanish speakers talking. Okay, if you're a San Diego resident and you've been talking to people who are Spanish speakers, even without knowing they're Spanish speakers, you might have heard this construction more often. So I could get a different result in a group of native speakers of English in Boston who are not in continual contact with people who might use that other high attachment. Does that make sense? People, it's your lifelong linguistic experience that is going to weigh in on how you interpret this ambiguous language. And so that's what I said. Bilingual Spanish speakers said that they think the brother was at the party, while English speakers will say that the actor was at the party. In general, maybe not in San Diego, okay? It is the language experience of a person that is part of the top-down perception here, which way you push the ambiguity. And sometimes I've done this with the class and I've found that the Spanish speakers prefer actor, okay? And that could be just from immersion in the English language and talking with English speakers, that from their language experience, it's usually low attachment that is practiced, okay? I brought that up here in your, I don't think there's anything like that in your textbook in the perception chapter. In fact, I know there's not. Because I teach a class here, I know you're taking 211, which is an under-division course right now. You're taking cognitive psychology. I teach an upper division course that I want you to know about if you're interested in the questions that I just brought up, questions of language, questions of bilingualism. It's Psych 380. It's called Intermediate Cognitive Psychology on your schedules, but it is taught as a topics class. And in the fall, and right now, I'm actually teaching it right now too. When I teach it, I teach cognition, language, and bilingualism. And it's new that this is a topics class. And I'm the only one who's taught it so far. So far, that's the only topic there is. But when other professors teach it, it might be more cognition and memory or cognition and emotion. But for the time being, and next fall, and probably next spring too, it's going to be cognition, language, and bilingualism. And to give you an idea, this is my selling point, because most of you will be ready to enroll on this next fall or next spring, I believe, because you should be sophomores now. So we take a cognitive spin, I take a cognitive spin, and I talk about the origins of language, why humans talk, how did they learn to do this, and why did they learn to do this. We also talk about language in the brain, native language learning, how babies accomplish this feat so quickly, with so little instruction. We are not teaching our 6-month-old the difference between a ba and a pa. They're just figuring it out, okay? So we'll talk about native language learning, second language learning, what it's like to learn a second language either simultaneously when you're a child or as an adult. Why is it so difficult? Critical periods in language learning. Is it so difficult to learn a language as an adult because there's a critical period for language learning? And if you haven't learned before the age of 7, well, you're just shape. We'll talk about reading. How do we accomplish this feat? These are just squiggles. They're squiggles on a page. Yet we read very confidently, and it only takes us 3 or 4 years to learn to read. We'll talk about the bilingual lexicon. So a lot of students here at San Diego State are bilingual, and they're navigating 2 languages all the time. You probably have 2 words for everything, but almost, right? And you navigate, you must inhibit one while using the other, right? You must listen in one language. It's inefficient to have to listen in both languages. So you have to turn it on and turn it off. You have to pick the right words. How does this all work? So efficiently. Okay? The bilingual advantage. This lifelong situation of managing 2 languages is theorized to produce introduce a bilingual advantage, which means that bilinguals have better executive function. They also might be more creative. There are a lot of theories out there that say very good things about bilinguals. This is different than, 1940, where parents didn't teach. Italian parents didn't teach. Immigrants didn't teach their kids Italian because they thought bilingualism was bad for the brain. It's a complete flip, and we talk about some of those differences of the way we look at bilingualism. So that would be another pretty image. That's all I have for you today, and I'm going to start my class.
So, obviously, no, I wasn't here last time. Professor Midgley was here, and we kind of decided to do that, to have her teach the class. It's part of, I guess, a sales pitch for the next course after you. For those of you who find cognition interesting, probably one of the next courses you want to think about in your junior and senior year would be the course that she teaches, Psych 3, which is MD in cognitive psychology. And as she told you, the way that particular version of the course that she teaches works is that it is a bit of a review of what we're going to be talking about, but most of the class is actually focused on a particular topic area within cognitive psychology, which is language and bilingualism. So those of you who find that particular topic interesting, I encourage you to think about that as a good follow-up. All right, so last time I lectured, we didn't quite get all the way through the last of the techniques or methodologies used in cognitive psychology and cognitive methods. So we did, out here, we talked about lesion studies and classic neuropsychology, structural imaging, CAT scanning, and MRI scanning that have been used to augment and make classic neuropsychology more viable, easier to do. We then talked about stimulation techniques like intracranial and transcranial magnetic stimulation. These are techniques where instead of monitoring what's going on in the brain, we actually alter how the brain processes information by stimulating the brain in some way. And then we talked about functional imaging techniques, which really have been the kind of mainstay of cognitive neuroscience. What cognitive neuroscience has kind of added to cognitive psychology largely comes from studies in this area, where you can study humans, just like you do cognitive psychology, and you can study intact normal individuals, people without brain damage, people without some type of disorder, the kind that you have to study when you're studying neuropsychology or stimulation. Instead, you can study right everyday undergraduates, right? In fact, most of the studies that have been done on people like yourselves. Many of you psychology majors know that one of the requirements of interest psych is you have to be involved in an experiment. Many of the participants come from modern functional imaging techniques, so people like yourself. Of course, the advantage being that these functional imaging techniques, for the most part, are non-invasive. You can do them on people without any type of damage done to the nervous system itself. In particular, fMRI has been very, very useful in this type of research. fMRI uses just a regular MRI machine that's been tuned, if you will, to allow you to take a functional image of the brain. In fact, we just recently, in the last year, we now have an fMRI facility here at SDSU. It's right next door to the engineering building. So yes, this has been a huge boom to cognitive science and cognitive neuroscience. The ability to bring everyday people into a scanner and to do the kinds of experiments, the kinds of cognitive, perceptual, linguistic experiments that we'll be talking about all semester. And to do them in a way that allows us to actually image the brain as we do those experiments. Actually figure out where in the brain the various perceptual and cognitive and linguistic processes are taking place. However, I'd be selling cognitive neuroscience short if I didn't tell you about this last group of techniques, which in fact, for the most part, also can be done non-invasively. In other words, you don't have to invade the body to do this. You can do these in everyday people without any kind of lasting damage. Well, most of them. Single unit recording and intracranial EEG, of course, that's not true. You do have to go inside the brain to do those. But the last two techniques, MEG and EEG, and the term surface here refers from the fact that you can actually get these electrical events from the surface of the scalp, right, by putting electrodes on the scalp. So let's turn to electromagnetic recordings. I'm going to skip over the first part because I talked about this in some detail last time. So this is single unit recording, almost exclusively done in animals. Some of our best information, particularly with regard to sensory and perceptual processing, comes from this technique, because you can put electrodes right into or right next to the neurons that are actually generating these sensory and perceptual experiences in the brains of an animal. So much work has been done in lots of different species, from mice and rats, cats and dogs, although that's not done anymore. That's been done back in the 60s, to studies in monkeys. Lots of studies have been done and are still done in monkeys using this technique as a way of listening to what individual neurons are doing as an animal is perceiving, sensing and perceiving the world around it. Here's the slide I used. So this is a monkey with electrodes in the brain. The monkey is processing some type of computer display. And we're actually listening to a group of neurons in this monkey's brain as the monkey performs some type of perceptual task. And from this kind of experiment, you can isolate the individual neurons and the properties of those neurons in the sensory parts of the brain, the perceptual parts of the brain, and figure out exactly what they're doing. You can do something similar in humans, right? Because these, again, are patients who have epilepsy and who are undergoing surgery to resect or to remove a part of the brain that's causing seizure activity. But before they do, frequently neurosurgeons will place a grid of electrodes right onto the cortex, and you can record the EEG from the cortical surface, not from the scalp surface, but the actual surface of the cortex. And the advantage of this is that, of course, these electrodes are much closer to the parts of the brain that we know are involved in, for example, sensory and perceptual processing. So this is a technique with higher precision than the next techniques which we'll talk about, which are the ones that are non-invasive. All right, so non-invasive electrical recording is possible because, as it turns out, when your brain is generating all of these electrical events during sensory and perceptual, linguistic, and so on processing, some of that electrical activity actually leaks out of the brain and conducts all the way to the surface of the scalp. And you can record that electrical activity by placing electrodes, which are small metal discs right on top of the scalp. And usually you inject a small amount of electrolyte, so it's basically salt water, that makes the electrical contact between the metal electrode that is here and the actual scalp. How many people have been in one of these experiments? We regularly recruit people like yourselves to be in our experiments where we use this technique. And the worst part of it is sometimes the experiments are a little boring, and your hair gets a little bit of this kind of electrolytic gel that washes out very easily. Otherwise, the experiments we do are really no different than the experiments that our ancient colleague Donners did. Right? We have people looking at stimulus displays and making decisions and pressing buttons while we record their electrical activity. All right, so yeah, this is just a schematic of the way the system works. So the person that's a subject or participant in the experiment sits in a room, they're wearing those caps, the wires are coming out of the caps. They're maybe reading words off of the computer monitor, and we're actually recording their EEG while they do that, their electroencephalogram. So the electroencephalogram goes to these wires, it goes into this box here, which is an amplifier. Now, what's an amplifier? All of you who have stereos at home, parents who have stereos at home, and people that actually have stereos at home, right? Use your phones for music. But if you, your parents have like an amplifier at home where they play their records or DVDs or CDs for music, it's basically the same thing. What this device does, is it takes the really tiny electrical potentials that are being recorded, that are being produced in the brain, and it enlarges them. So why do you have to do that? Well, the EEG is actually in units of, I think it's microvolts. So what's a microvolt? A microvolt is 1/millionth of a volt. Those of you studied electricity before, 1 volt is actually fairly small in itself. A little AA or AAA battery is 1.5 volts. So the brain waves that are being produced and are coming out to the scalp are less than 1 millionth of what a AAA battery is out. So this is the reason we have to amplify the signal. It's too tiny to actually do anything with it unless you boost it up. The same way, a stereo amplifier boosts up to very small electrical potentials coming off of the CD or coming out of an MP3 player. Same basic principle. You take a small signal and you just multiply it, making it much larger so we can now do something with it. And actually, in this case, what we do is we store those electrical signals in the computer. And then later on, we can go back and actually plot out on graph paper, on regular paper, what the brain waves look like. And what you get is something that looks like this. Right? Once the EEG has been amplified and recorded and stored, you get traces that look like this. And this is time going in this direction here. And what you're looking at here are really just kind of raw EEG waves that are being displayed on a computer monitor after they've been recorded from a participant. And the main thing to note from this slide is that in looking at this kind of EEG like this, one of the things you can really see very clearly is that the waves differ as a function of, for example, your conscious state. So if you're in a deep sleep, the EEG waves that you produce are much bigger. They're these really slow oscillatory patterns like this. If you're just kind of in a regular kind of sleep mode, or you spend most of the night actually, you get some of those big waves like we saw down here, but you also see a little bit of faster, smaller wave activity as well. And by faster, I mean the wave goes up and down more quickly. And then lastly here, in a state of, right perhaps when you're first falling asleep, you can see the waves are even smaller in amplitude. They don't have nearly as many of these big, slow, oscillatory patterns to them. And then lastly, the top two traces are the traces that you see when people are awake. The two prominent types of activity that can be seen in the EEG when someone's awake. For example, when you're very alert, right? Like probably when you first walked into this class, most of you were producing most of the data. Maybe by the time the class is over, maybe you'll be more in a state. Hopefully, most of you won't be here quite yet. But these are really, really consistent kinds of waves that you see in everybody. Right? Everyone produces this type of activity. And herein is some of the problem with EEG, as I'm showing it to you here. And that is that it's not highly specific. Yeah, so here are the two awake patterns you see. So here are those really small but very fast brain waves that are sometimes referred to as theta waves. And here are the much larger and slower. Slower again meaning that the waves are moving from peak to trough much slower than they are up here. But they're higher in amplitude, and these are so-called alpha waves. Alpha waves are associated with being very, very relaxed. And in particular, the visual parts of your brain, which are at the very back of your brain, produce a lot of alpha waves when you close your eyes. As soon as you cut off visual input to the visual parts of the brain by closing your eyes, that part of the brain generates a large amount of alpha. And then as soon as you open your eyes, the brain waves pattern change from this to this. So a really obvious change. In fact, we could wire each and every one of you up here, and I could probably demonstrate in 90% of you a really clear pattern like this. The other 10%, we probably see some of this, but maybe not quite as obvious. So this is a very consistent pattern across people. But this is about as good as it gets in looking at EEG. You can tell whether someone's wide awake and actively processing information versus when they're more relaxed and perhaps in a state of contemplation. But that's about it. So you might be wondering, why am I telling you about EEG with relation to cognition? If in fact, really all you can tell is if someone's very alert or not very alert. Well, the answer is there is a way to do it. There is actually a way, buried within these signals, are are actually other electrical signals. They're just so small compared to what we're looking at here that you actually can't see them. They're tiny, tiny changes that we know actually track perceptual and cognitive processes quite well. And I'll show you those in a minute. But before I do, let me tell you where the EEG is coming from. I mean, you know it's coming from the brain. But because we're recording it from electrodes on the scalp, it's not quite so obvious exactly where the brain is coming from. And it's actually required many, many studies, some of them in animals, some of them in patients, to figure out exactly what is the source of the brain itself. And the answer seems to be that, I guess luckily for us, because this is why the technique is so useful, is that most of the activity that we see in those electrodes on the surface, most of that EEG, actually is coming from neurons in the cortex, right? Remember what the cortex is. The cortex is that small gray outer mantle that covers the whole surface of the brain. Right? It's what you see when you see like someone showing you a picture of the whole brain. You're seeing the cortex, but in fact the cortex is actually very thin. It's only a few centimeters at most, and in some cases only millimeters thick. And of course underneath the cortex is all that white matter that is the axons, right, that allow neurons to communicate with each other. We know the EEG is mostly coming from electrical activity in the cortex itself. And this is a blow-up of what the cortex looks like. So if we took a small segment here, say right here at the top of the cortex, and we were to really enlarge it so that we can see the individual neurons, this is what you'd see. These are the really big neurons that are called the principal neurons of the cortex. Principal because we think that most of the information processing that the brain is doing is happening in these neurons. These neurons are doing most of what we would associate with sensory, perceptual, and cognitive processing. And they're everywhere, right? You could get, if you just take a tiny little piece of cortex and look at it, this is what it looks like here. But we could go anywhere on the cortex, and if we blew up the image, you would see something very much like this. There's really uniform concentrations and views of what neurons look like as you go out throughout the cortex. Very, very similar. A neuroanatomist could tell you differences. They could tell you by looking at a slide like this, more than likely which part of the cortex this is coming from. Because they would know based on lots of other properties. But by and large, this is the kind of schematic, if you will, of what the cortex looks like. And it's these neurons, and of course, when they fire potentials, that actually result in cognition. Now, I'm going to show you in the next slide here, kind of just a cartoon-like image of one of those neurons, right? So here are what's called the cell body of those neurons. This is called a dendrite here, and the axons are coming out the bottom. So in this next slide, that's what we're looking at. Here's the cell body. Here's that dendrite that's going up towards the top. And here's the axon going out the bottom. And of course, these axons, once they leave the cortex, are highly myelinated. That's what makes that white matter. So basically what we know from lots of studies, as I said, is that the EEG is produced actually by a circuit of activity in these long dendrites that project all the way up to the surface. Go back to the slide here. These dendrites here, which go all the way up to the cortical surface, there's an electric circuit in those dendrites that we know is what is responsible for generating the EEG. And like I said, lots of experiments have shown this. So it's basically activity in one of those circuits. And just to give you a bit more detail, this isn't a super critical part for you to know, but what induces the activity in that circuit is actually an axon coming from another neuron. That's what this is here. You can't see the other cell, it's cut off. But this is actually the end point of an axon, just like this one, coming from some other neuron. It's coming into our target neuron here, and it's this axon that's going to induce a current flow, a current change in this dendrite. And that's what the EEG is based on. Now, when you're looking at the EEG from the surface, it's obviously not a single neuron that we're looking at. What are we looking at? Well, we're actually probably looking at tens of thousands, maybe even millions of neurons, dendrites, and axons like this, all kind of synchronously firing. Right, so we've got one electrode that we're looking at, and we're looking at the EEG activity. You're looking at the synchronous firing of all of the neurons underneath that particular electrode. And that, like I said, could be tens of thousands or even millions of neurons firing synchronously together. So that's what produces the EEG. Another technique that's closely related to EEG that I'll introduce now, before we go on to talking about the technique that I use, is a technique called magnetoencephalography. Now, all of you have heard of EEG before. How many people here have heard of MEG? And maybe a few of you that have gotten another course in psychology have heard about MEG, magnetoencephalography. The reason it's not so well known is because it requires a machine like this to record it. And this machine is as expensive or more expensive than an MRI machine, so millions of dollars. Whereas the machine necessary to collect EEG, like I use in my lab, is on the order of thousands of dollars. So, very, very expensive machinery. Why would anyone use this? Well, as it turns out, when neurons, as I was showing you in the previous slide, are firing in those circuits, right, that generate the EEG, they also generate a magnetic signal. Any of you that have had a course in physics or engineering probably know this, that anytime you have a current flowing in a circuit, right, so if you've got a AA battery, you've got it hooked up to a light bulb, and electricity is flowing from the battery to the light bulb to light it up, there's an electrical current flowing, but actually you can measure, if you have a very sensitive device, that that same circuit generates a magnetic signal. And if you have a really, really sensitive device, like the MEG machine, you can monitor that magnetic signal. And that's basically what it's doing, is it's monitoring the magnetic equivalent of the electrical activity we see in EEG. Now, why do that? Right? I mean, you've got the EEG, why would you want to bother with the magnetic signal? Well, the answer is, to put it really simply, is that the EEG signal, as I hinted earlier, is a little bit messy. It's very difficult with the EEG recorded from the scalp to figure out exactly where in the brain it's coming from. We know it's coming from the cortex, but to isolate the exact location of the cortex as producing a particular EEG pattern on the scalp is actually impossible. It can't be done. It's what's called in physics or math an ill-posed problem. There's no method that would allow us to backtrack from that electrode to the neurons that are producing the signal. And therefore, EEG has what we call really poor spatial resolution. What does that mean? It basically means that we can't figure out exactly where in space, in other words, where in the space of the brain the signal is coming from. Any given EEG that we see on the scalp, it's not possible to know exactly where it's coming from. So that was a long explanation for why MEG is interesting. MEG, it turns out, does not have the same limitations that EEG does. It's much easier, mathematically, to localize the source of the magnetic signal. So MEG can be used in the same way EEG is, but with better precision at figuring out where in the brain the signal is coming from. Why is that important? Well, it's what cognitive neuroscience is about. Right? Cognitive neuroscience is interested in understanding where the perceptual and cognitive processes are actually coming from in the brain, how they interact with each other to give rise to cognition. And so knowing where something is in the brain turns out to be pretty important. And MEG is just better at doing that. The downside, of course, is that it requires a really expensive machine to do it. And it also requires a special room. Because the Earth's magnetic surface is several orders of magnitude greater than what your brain produces, you have to have this special room that shields out all of the magnetic signals coming from other sources like the Earth, like any other pieces of equipment. Because the magnetic signature of your brain is tiny, 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 just like the electrical signal. So, yes, a great technique for helping localize things in the brain, but very expensive to use it. And in fact, there's only a handful of these machines around the world. Actually, there is one here in San Diego. But yeah, you can probably count on your fingers and toes the number of MEG machines around the world. So it's not a super popular technique, largely because of its expense, but also because no one has figured out yet how to use it clinically. This is why people are willing to spend millions of dollars on MRI machines, is because, yes, they have great clinical usefulness. MEG so far is not proven to be all that helpful clinically. And so the machines are only used for research pretty much. All right, so let's move on to the very last topic, time-locked electrical recording. What does this mean? Well, so far I'm talking about MEG and EEG. I just told you about what the EEG signal and the MEG signal look like if you record it from the brain. You get this kind of continuous squiggly lines that come out of the brain. And I've already told you that those squiggly lines aren't very unique. They're not very good at telling you what the differences are in different kinds of cognitive or linguistic or perceptual processes. Right? So the brain waves that you produce when you're listening to music versus listening to someone speak versus reading a book versus watching a video without an auditory soundtrack all look really similar. The EEG looks almost identical in all of those cases. And so the raw EEG by itself is not very useful at trying to isolate the different perceptual and cognitive processes. But as I suggested a minute ago, buried within that EEG are signals that are unique, in fact, very unique with regard to all of the processing that's going on. So how do you extract those signals? If they're tiny, tiny, and they're buried within the EEG signal, how do you actually pull them out so you can see them? Well, the answer is you basically collect regular EEG, which is what this is. This is a participant in our laboratory in an experiment. These are all of the electrodes that we're recording electrical activity from, and this person's watching some type of stimulus display in front of them, and we're monitoring their EEG as they do that. So this is how it works. So in one of these experiments, right, the person's sitting here and they're reading words flashing on a computer screen one at a time. And while that's happening, we're recording their EEG. And what we're doing is here, what you see here are when and what kind of stimuli we're presenting to the subject, the participant. So at this point in time, we presented the word car. About a second later, we presented a word like pen. A second later, a word like cat. A second later, a word like dog, and so on. We just present lots and lots of words, one after another, and we record the EEG throughout all of that time period. What we do then is what gives this technique its usefulness. We take the little segments of EEG, so we take this segment, and we take this segment, and this one, and so on, and we average them together. What does that mean? Well, it's what you guys learn when you take statistics or even high school math classes. You all know how to compute the mean, right? How to compute the average. What we do is we average these waves together. And specifically what we do is we average all of the waves from one condition in an experiment together, and then all of the waves from a different condition, we average those together. And the resulting waves that come out of that are then superimposed, allowing us to compare the two conditions in an experiment. So for example, in this case, what we could do is we could look at the averages of all of the words where the word previous to it was completely unrelated to it, right? The word car and pen had nothing to do with each other. And then we can average together all of the words where the previous word was, for example, a word in the same category. The cat and dog are both cats. So what you're looking at down here is actually a whole bunch of words where the previous word was a semantically related word, and a whole bunch of words where the previous word had nothing to do with the current word. And I think what you can see is that unlike these kind of very chaotic squiggly lines up here, you can see some nice patterns here. So in the case of the unrelated word, like pen, I colored it yellow because that is the yellow line. And the word dog, words like dog, is the white line. And what you're looking at here, this is the point in time when the stimuli were presented. This is basically time 0, right? As soon as the stimulus comes on the screen. And then each of these little marks on the x-axis here is 1/10 of a second. So from the time a word is first flashed on the screen until a point in time when you can actually start to see these two conditions producing very different waveforms. That actually happens 2/10 of a second after those words come on. So within 2/10 of a second, your brain is differentiating words that are preceded by semantically related words from words that are preceded by unrelated words. What is this telling us? It's telling us something about how your brain is processing the meanings of these words. And it's telling us a lot about when that happens, right? When with respect to when you actually read those words. So within 0.2 of a second, your brain realizes, in one case, that the word you're looking at is not related to the word before it, with the yellow line. And in the other case, that the word is related. And this time, basically these differences from about 0.2 of a second out to about 0.4 of a second is a really reliable phenomenon. We'll get to this when we talk about language in a few weeks. But this is a very reliable phenomenon, and this technique really illustrates how you can use brainwaves to very, very carefully catalog and categorize how long it takes for certain things to happen in the brain. So this technique is really good at telling you when things are happening. Now, just by way of comparison, if this had been a reaction time experiment, where people had to press a button, let's say you had to press a button whenever you detected a word that was related to the word before it, where would that button press be happening in time? Based on the Donders experiment, so imagine how quickly can people press a button, make the decision about whether two words were related or not. Yeah.
410, 440 milliseconds.
That's good. So that you're in the ballpark. Probably 100 milliseconds later on average. You might find some people who are really fast and might tick off a response within 0.4 of a second. So where is that on this graph? Well, that's out here somewhere. So the actual response you produce, if you were going to estimate where these effects of relatedness between words occur, you'd have to say, well, 400, 500, 600 milliseconds. These event-related potentials, or ERPs, are telling us that, well, in fact, already by 0.2 of a second, 200 milliseconds, the brain has already seen that difference. So you can see this technique has the ability to much more carefully localize in time when these various phenomena are happening. You might remember that I made a very big deal on the first day when we started talking about methods about time and how important time is with regard to where and how things are happening in the brain. So this technique turns out to be very valuable in that regard. It's very good at talking about when things are happening relative to other things. Not so good at telling you where they're happening. Right? For the reasons I mentioned, it's hard to know from scalp recordings of EEG and scalp recordings of ERPs exactly where they're coming from in the brain. All right. So I just want to finish with this slide here. This is a pretty complicated slide, not super important to fully understand this. But what I'm showing you here is kind of a classic picture summing up the whole field of cognitive neuroscience. So what is plotted here are basically a whole bunch of techniques that we've talked about, right? So we talked about CMS, we talked about ERPs and energy just now, we talked about unit recordings, optical imaging, functional MRI. Each of these kind of colored circles here is depicting what is called the resolution of that technique. And what do we mean by resolution? It's just talking about temporal resolution with regard to ERPs. Right? ERPs actually measure things down to the level of milliseconds, thousandths of a second. We can differentiate things happening on the order of milliseconds, or tens of milliseconds, with EEG and ERPs. So they have really good temporal resolution. And where is that shown on here is on the x-axis. Right? The resolution in terms of time is on this axis here. So when you look at a technique like MEG and ERP, what we're seeing here is that, yes, it goes all the way down into the millisecond range, and it's also sensitive to things happening all the way up in the, well, approximately 1 second range. Right? So that's its temporal resolution, is this kind of extent across the x-axis here. What about its spatial resolution? In other words, how good is it at telling you exactly where in the brain something is happening? Well, the reason MEG and ERP are all the way up here is because of the problems I mentioned earlier, right? They're not very good at telling exactly where things are happening. MEG may be a little bit better than ERPs or EEG, but not great, right? So what you're looking at here is pretty much this technique it can tell you, yeah, it's happening in the brain. We know it's happening in the brain, but we don't know exactly where. So so its spatial resolution is not great. Let's look at something with really good spatial resolution. Well, unit recording, right? So recording from single neurons or a handful of neurons, including electrodes in the brain, you can really get great resolution here by column, layer, and neuron. We're talking about literally single neurons. So the spatial resolution is quite good. And the temporal resolution is pretty good as well. And you can get information down to the millisecond level and all the way up to the second level with this technique. All right, so you start to see how this works. Let's look at fMRI here. So here's functional MRI. And notice that its spatial resolution is better than EEG and imaging, right? It's going all the way down here to the level of, well, maps and columns, which are relatively small areas of the brain. However, notice what is traded off. What's the problem with MRI on this graph? It's temporal resolution, right? Notice that the temporal resolution of fMRI is really in the 1 second and longer range. And that's, of course, because remember what fMRI is measuring. It's measuring changes in blood flow. And while blood flow tracks very nicely with cognition, it still lags behind it. It's what we call a sluggish signal. It takes a while for, when your brain starts working harder in some area, it takes a while for the blood to increase its flow to that region. And for that reason, the MRI signal actually does not have very good temporal resolution. You cannot distinguish with fMRI the kinds of things that I was showing you earlier with those word pairs. Pairs of words that are related or not related. It'd be very difficult for you to know exactly when differences between related and unrelated word pairs actually are occurring in the brain. Although you might be able to figure out very nicely where they're happening, which parts of the brain are sensitive to that. You wouldn't know when. Because in fact, it takes, like I said, seconds for the MRI signal to actually start. Okay. So, yes, the bottom line here is that by looking at this, spend some time looking at this, you can get an idea of what the advantages and relative advantages and disadvantages of various techniques are in terms of their resolution in both time and space. And as it turns out, there is no perfect technique. There are no techniques that are superior across the board to other techniques. If you're interested in questions about time, some techniques are better than others, but usually there's a trade-off associated with that. So usually you're not going to be as good in space, you're not going to be as good at localizing in other ways. On the other hand, if you're interested primarily in localization, then other techniques like fMRI will be better, but you'll trade off the ability to localize in time when things are happening. Hopefully someday we'll have techniques that are good at all of these. But for now, one of the approaches in cognitive neuroscience, one of the most fruitful, is the combination of techniques. Right? So you try to combine together the recording of electrical signals like EEG along with MRI signals, in order to try to solve the problem of both when and where things are happening in the brain. All right, any questions about this? We're going to move on now where we're supposed to be, which is on perception. So, like I said, techniques are important, and we'll be obviously coming back to them a lot this semester as we move on to the various topics. So Professor Mitchell gave you kind of an introductory level lecture to sensory and perceptual process. And what I'd like to do now is kind of jump in at a slightly higher level and focus on a particular kind of visual, a certain kind of perception, which is visual perception. Now I'm going to talk about some of the other modalities as well in passing here, but we're going to mainly focus on visual perception, which of course is our ability to process information in the environment using our sight. Again, with Homer here, what happens in the case of a visual perceptual experience is, of course, is that there's an image in the real world that you focus on. That image comes into the eye here, it's projected onto the back of the eye, onto the retina. Right? The retina is basically the photochemical surface at the back of the eye where images are first processed. then that information is sent, it's converted into an electrical signal. Now, this is basically what sensory processing is. Sensory processing is converting some type of environmental energy, in this case light, into an electrical impulse the brain can then deal with. That happens in the retina, and then the information is transmitted as electrical impulses all the way back to the back of the brain, as I discussed earlier, to the part of the brain called the occipital lobe. And then the information actually, surprisingly, goes all the way to the back, and then it starts to make its way forward as you try to figure out what it is you're looking at. So the process of visual perception is one whereby the information initially arrives at the back of the head and then slowly makes its way towards the more front and more interior parts of the brain. One of the reasons I want to concentrate on the visual modality is that arguably it's our most flexible modality, our most flexible sense. It allows us, for example, to see things that are very close to us, right? You can see objects, within a few inches in front of your face. Right? So, if a cat has climbed up into your lap and staring right at you, you can easily make out the details of the cat's face with your vision modality. This is what we refer to as a proximal stimulus. Proximal meaning is very close to you. So the visual modality is capable of doing very fine brain processing for things that are very close to us. Right? Hence, our ability to read. Reading is a really good example of a very, very elaborate visual perceptual skill that we have. Right? We can read those tiny little squiggles that make up letters and then together make up words. And we can do that very efficiently with our vision. It's also possible, though, with vision, and this is perhaps the most flexible sense, to process stimuli that are very far away. So, for example, things that are hundreds of thousands or millions or billions or even light years away can be processed by our visual systems. Photons of energy, right? Light rays coming from very, very distant objects arrive at the retina and allow us to actually get some information about what it is that's very far away. And these are what are known as distal stimuli. A distal stimulus is something far away, and vision can do both of those. Hence this kind of flexible notion, right, that can be used for processing at lots and lots of different distances. Obviously, as I said, there are other modalities. The auditory modality, right, your sense of hearing, is also pretty versatile. When we hear, of course, what we're processing are sound waves. Sound waves are actually changes in pressure of the air around you. Some object makes a noise, right? The book falls off of the table. What happens is that basically it upsets the air around it, and those sound waves, changes in pressure in the air, reach your ear, and you then hear that as sound, a change in sound. So the changes in pressure that strike our eardrum are basically what allow us to hear. So those sound pressure waves here are illustrated by diagrammatically here by these visual angles here. And of course, all of that then ends up being processed in the auditory parts of the brain, which we call the auditory system. And this is just a diagram here of what that looks like. So someone's speaking, those sound waves reach your ear, they travel through the ear canal, and they stimulate a set of organs in the ear, which convert, just like in the visual modality, right? The retina converts light rays into electrical impulses. The inner ear here converts sound waves into electrical impulses. And those electrical impulses then traverse several circuits here and make their way into the cortex. Very similar to what happens in the visual modality. And in fact, the electrical signals that are traveling in this circuit are actually very similar to the visual ones. And it's even possible, it's been demonstrated it's possible if you go in and very carefully move those neurons around, it's possible actually to stimulate visual parts of the brain with sound waves, and likewise auditory parts of the brain with light rays. And people then experience, or these experiments that have been done in animals, the animals actually experience those different modalities as something completely different. Right? You're actually hearing light and you're basically seeing sound. The point is that the electrical signals are very similar in the two modalities. It just happens that the signals end up in different parts of the brain, which then convert them into the familiarity of the particular modality that we're talking about. There are actually people, neuropsychological patients, who have a disorder where they actually, there's some short circuits in these various pathways, and they actually do sense visually when they hear a sound. They'll actually have a visual sensation, visual perceptual sensation of the sound, and vice versa. They'll actually think they are hearing sounds associated with visual stimuli. This is a disorder called synesthesia. Very interesting. Brain disorder. All right. So the auditory stimuli in the auditory system is like the visual system also, not only in that they're both these electrical impulses, but they also, this system is capable of processing both proximal stimuli, so stimuli that are very nearby, right? So sound sources, right, as close as the eardrum itself, right? A mosquito inside your ear, everyone's had that sensation, right? So that's a very, very proximal stimulus. So the stimulus in that case is like literally on top of your eardrum. But you can also, of course, process sound that originates from stimuli very far away, or distant stimuli, right? So thunder from lightning miles away can cause a change in the auditory system. It could be perceived as a sound. Lots of other modalities, of course. The somatosensory modality is probably the next most important one for us as humans. And the interesting thing about these somatosensory modalities is it's almost completely devoted to processing proximal stimuli. And so our sense of touch, for example, is an example of a somatosensory modality. Of course, the way that works is some kind of stimulus touches your skin, it sends impulses on nerves, again, into different parts of the brain, part of the brain that's specialized for processing somatosensory information. For example, this is the touch of pencil. Another system that runs in parallel with our sense of touch is the pain system, of course. All of the organs of the body, all of the surface of the skin have pain receptors. There's temperature receptors, there's receptors for balance. Most of these are in the middle ear, close to the auditory system. And of course, all of those nerves feed into the brain. And in particular, the very top of the brain here is where the somatosensory parts of the brain are. Other modalities include the chemical senses. They're mostly proximal, right? Stimuli that are fairly close by. Very close. In the case of the sense of smell, most of the senses of smell are relatively close to us. Originating from something relatively close to us, but not always. All smell. And fire season here, when you fire even miles away, you can smell what's the remnants of the fire. So that would be one kind of counterexample for the proximal notion of smell. The source can be distant, but you actually sense it as happening in your nose. And like the other modalities, there's a similar kind of transduction of information, this process of converting a stimulus from the environment into an electrical impulse is called transduction. And the transducers for olfaction are in the top of the nose here. And we send signals then into the parts of the brain that are important for processing odors. And then lastly, taste. Taste and smell are actually closely related in many ways, but the different brain areas are responsible for processing. And taste is almost completely proximal, right? In fact, the stimuli have to be inside your mouth for taste to actually, in most cases, for taste to work. So really, the taste modality is very much devoted to proximal stimulation. All right, so let's get back to visual perception. Like I said, visual perception is arguably our most important sensory and perceptual system. Another reason that I spend a lot of time talking about this is it's also the modality of the system that we know the most about. There are way more people studying the visual system than any other of the modalities. And so we know a lot more about it. And of course, what's it for? It's for doing this, which we're all doing here. It's allowing you to see an image like this and to very quickly categorize and figure out what it is you're looking at. Although it's also quite obvious when you're processing visual information that the stimulus basically elicits a similar response. In other words, the process that there's a cat there. The stimuli don't always look the same. All right, so here we have a very, very different image of the cat, and yet all of you, I'm sure, were relatively quickly able to recognize what this object is. You can probably recognize this one as well. And that one. This presents to you some of the problems that the visual system has to solve, right? So probably within probably a second or so, all of you realize that you were looking at both a cat and a rabbit here. And yet, there are lots of similarities between these two images. How does our brain allow us to differentiate between this creature and this creature? And this one, right? I mean, again, another example of the same category, but a really different example of the same category. And of course, here's even a harder one, which we grew up on cat dog. Which is this, right? Another really good example of our visual perceptual abilities is our ability in lots of different categories to recognize similar objects, and in some cases dissimilar objects that belong to the same category. Right? So all of these are examples of houses. And probably all of you can rapidly recognize these, right? So our ability to do this task, right, to process visual information and to do, I call it in here, but to recognize the individual things that are in the image you're looking at, turns out to be what the visual system is evolved to do. It can also do this, of course. It can do more complex kinds of processing. This is what we would call scene processing. So all those previous images I was primarily showing you a single object, but here there are many different objects. And scene processing is even more difficult than processing individual objects. In part because there's just a lot more stuff, right? But also because there's confusing information. So probably all of you immediately recognize this as a body of water. But early computer programs that tried to do scene recognition would very easily be fooled by the image of the upside-down mountains in the lake here. And so, seemingly an easy task for us is actually quite a difficult task for computer programs. Of course, you can also recognize an object superimposed on a scene like this. So, yes, scene perception is basically an example of a more complex example of visual stimulus processing. And your ability to do it, right, to process a scene like this and to extract objects from it is really what the visual system has evolved to do. Our visual systems have evolved to allow us to process information from objects and scenes. What we're going to do now is we're going to spend most of our time talking about this aspect of what the visual system does. Because it really, in some real sense, being able to recognize individual objects is what really differentiates our visual system from lots of other types of perceptual processes. So yes, the topic areas are sometimes referred to as object recognition. How do we recognize all of the objects we're capable of recognizing, which number in the tens of thousands? Right? The human brain is capable of recognizing tens of thousands of different objects. I say tens of thousands because no one actually knows what the upper limit is. And certainly when you start talking about very, very similar objects, like all of those different houses that I showed you, and if you consider all of those different objects, then the number of objects that we can recognize starts pushing up into the billions. So it's a pretty amazing thing that we're able to do, and we're able to do it very efficiently and quickly. Again, this type of processing, you know, mostly happens in less than a second. Your brain is able to take the visual information striking the retina and convert it into a recognized object in less than a second in most cases. And in fact, arguably, this is what our visual systems are doing most of the time. Our visual systems are usually doing the task of object recognition. They might be doing some other things alongside it, things that enhance object recognition, but this is probably the major task that our visual systems are tasked with. Right? Is they're trying to recognize all of the objects in the world around us, all of the objects that we basically focus our visual system on. And of course, classic example here, right? Finding your car keys and picking them up involves this very, very precise object recognition. So you have to recognize these are your keys. And you have to be able to use that information, in fact, to help your motor system, the system that guides your hand, to the location where you pick them up. So it's a really complicated set of processes to go on. Let's try to kind of keep it simple right now in the beginning as we talk about what must be happening when we recognize objects. Oh, I guess I should say this before I move on, is that while we're going to talk about object recognition in the visual modality, one could argue that all of the modalities are actually optimized, or most of the modalities are optimized for doing object recognition. So it's possible to recognize objects that are auditory, right? You're doing that right now. In some real sense, the words that I'm speaking are auditory objects. You're recognizing these English words that I'm speaking, and you're obviously assembling them together into sentences and the basic concepts that I'm trying to transmit to you. There are also haptic or touch-based recognition. Perhaps a bit closer to the visual modality, right? So I've got this laser pointer here, and I put it in your hand, and you're able to move your fingers and hand across it. I'm not guessing exactly that it's a laser pointer, but you probably be able to get some pretty good information about the properties of the object from feeling it with haptic sense. Chemical senses also deal with objects, right? The smell of particular things that were familiar to us. In fact, we've organized our perceptual world as collections of objects within scenes. So there's a very similar sense in the olfactory modality of a scene, right? It's the smells of the environment you're in. Think about walking into the kitchen of a restaurant or into the mom's kitchen when she's cooking. All of the different smells that you can pick up on as you move through a scene like that. But like I said, visual object recognition is where we're going to spend most of our time. We process objects about specific information about specific objects. But what does this involve? What's involved in recognizing an object? Well, there's a number of different properties of objects, as it turns out, that are important in recognizing. Probably most important is their shape. And shape perception is basically our ability to recognize the shapes of the objects that we're looking at. The interesting thing about shape perception is that while in most cases we don't have much trouble recognizing the shapes of objects, there are cases where there could be problems. And Professor Midgley showed you some of these last time. She showed you some visual illusions as a way of thinking about how object recognition actually happens, understanding how we process information that is not actually what it appears to be, turns out to be important. And perceptual psychologists use illusions to better understand what the system is doing. For example, in the case of shape perception, here's one of the classic illusions that people have studied. This is called the Necker cube illusion. And looking at this, how many of you see this kind of front square here of the cube as a plot? I'll give away the answer. How many of you see this part here as the front of the cube? Now think about it the other way around. Think about this part back here as being the front. Can anyone get it to flip around? Yeah, so this is the reason this is called the Necker cube illusion, is that once you do that one time, you'll notice that the cube keeps switching back and forth on you. What's the front and what's the back? Right, so yes, these are the front edges for most people on first looking at it, but if you think about it for a while, you can actually get it to flip. I can't get it to flip from my angle. No, sorry. So when this becomes the front, it's higher up, right? So this is a higher up part of the cube. So what's going on there? What is going on in our brain that is telling us, giving us kind of conflicting information about where the front and where the back is? That's obviously an important clue to object recognition, recognizing this is a cube. Another important clue to object processing is the fact that objects rarely are stationary. In these idealized examples that I'm showing here on a flat screen, most of my objects have been stationary. But in the real world, objects tend to move around. And that, of course, both complicates, in some cases, but also enhances our ability to recognize objects. If objects are moving, in some cases, it becomes easier to process them. And this particular type of processing is called motion perception. Our ability to process motion turns out to be very, very good and very helpful in many cases. And the reason I'm telling you about these separately is that there's good reason to believe that actually the areas of your brain that process these two attributes of objects are different. Part of your brain that tells you about the shape of things is a different part of your brain than the one that tells you about whether an object is in motion. And that's a really key piece of information. Even though we kind of experience the world as unitary, right? So when you see an object, you see both its shape and its motion. That's an illusion of itself, that this is a unitary thing happening. Actually, different parts of your brain are doing those two different parts of the task. So when you experience motion, the brain areas that are doing that are different than the ones that are telling you what the shape of this object is. So there's an example of how important motion perception is. So that's about an 8-second little video there. What did you see? Maybe I'll get closer to this and see if what was in that image? Yep. Someone put a plate in the dishwasher. Who put the plate in the dishwasher? Anyone tell? The kids, yeah? How do you know that? Yeah, I think he was fairly taller than the dishwasher. Does anyone able to tell what race the kid was? No. He's not Caucasian. That's another good guess. Yeah, there's not much information, right? Because you always see from behind. Those are the, yes.
He's Asian.
Yeah, he's Asian. So he's Asian, and you don't ever really see his face, but there's important information, right? And even in the shape of the head and the coloration of the skin gives you this kind of insight into who it is you're actually looking at. Boy or girl? Boy. Yeah, so you extracted a ton of information from that tiny little low resolution video. I apologize that it's not high quality as it could be, but I think it's kind of good that it's not because it shows you just how important the motion part of this is, right? The moving of the parts of the image relative to each other tells you a huge amount of information about what's going on. And it also illustrates, I think, one of the points that Professor Mitchell made last time, this notion of top-down information, right? The idea that we all have stored in our brains lots of information about the world and how it works, and we use that information when we perceive the world. This idea that we just kind of passively take in information, right? It just kind of pours over us, and we process the information purely as a stimulus is wrong. Perception really is an active, constructive process. It involves, obviously, the one part, the incoming information, but also your previous experiences. Right? Experiences at the level of individual objects, but also the social experiences that you have. Right? The idea that loading a dishwasher is something that we're all familiar with. And it's something that maybe our children do at some point. This is why a child might be doing this. This might be a chore. Right? All of that information becomes available when you process this type of really simple perceptual information. All right. Another aspect of visual perception is that objects can be close by or far away. Right? So in this case, being able to judge if this car is close is obviously an important part of crossing the street. This particular ability that we have is what's called depth perception, our ability to process the relative positions of objects in the environment and their distance from us and from each other is an important part of what object recognition is doing. So, as I mentioned earlier, I showed you with the necklace, there's a lot of ambiguity in vision. Most of the examples I've been showing you are fairly unambiguous, but even in unambiguous circumstances, it turns out there's stuff that's not perfectly clear. Vision frequently recognizes objects under clear and obvious circumstances, like I've been showing you, right, like this. But also, probably equally often, objects or entire scenes present us with ambiguous information. What do I mean by this? Well, there's actually, many different things, actually. So here's an example of a somewhat ambiguous picture, partly due to lighting, impoverishment of lighting. Right? There's not a lot of light on this particular set of objects. And yet, your visual systems are probably able to process this information fairly well. It might be a little bit slower than if these subjects had been perfectly lit. But what can you tell me? Someone who got up close. What do you see here? Native American woman with a child on back, right? So yes, probably most of you can see that. And yet, if you look at the child, or what little bit of information we have about the child, we've got one arm, some really, very, very in the shadows features here. So a little bit of a nose and a cheekbone here, right? And I guess this could be hair, but it could be a fur coat as well. Very, very small amount of information here. It allows you to extract this kind of very precise information about what we're looking at. So we can recognize objects under non-optimal conditions. And many times, though, objects can actually play tricks on us. Right? And of course, these are the illusions that we talked about last time, which I showed you. Perceptual psychologists actually use illusions to better understand what the visual system is doing. Because illusions really kind of pit many different cues against each other and make perception not as easy as you might imagine. So here's a really good example of a particular kind of illusion that's important. And this is called a figure ground illusion. So how many people see a woman standing here? But how many people see a face as well? So there's a face here, we haven't seen it yet, right? There's a chin. The woman's kind of the nose of the face here, the eye. This tunnel here is an eye, and this is another eye. Everyone got that? Yeah. This is a figure ground illusion. It's called that because, in fact, if you're looking at it and you see this as a woman, she is the figure, and all of this other stuff is the background. But in fact, that illusion allows you to switch the figure in the ground, and the figure becomes the face. Right? And all of these other things really are the background for the face. There's another classic one. Maybe you've seen this one before, the old lady and the young lady. I'm having trouble seeing both pictures. So the young woman, this is her chin, and she's looking away from you. Here's her eyelashes here, you can see just a little bit of her nose. Here's her ear and earring. And the old woman, of course, this is the old woman's nose. This is her mouth, and her chin is down here. And she's wearing, some type of shawl. And of course, once it's been pointed out to you, one of the things that happens is this thing is kind of like the Necker cube, right? It keeps going back and forth like that. So these different visual cues, right? These different, basically, shape and depth perception cues are flipping back and forth because they're basically being contrasted with each other by your visual system. So one of the things that our visual system is doing is it's doing this task. It's trying to figure out what is the object and what is the background the object is on. Now, in some cases that's really simple, right? In a case like this, the background is black and uniform, and the figure is clearly something with texture and shading differences that allow you to see it as a three-dimensional object there. But this is what we mean by figure and ground. And it's kind of simplest form and easiest form. But in many cases, as we showed with some of those illusions, These two things are actually kind of pit against each other. And in this particular case here, of a woman sitting in front of her vanity, looking at her image, you're seeing her as a figure and her image as a figure. And the background is the mirror that she's looking at there. Of course, the other image, for all of you can see, is the skull, right? That is, here are the two eyes of the skull, here's the mouth, this is the outline of the skull. And again, that then becomes the figure, and the image of the woman is basically the background. Another type of illusion that's useful in understanding object recognition are these so-called impossible figures, which I'm sure all of you have seen these before. Right, where if you look out here at the end, it looks like three different tubes, but if you come down here, it's very hard to figure out where is this tube's physical origin. Right? It seems to go against the laws of physics here. That objects like this, obviously, they should be doing what this is doing, not this. Some examples of impossible figures that cause our visual systems to have particular trouble. And of course, Escher is the most famous of the impossible figure artists, right? Lots and lots of pen drawings, basically pitting all of these different cues that we're talking about, these visual perceptual cues against each other, making it very unsettling to look at these images. I love this one. So the water flowing up is what's flowing down. So one of the problems our visual systems have is that we actually only see two dimensions. Your retina is flat, right? Your retina is like this screen. It does not directly perceive the third dimension of depth. So depth perception is something that the brain actually computes. It has to derive it from a variety of features. So when you see a cube like this, for example, if I present you with a cube in front of you, it looks like a cube. If I present it at the right angle, the image that actually displays on your retina is a flat image that's not cube-like. A cube presented in front of you with it ever so slightly tilted presents this image on your retina. So how do you extract the cubeness from that? How does your perceptual system actually derive the fact that it's looking at a cube? It's tricky how it does it. It uses a variety of so-called depth perception cues to do it. The first of these is something called linear perspective. These are actually, the cues I'm going to tell you about now are all ones that artists have discovered kind of on their own through using various artistic media. Right? And in fact, they didn't always know that. So linear perspective is one of those cues. And of course, this is the classic example, is the parallel lines of a railroad track as they recede in the distance get closer together. Right? So two parallel lines as they recede in depth get closer together. Your brain uses that cue to give you depth. Right? The reason you see this track as being receding into the distance is because your brain extracts from that this information about what parallel lines do when we look at it. Artists, as I said, use that information in drawings and in paintings to basically perceive depth. However, the ancient Egyptians, the artists from ancient Egypt, actually were not able to use that. They did not know about linear perspective, and so all of their drawings were of this kind of flat nature. It wasn't until much later, and obviously clearly illustrated by Renaissance painters like Da Vinci, painting the Last Supper here, where linear perspective is used throughout this painting, right? You can see here all of these different lines all emanating from Jesus in the center of the painting. Other illusions are illusions that give us information about depth. So illusions of motion can be used for that. You probably see this as there's some movement going on there when we're actually disappearing. You see that moving? There's no movement going on. That's just depth perception. And motion center parts of your brain are actually seeing motion happening. Perception is really important, as I mentioned. Object perception, I'll just show you one more slide here. So, does anyone see a person in this image? chance to figure out what it is that you're actually doing. All right, so this is probably a good place to stop. Is the question of object recognition. The main task that your visual systems are trying to accomplish is to recognize all of the potential objects in the world around you. So, examples of objects are things like myself up here, you're looking at the screen, obviously the clock. Any individual thing within a visual scene, a scene of course being a collection of objects that your visual system is able to take in at any one point in time. All right, so as we mentioned, there are a number of challenges, if you will, to perceiving objects in the world. One of the big challenges is the fact that the retina, basically a photographic surface at the back of your eye, that part of the eye that transduces the light rays that come into your eye and convert them into an electrical signal that then goes into the brain. That retina, two of them, right, one in each eye, is flat. Which means that just like a camera, it's really only capable of registering a two-dimensional image. Right, so things that have an X and a Y coordinate. What it can't pick up directly anyway is the third dimension of space. Right? The thing that gives objects in the real world their third dimension, the Z direction, if you will, the depth of objects. In order to perceive depth, you actually have to manufacture that. So it's part of the perceptual process. Your brain is actually inventing, if you will, or is inferring what that third dimension is. Now that might seem strange to you, right? Because we have a very powerful sense that the world is three-dimensional, right? It's not a flat the screen up here. And yet we can demonstrate that even in a two-dimensional world, you can still see three dimensions, right? So all of you can see that this is actually a cube. When in fact, the image that's projected onto your retina, if you're looking at a cube as we are here straight on, what you're actually projecting onto your retina is something that has a shape like this, right? It's basically the corners of this object. So the question is, how does your visual system do this? How does it recover? How does it manufacture, as I said, that third dimension? And the answer is it uses a number of different cues, a number of different ways of taking the information from those flat images on your retinas to manufacture the third dimension. Now, some of you probably know that one of the cues it uses is the fact that we have two eyes. Actually, I don't have one retina, but we have two retinas. And that the images that project onto those two retinas are actually slightly different. And your brain actually uses that information to help construct the third dimension. Now, obviously, that's not the only cue, because probably in a class of this size, there's probably some people who don't see really well out of one of their two eyes. And yet, nevertheless, you still see the world in three dimensions. Right? So it doesn't absolutely require that you have two perfectly working eyes to see the world in three dimensions. In fact, all of us can close one eye, and while the image that we look at isn't quite as good, in fact, we lose information from one side of the world to some degree. Nevertheless, you still see three dimensions. So there must be other cues, right? There must be other information available to that flat retina that helps us see that third dimension. So let's talk about some of the cues that the visual system uses in order how do we see this third dimension? Just like here. There are these depth perception keys. One of them is binocularity, right? That's the fact that what I just mentioned. You have two eyes, binocular vision, versus the fact that most people are able to use two eyes to help them see depth. But there are also other cues that are available that are what we sometimes call monocular cues. In other words, you can see these cues with a single eye. And with one eye open, you can still perceive depth. And one of those cues is one that I mentioned last time. It's called the rear refractive. And that is the cue seen here, the rear refractive illusion. Right? The idea that if two parallel lines appear as they recede in distance and get closer together, our brain sees that as a cue to distance, as a cue to depth. We're able to use that then to actually recover third-dimensional information. And as I mentioned, artists have kind of discovered this particular aspect of depth perception. Right? They exploit it in paintings. Early artists back in the Renaissance, in particular, really used this. Right? So I mentioned that Egyptian artists over 2,000 years ago, almost 3,000 years ago, did not know about linear perspective. But certainly by the time of the Renaissance, and painters like Da Vinci, they were well aware of the linear perspective cues. Here, there are tons of these cues that are being used to this particular painting, a third-dimensional appearance. So, these kinds of illusions, if you will, right? So Da Vinci is using an illusion of sorts, right? He's using a trick. He's tricking our visual systems into thinking that when we look at a painting, that we're seeing depth, when in fact there is no real depth there. It's a flat image on canvas. So other illusions help us figure out what some of the other cues that the visual system might use in helping us see objects. And as I mentioned, one of those is our ability to see motion. Right? So in illusions like this, even though there is no motion here, your visual system probably is seeing some kind of waviness or quiveriness of the image up here. And that's because the way this image was created, in fact, sets the motion cues, parts of the brain that detect motion, sets them into actually working on the image. And this is like a really powerful motion type of illusion that the motion parts of the brain are working on. So yes, motion perception is a really important cue in object recognition. And it's really important cue in depth perception as well. Right? The fact that we're able to move around in the world and that objects are moving in the world helps us see depth. There's a really powerful understanding over here. I'm sure all of you have seen it. It's been used very frequently now on television and in YouTube videos where you're looking at a flat image, but it appears as though you're moving with respect to the objects, perhaps the scene you're looking at. And as soon as that starts to happen, it's almost as though a three-dimensional component of the image really starts to jump out at you. It's almost like you're looking at a stereo image. Right? Stereo images are another way that we can fool the visual system into seeing three dimensions, right? By putting two slightly different objects onto the two retinas, right? So they do this in three-dimensional movies, basically by superimposing two views of an object from slightly different perspectives. Gives this very powerful illusion of depth. And I think this is the slide we ended on last time, is that it's very difficult to see the outline of the person here. I mean, once you've seen it, you know where the person is, so like here. But as soon as you start to move one object relative to the background, you get this very powerful sense of object recognition, right? That that's a person. So yes, objects, there are these cues. There are depth cues, there's motion cues, there turn out to be lots of other cues. But the main thing that you're trying to do, one of the main things you're trying to do when you're recognizing objects is, first and foremost, is you're trying to separate the object that you're looking at, what I'm calling here the figure, from the background that it's superimposed on. As that previous slide showed, right? Basically trying to separate the object from its background is a really important part of seeing the object. In many cases, that's a really easy thing to do, right? If the background is uniformly colored in the foreground. The object is colored differently and shaded differently. It's really easy to do that. But in many cases, it's not so easy to do that. So what we're doing when we're doing this figure ground perception is we're trying to segregate figure from the ground. In other words, we're trying to separate these two things from each other to see them as distinct. And once you're able to do that, of course, you can then actually process the figure or the object itself. Of course, our big question is, how do we do this? So here's a picture of a building, and obviously, probably, again, within less than half a second, all you recognize this is a building on a kind of white gray background. So how is our visual system actually recognizing this object? Well, one thing that we think is going on very early in the process of recognizing objects. So in other words, as soon as this information starts to get into the visual parts of your brain, what your brain probably is doing is that it is doing this. It's doing something called edge detection. It's detecting all of the corners and straight lines of the objects that you're looking at. This seems to be a first step that's involved in recognizing that that object is actually a building. So yes, processing the edges is an early aspect of object recognition. So really, this detecting those edges is something that happens before you actually realize what it is you're looking at, before you realize it's a building. Now, of course, again, it's important to remind you that we don't have conscious awareness of this, what's going on when this happens. This is happening so quickly in the visual parts of the brain that you're not really even aware you're doing this. And this is happening literally. It takes half a second to recognize that building. Detecting the edges is probably happening in the first tenth of a second. Right? So long before you've actually gotten to the point where you say, oh yeah, that's a building. After you've detected the edges, what seems to be happening is that we start to become aware of the various shapes that make up an object. So in the case of the building, we start to recognize the kind of angular shapes that are coming together. And part of that is recognizing where there are discontinuities in the shape. In other words, where things change. So when two angles come together, two flat surfaces come together and form a corner. Visual system seems to be, very soon after we detect the edges, we start to do this. We start to see this type of feature, or process this type of feature. We start to recognize the shapes that go into making up the object. This is kind of an example of an illusion that shows the visual system trying to do this, right? So one of the powerful things about this illusion, of course, is that really what you're looking at here is four passionate. But I'm sure most of you have a very powerful illusion that there's also a square here. Right? Even though there is no line that connects these two things. So this is an attempt by your visual system to basically define that figure, right? That shape that's in the middle here. And you're doing that first by finding the edges, right, which are here, here. And then the shape perception system starts to superimpose on those particular edges this shape, the shape of a square. Another, of course, important part of recognizing objects is their color. Perceiving color is something that the visual system is doing kind of alongside of these other things. Alongside edge detection and shape perception, we're also perceiving color. So I'd like for all of you to stare at this. Seeing this illusion of what's going to happen. It stares at this black dot here for a little bit, and then I'm going to change the image. And this will tell us something about the systems in your brain that are involved in actually perceiving color. So, what did you see? What happened when I changed the light back there?
Red and blue.
Yeah, most of you saw the regular black, right? So what does this tell us? What's going on? Why is the color perceptual system doing this? Well, the one that seems to best explain this particular illusion, what's called an opponent illusion. Basically, what's happening is I'm showing you those green and black, and obviously the reverse colors in some sense, right? What are called the complementary colors. And what happens is that we're saturating the receptors in the visual system that process those colors, and so when you take those colors away, the system compensates by going to the other extreme. And people have argued, scientists have argued, that this suggests that there are actually two opponent systems in the color detection system. The red-green system and the yellow-blue system. And that basically these two are in constant, the two colors on the extremes of these are in constant conflict with each other for the same spatial location. Yes?
Is that kind of what happening with people that
Yeah, so what's going on with that? So good one we're talking about here. So people that are colorblind, actually there are some glasses, I think that you're red, I think it's red and green colorblind. There's some glasses colorblind. Yes, it is. It's part of what's going on, and it's compensating for the opponents. What's going on in people that are red and green colorblind is that the receptors that code for red and green are actually, you don't have as many of those. And the ones that you do have seem to be tuned to one extreme. People that are completely colorblind actually are missing all of these receptors, and they rely on a different type of receptor that actually see shades of gray. And red-green colorblind, yes, we can help correct that, which with glasses that basically compensate for the imbalance of this type of processes within the visual system. The important thing to tell you here is that probably all of you, many of you have, of course, we've learned about rods and cones, right? And you know the cones are the receptors that pick up all color, three of them. That are tuned for different wavelengths of light, and they roughly allow for us to see the three most important colors, red, green, and blue colors that we're able to see. So, knowing that, you might be wondering, well, what is this? What does this have to do with that? And the answer is that the trichromatic nature of the receptors, the three color receptors of the eye, by the time the color information gets into the brain, right, the parts of the brain that actually process color information, the system seems to instead have this opponency type of relationship. So the neurons in the visual parts of the brain that process color, they certainly care about the red, the blue, and the green information coming in, but they organize that information into these two kinds of opponent systems. And that's what accounts for the illusion that we just looked at. All right. So we've talked about some of the different processes that are involved in dealing with visual information that allow us to recognize objects. An important thing here in talking about this collection of things that are going on is the idea that, this is something I've been starting to harp on, I'll continue to harp on last semester, is that the processing that's going on is in part hierarchical. Right? And by what I mean here, this is a nice illustration of hierarchy. Before you can do shape perception, you have to have detected the edges. Right? Obviously, before you can detect the edges, you have to have excited those rods and cones in the retina. So the rods and cones get activated, they send the information into the visual parts of the brain. We then detect the edges of the objects we're looking at. We then start to detect the shapes based upon the edges. And then we also detect the color, and we're going to detect many other things. But the point is that one has to happen for the other. And this is hierarchical. Right? You go from what I'll call low-level processes, which would be what happens in the retina. Light rays actually activate those first cells in the retina. And then the signal gets transmitted into the brain, and then you start to activate edges. And after the edges are activated, You then start to activate the shape perception areas of the brain and the color perception areas of the brain, and you see eventually the actual object representations of the cells in the brain. So then we've got this notion of hierarchiality, right? The idea that you're going from low to intermediate to higher levels as you recognize an object. And of course, the other important part of that is that this is obviously taking time. So yes, in that sense, object recognition is somewhat sequential, right? There's steps involved. You have to do this, and then you have to do that, and then you have to do that. And only after you've done all of these steps do you actually recognize the object. However, I don't want to give you the impression that everything that's happening is sequential. One thing has to happen before the next, and so on. We also know that one of the really important tricks our brains are able to play is that they can process information in parallel. And a good example of that is here. So while I said that you have to detect the edges before you can detect the shape, notice I didn't say that you have to detect the shape before you can detect the color. Shape perception and color perception likely are happening at the same time. That's what we mean by parallel processing. Parallel means that basically two different parts of the brain can be working to solve different kinds of problems at the same time. And in fact, in the case of the visual system, I'm grossly oversimplifying what's going on. There are probably as many as 35 different brain regions simultaneously working on figuring out what the object is doing. I'm going to go into what all of the different processes are. I actually don't even know what all of those objects are. But there's evidence from people that study visual perception that there are many, many different brain regions doing many different computations to help us figure out what it is that we're looking at. And this is the notion of parallel processing in the brain. And it's a trick our brain has basically acquired through the course of evolution, right? The fact that the individual units that process information in the brain, the neurons, that are doing all this, are actually pretty slow. Neurons process information, well, certainly on our time scale, neurons are fast, right? A neuron can fire an action potential in probably a few milliseconds, a few thousandths of a second. But when you start to think about how many action potentials you have to do something even as simple as recognizing a primitive object, we're talking about millions of neurons. Right? Millions of neurons are involved in that process. And if they all had to do processing sequentially, right? So one neuron had to process information and it sends it to the next one, and then the next one. By the time you got to 1 million neurons, each of them takes 1 millisecond. We're talking about 1 million milliseconds. That's actually 10 seconds. All right, so obviously that's not what's going on. It doesn't take us 10 seconds to recognize objects. So what that means is that many of those neurons that are involved, many of those billion neurons that are involved in recognizing an object are actually doing things at the same time. They're processing information in parallel. This computation, we're doing color perception is going on while the computation for shape recognition is going on. In fact, it's probably even more complicated than that. It's probably not just a matter of them being parallel. Some processes, like for example, I said you have to do edge detection before you do shape perception. It's probably the case that you start to do shape perception even before edge detection is finished. This is what we call cascaded processing. Right? One process starts to work on something, and if it starts to get the problem solved, right? Maybe the problem isn't completely solved yet. Maybe not all the edges have been detected. But maybe enough of them have been detected that the shape system can start to work on the problem too. Right? So you've got the edge detection thing working, there's the shape detection thing, you're waiting for it, and as the edge detection starts to send information out, saying, yeah, I've got some idea about what the edges are, the shape system can start to say, oh, well, maybe some of those shapes are rectangles, and maybe there are rectangles in these locations. This is what we call cascaded processing, kind of like what happens right with a waterfall. One waterfall falls down into a pool, and then water from that pool falls down to the next pool. All the water is continuously flowing. Right? It's not that one pool has to completely be filled up before it can be emptied into the next one. mean by cascade? So, yeah.
When you say hierarchical, it doesn't mean exactly step by step, but the extension is like just like start first, and everything else will happen before it finishes. So like it's not at the same time, but it's pretty much like it doesn't have to be like 1 and then 2 and then 3 or like.
Exactly. Yeah, it doesn't have to be 1, then 2, then 3. It can be 1, and now let's start getting 2 going. 1's still finishing what it's doing, but 2 can get started now, and as 2 gets going, it can pass a little information on to 3, and then 3. Okay, so it's hierarchical, cascadedly hierarchical is the way this terminology works. So you can see the brain is a complicated place, right? It's doing some things, at least partially sequentially and hierarchically. It's doing other things completely in parallel. And eventually all of this information comes together, allowing you to actually recognize what the object is. And of course, the important underlying point here is that we're not aware of any of that. Right? All of this is going on outside of our awareness. What are we aware of? Well, you're aware that you've seen an object. And as we move into talking about attention, it's going to be obvious why this is the case. Why is it that we can't kind of consciously introspect about the low-level things that are going on in perception? And the answer is, if you were doing that all the time, think about how distracted you would be. Right? I mean, if you were sitting there thinking about all of these edges and shapes and things that go into making up an object, and were concentrating on that, you wouldn't have any attention left over to think about the things that are important. Like, what is that object? And, is it something I need to be aware of? And is it something I need to interact with? Whatever. Right? And what that, of course, tells us is that when we're processing information, we have a limited ability. We can only do so much, right, before we get overloaded. And so, if you think that not being able to focus on these low-level things is kind of unfortunate, think again. Evolution has probably spared us from paying attention to things that really don't have any real impact on how we need to interact with the world. All right. Yes, I already said this, but just to reiterate, all of this implies that while it seems as though we recognize objects instantaneously, in fact, it does take time. As I said, on average, for most objects that are fairly clear, fairly well distinguished from their background, object recognition happens in something around a half a second, somewhere between half a second and a second. That's time. It seems like a short time, but nevertheless, it's time, certainly when compared to how fast neurons are doing things in a range of milliseconds to tens of milliseconds. All right, so let's just quickly, I'll show you one brain here to give you an idea of where is all of this happening. So I've already told you that the back of the brain here, this gray area here, is where the visual system first receives information. So the eyes are up here, right? The eyes are up here. And there's the optic nerve that takes the information all the way back, and eventually that information arrives in the occipital lobe. It takes about a little bit less than a tenth of a second for the light rays to excite your retina and for that information to make its way back. So the first tenth of a second, your visual system isn't starting to figure out anything yet about the objects that it's looking at. It's roughly from a tenth of a second to 5/10 of a second when things start to happen. And the reason they start to happen is that the information, as it arrives in the neurons here, these neurons do some work on the information. They probably start to detect the edges. Right? So the edges are being detected right back here, kind of the first step in object recognition. And then the information starts getting passed along to new neurons. And surprisingly, right, the information started out up here in your eye, it goes all the way back here, and then it starts slowly working its way through different brain regions back towards the front, where the eyes were to begin with. A strange set of circumstances that probably an accidental set of circumstances that this is how it happens. All right, so yes, you do edge detection, then the information is passed along, we're doing shape perception, color perception up here, and then eventually the final product of recognizing the object is happening down here, actually in the very kind of lower front part of the temporal lobe. This is the temporal lobe. So we know this. This is the pathway that information flows through as we recognize objects. How do we know that? Well, because there's been lots of fMRI studies done where we have basically put people in various different kinds of experiments and we very carefully manipulated what kinds of objects, what kinds of visual information we're presenting to them. And we can look at the brain areas that get greater blood flow during those different kinds of experiments. And so we can kind of trace, if you will, the pathway of activity through the parts of the brain that are important for recognizing objects. Yeah, so it's certainly edge and shape. Color I kind of got off to the side here, because it's probably going on in parallel with shape perception. But it's a different brain area, right? So color perception is happening up here, shape perception is happening down here. And I haven't drawn it on here, but motion perception is happening up here, and it's probably happening simultaneously with shape and color perception as well. So that's the parallel notion. The idea of parallel processing is that shape, color, and motion perception are all kind of happening simultaneously once edge detection is finished. So once edge detection is gotten far enough along. Edge detection is certainly happening in the occipital lobe. There's kind of a boundary between the occipital and temporal lobe where shape perception seems to be happening. So some of it's probably still happening in the occipital, but some of it is also in the temporal lobe as well. So a good question, one that it depends on what aspect of shape perception you're talking about. Yeah, one of the things obviously I've done here is I'm simplifying to get a lecture and a half to talk about this. This is an area of research where there's incredibly complex things going on with lots of different experiments. There are many, many different areas involved here. I'm glossing over a lot of what we know about how the system works. This is the basic idea. And this basic pathway here, what is sometimes called the ventral means lower down in the brain, is very important for object recognition. You might notice there's another arrow here, though. So after edge detection, information also passed up to these areas in the brain up here, higher areas. It's called the dorsal stream. So these suppose the dorsal stream is good. If the ventral stream is recognizing the object, what's the dorsal stream doing with information from the visual system? Yes? There is some element of telling the brain what to do here. Yeah? Yes? Exactly. Yes. So location, right? I mean, objects are placed in space, right? Particularly in a scene where there's lots of objects. Where things are relative to other things is one of the really important things that's happening in this pathway. And this is the so-called where pathway. The dorsal stream is the where pathway, and the ventral stream is sometimes referred to as the what pathway. So what is it I'm looking at? What is the object I'm looking at? And where is the object that I'm looking at? Where in the visual world? So these are two very important findings that came out of lots of different research using lots of different techniques. Now it's fairly well established that this is some of what your brain is doing when it's processing the visual world. All right. So obviously there's more to object recognition. There is a component that we haven't even talked about yet, which underlies the recognition part of this, right? The fact that when we say you recognize something, that means that you're identifying that this is something that is familiar to you. You've either seen it before, or you've seen something like it before. And that implies that memory is involved. Right? You've stored information somewhere that allows you to say, oh yeah, that's something I recognize. That was a building. Or that was that little purple figure I showed you, which is called a greeble, by the way. Greebles were invented by people that study object perception. They wanted little objects that people would recognize as objects, but didn't have any preconceived notion of what they were. Right? So in other words, you didn't have a memory representation for these. Or some kind of precise one. So yes, memory is obviously a really important part of object recognition. And of course, one of the things we know you're doing when you recognize objects is you're matching a representation coming from input, right, from the perceptual screen, like that ventral screen we showed. So you're forming various neural electrical representations of what it is that you saw. And remember, even though I've drawn pictures on top of the brain there, what's actually going on in the brain are electrical patterns that represent those objects. Your brain has converted the actual object into an electrical pattern in the brain. So yes, when you see something like a lamp like this, you might imagine that previous things that you've seen that are kind of like it, maybe the same color and the same shape, might be involved in the recognition process. And so when you're seeing one of these greebles and you're trying to figure out what it is, it might partially activate many of these things that you've seen before that have a similar shape. Now, of course, none of these are actually that object. So eventually, you're going to say, yeah, I reckon that's an object, but I don't know what it is. So in a sense, you're not doing object recognition here. Your brain is registering the shape, but you're not recognizing it as something familiar. You've never seen this object before. Now, of course, once you have seen one of these objects, one of the things your brain can do, and we'll get to this when we talk about memory, is you obviously can form a new memory representation for that object. And so like 2 weeks from now, if I show you one of these, your brain will say, oh yeah, that's one of those greeble things Professor Holcomb showed us 2 weeks ago. And you'll do that because your brain will have, if you will, memorized, will have stored a representation for that object. But yes, under normal circumstances, if you're seeing this object for the very first time, your object recognition system is going to say, hey, I didn't find anything that looks like that. So that's not an object that I know. I don't recognize that object. However, if this was the target, right, this is the thing that I showed you. Obviously, you have seen green balloons before, and stored somewhere in your brain is a representation, a memory for a green balloon, and this would allow you to recognize that object. And probably be able to do other things then, right? But once you recognize an object, I mean, object recognition just isn't the end of the road. It is part of, obviously, other things that happen. So, for example, if I put you in an experiment, I say, name that object. Once you match that object to a memory representation, you then can activate the language system that allows you to actually name what that object is. Clearly, you couldn't do that here. You've seen this for the first time. You might be able to describe the shape, but you wouldn't be able to come up with a name for it. And once the representation is matched to some kind of stored memory, you can get this kind of activity that allows you to say, I recognize it. So that's what we mean by object recognition, right? Object recognition is basically the whole point of that perceptual operation of taking all of that visual information, processing the shape, and its motion, and its color, and all that. The whole reason for that is so that you can activate some type of memory for either that object or some similar object, and allow the brain then to do something with that information. Right? To use it for some other task that you have been presented with. So, in thinking about that, I'm thinking about how these processes actually work, the processes involved in doing the recognition. Once you've activated the perceptual information, how do you then actually do the recognition? How do you match up that green balloon that you're seeing to a previous green balloon you've seen in the past that allows you to say, yes, I recognize that as a green balloon? How does that work? Well, cognitive psychologists have been thinking about this for a long time. This is one of the early areas that cognitive psychologists identified as being an important part of cognition. Object recognition, recognizing things in the world around us. And one of the first theories that cognitive psychologists came up with to explain how it is that we do that, and what they refer to as template matching. And this is kind of the simplest notion. Probably, well, it's the one that was kind of inherent in what I showed you in the previous slide, right? When I showed you back here, what was going on. This is what we call a template matching kind of theory. So what is template matching? Well, basically what template matching implies is that the object to be recognized, in other words, that perceptual representation that the visual system serves up, if you will, the memory system, is compared to some kind of template, some type of representation you've stored in your memory from previous experience, previous balloons that you've seen, perhaps previous green balloons. So inherent in that notion, right, were those double arrows that I showed you, right? It's comparing those two things. See, is this the same thing? Am I looking at, is what I'm looking at the same thing that I remember having seen before? So yes, so I'm Homer here, looking at an apple, and he's trying to figure out what it is he's looking at here, right? He just picks this thing up and he holds it in front of his face, and he now needs to recognize that object. Of course, we know Homer likes donuts, so perhaps the first thing that his memory system serves up, if he's looking at a round object like this that could be edible and you can hold in your hand, is perhaps a donut, right? Obviously, this is not a donut, so you're not going to find a match there. But as he searches through his other memories for similar objects, he might find stored there something that looks more like the actual object. And in this case, as indicated here by the red circle, this would basically be what happens when you actually recognize it. When you find a match, you recognize it. So think about it. What this implies is that you've stored in your minds examples of all of the objects that you know. And then what happens when you recognize objects is that you form some kind of perceptual representation, and you then match that to all of those possible objects until you find a match. This is what template theory proposes. There's a match, and the object is recognized. Now, some of you probably already thought about what some of the problems might be with this. Give me an example of what you think some problems for a theory like this, what they have to overcome. Yes. Yeah, so lots of things can have very similar shapes, right? So in fact, the donut and apple example is kind of close to that idea, right? But they have kind of similar shapes. How do you know? How do you distinguish between two different things? I mean, you have to have a pretty precise representation to do that, right? Okay. Sure, yeah, so the Tide Pod craze that come around in Mount Palmville, right? The kids were mistaking Tide Pods for candy because they have similar shapes and colors. Yeah, so it's kind of an example. Yes? Yes.
So like if you say that information to set a lot of the figure out that was happening.
Yeah, so what would template theory say about that? Right? Yeah, so there's lots of different kinds of apples, right? Lots of different colors, lots of different sizes, cartoon apples, there's real apples. How do you, if you have a template, how do you resolve that? Well, you could have a template for all of them, right? The idea would be you just have to have lots of templates. But what does that suggest, right? I mean, as I said, you probably recognize tens of thousands of different objects. And each object has lots of different possible ways that you look. Does that mean you have to store all of those? Template theory would say that, right? If it already has a system, it's a good match, right? It can tell the difference between things that are slightly different. Then yes, you would imagine template theory has trouble with this kind of explosion, if you will, of the amount of information you have to store in memory. You have to store vast amounts of information about all the different objects, you know, and all the different possible ways they could look. In fact, we didn't even go into the fact that an apple, depending on what orientation it's on, looks very different. I think I have some slides. So yes, this is what we refer to as the canonical version of an apple. Right? The standard view, if you ask someone to draw an apple and draw its colors, is probably what most people would do. But of course, yes, there's no apples, which is perfect. And there are apples that have been cut in half. Right? And all of these are different shapes. And a quick perusal, right, through various databases of apples. And we're not even beginning to touch on how many different ones you're all able to recognize. So template theory has this problem, right? There's an explosion of information that can be stored. You can imagine that our visual systems have to have pretty much exact replica of what it is it's trying to recognize in order to match the template to the object. So one theory that's come up with a way to kind of get around this problem, get around the problem of there just being, you'd have to have too big of a memory in order to do this. There's something called prototype theory. So prototype theory is like template theory. It imagines that there's a comparison process that goes on during recognition. Perceptual representations compared to some kind of memory process. But this system tries to limit how much information you have to store by assuming that what we actually store is a few abstract prototypes of common objects. So in other words, rather than having to store 10,000 different views of an apple with different colors and different orientations and bites taken out of them and so on, that instead what you do is you store a representation of an apple that's a little looser in terms of its definition. And it's not exactly the same as what you're looking at. Now, how does that work? So how could you possibly then do this matching process? This is kind of a very loose kind of definition. And the answer is that in this theory, we imagine that there's an intermediate step. So you form a mental representation of what you're looking at. You've got the stored prototype of the category. And what you do is you do something like morphing. Right? Everyone knows what morphing is. We've seen tons of examples of this on the internet where you morph from one person to the next. You do this very smooth transition between objects or people or whatever. Prototype theory imagines that there's something like a morphing process. And you morph the mental representation that you have stored to see if you can make it fit the object that you're perceiving. And as long as you don't have to do too many steps to go from stored representation to the actual object, then you probably have a match. So you can see this theory is more of a probabilistic thing. Well, your brain is telling you that that's probably an apple. And the closer it is to matching, the actual prototype, of course, the more common it can be, probably the faster you can be at identifying. And then the more it deviates from the prototype, the longer it's going to take you, and probably the less sure you're going to be that what you're looking at is actually an apple. Well, that would be some kind of morphing process there. One problem, though, is how to constrain this morphing process, right? I mean, at some level, right? I mean, you might only need a single representation for everything if you can allow the morphing process to be extremely powerful. But of course, then you don't get any ability to actually recognize different objects. So you have to come up with ways to do this. You have to come up with ways of constraining the morphing process so when you're morphing the object into the mental representation you have, that you don't actually mistake an apple for an orange. So another very different approach to solving the problem of the recognition process is a kind of cognitive model called feature matching. So here the idea is you don't try to match the entire object to some stored mental representation. Instead, what you do is that part of what happens during perception is you actually take apart objects. You take apart objects into their features. So for example, if you've got a picture, you're looking at a robin, bird, that what you're actually doing when you recognize that bird is that you basically break it down into all the different features that make up that particular bird. So for example, there's a somewhere stored in your brain is some type of representation for a wing, for an eye, for a beak, for the orange crest of the robin, and you can imagine more and more of that, obviously, feet, and many, many other features that make up a robin. So in this theory, the idea is that as you basically take all that information from the edge detection and shape recognition and all that, what you're actually doing is, in part, is you're coming up with a bunch of features that actually make up the object. And that object recognition then is a matter of essentially matching the features that your visual system has suggested are there with some kind of stored list of features that actually make up that object. So in the case of a robin, it would be, yes, a list of, it has wings, it has eyes, it has a beak, it has an orange crest, it has skinny legs, on and on. And then if you get enough feature overlap between what the perceptual system is telling you is there and with your stored information about what that object, what features that object actually has, that is what happens when you say, yes, I recognize that object. Now, the big advantage of this kind of theory is that it doesn't require that you store nearly as much information, right? All you have to store is basically some information about the various important features that make up the various objects that you can recognize. It solves the problem of looking at an object from different orientations, right? Because again, the features are what are important. Might be a slightly different list of features from a different orientation of an object, but nevertheless, there's going to be enough overlap in the features that allow you to recognize it. And if the orientation is weird enough, right, you can't actually extract the features, this is what happens when you don't recognize something. So, what is that? There's something familiar about it, but I don't know what it is. I think you're looking at it from a very strange and unusual perspective. All right, so that's all I really want to talk to you about, object perception. I do now want to move on to talking about, we've got about 15 minutes left to talk about attention. What is attention? A layperson's definition of what that word means. What does it mean when someone uses this word in the real world? Okay. Focus. Yeah, focus comes to mind, right? Yeah. Conscious perception of information around us. Yeah, that's good. Engage, right? That's what the term engage in an activity. Obviously, that's important as well. So these are all common notions. When do people use this word in the real world? Did your teachers ever use it when you were in elementary school? Parents probably did as well. Pay attention, right? It's a really common use of the word. So yeah, all of these things kind of describe something very familiar to all of us. And yeah, there's not a completely consistent definition. So what is it? Well, here's a definition that I like. So yes, we all know what it is. It's such a common word in our language. It's the taking possession by the mind in clear and vivid form of one out of what seems several simultaneously possible objects or trains of thought. So one of the things you notice here is that several simultaneously possible objects, right? So attention implies this aspect of processing, which is that there are lots of possibilities of things to pay attention to at any point in time. And we have to decide which of those things are the most important to us right now. Right? When your teacher tells you to pay attention, she or he is telling you that basically that, the important thing for you to be taking in right now is what I'm saying or what I'm demonstrating, not looking out the window. So the interesting thing about this particular quote is it's actually made by William James, right, that early American cognitive psychologist in the year 1890 when he wrote his first textbook on psychology. So people have been thinking about this area for a long time. Right? They've been thinking about the concept of attention and how it relates to cognitive psychology for well over 100 years. So when I think about attention, when we think about attention from a psychological perspective, there's three different elements of attention that I think are important. The first of them is what we call orienting. The second is what we'll call searching. And the last is what I'll call filtering. These three different aspects of attention, right? This implies that attention really isn't just a single thing, but rather it's kind of a collection of different things that the mind is capable of doing. So let's talk about all of these. Let's start with orienting. So what is orienting? What does it mean when we say be oriented to something? Well, the simplest form of it is what happens here. It's called the orienting reflex. Many of you know about the orienting reflex. It's a rapid adjusting of the sense organs to biologically important events. That's the definition of it. In reality, if I were to do this, right, I can press the speaker here, it would play a very loud sound. So I won't do that. But that's a really good example of orienting, right? Really loud, intrusive stimuli tend to cause a reaction. And the reaction that we have is what we call the orienting reflex. It's basically biologically, environmentally, what I'm looking for here, genetically determined predisposition, right? This is something you don't do this voluntarily. If I play a super loud sound and you weren't expecting it and jolt, that's not something that you can control. It basically happens automatically. If you show it in its most obvious form, is what we call the startle reflex. Right? We startle when, right, when someone walks up behind us and we didn't hear them and then they say something, right? We all jump when this happens. That's an example of the startle reflex. It's an example of orienting. You can demonstrate this in a brand new baby, right? Just after birth, one of the things they do in the delivery room to make sure that the nervous system is intact in the newborn is to test this particular reflex. And you do it just by taking a baby and you lower them fairly quickly. And what they do is they throw their arms out to the side and usually also cry. Because basically what's happening is that that rapid lowering simulates what happens if the baby's falling. And putting your arms out to catch yourself, right, to spread the impact is something that obviously has survival value. So the orienting reflex is there for a reason. It's there to help us survive. And it's there in lots of different forms throughout life. Here's an example of experiments that are done in animals. This is a rat in a chamber. There's a really loud, big speaker here that can play a really loud sound. And of course, when you do this, the rat will jump, just like we do. You can do this in a much less obnoxious way in people by using a device like this, where instead of playing a loud sound, what you do is you just have a little puff of air blow on a person's eye. You've seen it. What happens when you do a little puff of air like this is people reflexively blink. So it's kind of a mini startle response that your eye is obviously, again, evolutionarily an adaptation to protect your eyes, right? To protect your eye from foreign objects that might be coming at you. So the interesting thing about this startle response is that it happens in this very, very large overt way when the stimuli are potentially threatening. But you see it actually in many, many other circumstances as well. In fact, there's some kind of aspect of the orienting response that exists in any kind of stimulus that's novel, right? So novel stimuli, of course, draw our attention. This is why orienting is considered part of what goes on with attention. So those of you who were watching the screen when I did that probably oriented. Right? This is a very obviously not a startle reflex, but just the fact that I turned a blue dot on up here. Your eyes probably looking at the screen immediately went to that blue dot. This is an example of an orienting response. You're orienting things that you didn't expect to happen in your life. This is actually used in experimentation with babies. In fact, this is kind of the dominant experimental task that people use when they study pre-verbal babies. Is they rely on the fact that babies will orient anything new. So if you're having a baby looking at pictures on screens like this, sitting on its mother's lap, one thing we know that will happen very reliably is if you show a baby two pictures, and one picture was shown on the previous trial, and in this picture, over here on the right is a new picture, the baby will always look to the new picture. Almost always look to the new picture. This is an orienting response. And you can very subtly manipulate or stimulate it to see if that baby's ready to perceive other things. If you show two pictures of cows that are very similar, and the baby doesn't orient to the new cow that you show, that would suggest they're not seeing the difference between them. The cows enough different that the baby can perceive it, you'll notice that they'll orient. So it's a way of saying, can babies perceive this difference? You can't ask them, or we can ask you. We can ask you, do you see the difference between the two cows? And of course, the yes or no. Babies, you have to rely on their orienting response in order to know them. All right. So this aspect of orienting emphasizes what we call automatic attention. Right? It happens without us thinking about it, without us intentionally doing it. It just kind of is a byproduct, right, of there being something new and novel for us to process. And it's mostly involuntary. And in this sense, it's kind of a variation on the fact that there are many stimuli in our environment that actually demand our attention, require us to pay attention to. And as hard as you try, you actually can't avoid doing it. Right? The only way to avoid attending to something novel and visual that's in your visual field is to close your eyes. So what is this for? Why do we orient? What is the biological significance of this? I mean, obviously, in this first slide that I showed you, the startle reflex clearly has survival. But why do novel things in our environment that are occurring for the first time or that are occurring unexpectedly, why would you wire up our system so that we automatically attend to this? Yeah, dangerous things for sure, right? I mean, anything that's novel, particularly in a hospital world, right, the world that we all evolved in, was much more hostile than our current world. These are the kinds of things that might be dangerous. And having a system that rapidly can deal with or can orient to potentially dangerous information gives you some survival, right? It gives you an advantage. You might be able to flee if you need to. You might be able to get ready to protect yourself if you need to. And imagine that you have two organisms, one that has that reflex and one that doesn't. Who's going to get eaten by the saber-toothed tiger? Right? It's the person, the individual that orients and quickly detects the potentially dangerous situation. And the one that's a little bit slower to do that is more often than not going to be eaten. And what does that mean evolutionarily? Well, it means that if you get eaten, obviously your genes, if you haven't reproduced yet, your genes don't get perpetuated into the next generation. And so you can see how a reflex like this could very rapidly predispose an evolving creature to really favor this particular trait, this particular ability. Okay, so that's survival value. It also is important because it helps us bring attentional resources to bear on a stimulus. And we'll get to what that means in a bit. But basically what it allows us to do is it allows us to do really high-level processing to potentially important information. This is what we sometimes refer to as stimulus-driven orienting. In other words, there's something about the stimuli out there that causes us to orient to. There's another kind of orienting, though, that we can refer to as goal-driven orienting. And while the kind of orienting we've been talking about up till now happens kind of automatically by virtue of the stimulus occurring, it's also possible to use probably some of the same brain processes in a voluntary way to help you actually train your attention on something important. So, for example, we can decide to attend to something. Right now, you're all deciding to attend to this screen based upon things that I'm saying. That's not novelty. Or orienting to here, but it's a decision to orient your attention to relevant information in the environment. Of course, you can even do it to internal stimuli, right? Your stomach growls, for example. Or perhaps a thought that comes into your head that, oh, I forgot this particular assignment. I need to work on that. So yes, thoughts can obviously cause us to orient, to change our attention, right, to something different. Most of what we'll have to say from now on actually will concentrate on this kind of voluntary aspect of attention. The idea that attention is flexible and that we can voluntarily move it around in a way that's beneficial and helps us process the world. But before we get to that, let me mention one other kind of orienting. What I'll call covert orienting. So all the previous kinds of orienting were overt, right? They were obvious. We either decide to orient to something or some stimulus in the environment causes us to change our orientation to it in some way. In covert orienting, that's not the case. What we know is that it's possible to attend to stimuli without the direct changes or signs for attending. Well, I'm talking about ability that probably is unique to primates. Probably has been honed to its best in humans. So, this is, probably some type of overt orienting, right? If a walks by, we should check him out. That's overt orienting. Covert orienting, on the other hand, which is a very sexist slide here, I'll admit. But it's one that's true, and everyone knows, we'll all resonate with this one. So here's a guy, here's his girlfriend coming up here, and he sees her coming. And of course, she's looking at him, and he's looking at her, and there's perhaps a cute girl walking over here. Yeah. So what happens? So, obviously he doesn't want to get caught doing something. Right? But he obviously looks at her, he might be in trouble with his girlfriend. But in fact, what humans are able to do is this. We're able to covertly attend to information in the periphery of our vision. Now, we can't do it as well. Your peripheral vision doesn't have as high acuity as your vision in front of you. Nevertheless, we can do this, and we do it all the time. Maybe it was blatant sexist way, but it does happen. That we are actually able to focus our attention on not exactly what we're looking at. So as I look straight ahead over here, I can detect things that are going on in this direction. And of course, one of the reasons for doing this is because we are a social species. And sometimes it's not advantageous to let other members of your organization know exactly what it is you're attending to. This happens also in Hemidreus baboons. Those of you who haven't been to zoo, there's a great exhibit of this particular kind of baboon. They're a highly social baboon with a very rigid social hierarchy. There's usually an alpha male in these groups. There are then also some non-dominant, younger, usually juvenile males. And this guy maintains order, and he does it by threatening the younger males. Sometimes we'll fight, but they don't fight very often. And the reason they don't fight very often is this nice, rigid hierarchy that prevents them from having to fight. One thing that will cause this baboon to attack this baboon, though, is if he starts checking out any of this guy's females that are in the street. And so, again, this baboon will learn, right, that even though he's looking straight over here, he can covertly check out what's going on. In particular, he can very carefully check out what the alpha male is doing. And he can do that with his peripheral vision. And this is something that those of you who are primatologists is a great area of research. But there's lots of data suggesting that this happens in these kinds of species. In the labs, you can actually test this by having people look at computer monitors and focus their vision on a central point here and present relevant information in the periphery. And we know that, this, in fact, this ability to divert your attention to the periphery, even though you're looking straight ahead, is something that we do all the time. And you can actually study it very clearly in cognitive experiments using this technique. All right, so let's move on to the second topic here. Let's see, a few minutes left. Try to get started talking about a second component of attention that I'll call searching. So one of the really important things that we're doing with our attention is we're using it to find things that are of interest and important to us. Right? You're all doing that a lot right now. You're looking down from your notes and up to the screen to find the things that are relevant, the things that I'm talking about, and writing them down. So yes, attention is involved in finding things. And you can actually study this in cognitive psychology using something called a visual search task. So if I tell you, for example, to find the red T, all of you probably almost instantaneously do that here. And interestingly, even with a much larger display, with many more distractors, like the distractors here at the blue, you can still do this, probably very rapid, very efficient. This is what we call a feature search. When the thing that you're looking for has a distinguishing feature that none of the other items in the visual array has, you can do this very efficiently and very rapidly. What we call an easy search. And it appears that we can do it almost automatically, right? It has happened. In fact, this is referred to as pop-out. That red T just kind of pops out of the image without really you having to do much at all to do that. Now find the red T. Obviously, if you do that, you probably did it fairly quickly, but certainly never would you do it as quickly unless you just happen to be looking at this part of the screen when this came on. And of course, this is a much more difficult kind of search. And it's even more difficult, right? As we add more elements, more potential distractions to the display. Of course, all of you see what the problem is here is now the distractors actually share some of the same features as the thing that we're asking for. And that makes the task much more difficult. And this is what we refer to as a conjunction search. And you all know that these searches are hard. And of course, the best example of this type of conjunction search, where you have to actually use what's called controlled attention to find the conjunction of two features. This is what's very difficult. And most of you all know that kind of standard conjunction search problem, right? But where's Waldo problem? Where is it? It's a conjunction search because, right, what distinguishes Waldo are his glasses and his red striped shirt. There's many other red striped shirts that make it much more difficult to figure out this conjunction. Obviously, if we turn it into a simple search, though, right, then none of the distractors have the same colors. Waldo pops out. So here is basically a graph showing you that relationship. So for really easy automatic searches, it doesn't really matter how many distractors you add to the search set, right? You can go up to hundreds. And people are just as fast at detecting a target amongst distractors that differ on a single feature. But as you add distractors for a conjunction search, people get systematically slower at finding the item. Alright, so this is probably a good place to stop. So I'm going to try to finish this up. It's Monday. My plan is to race through the rest of the session, start then talking about the first of our mini memory topics for the rest of the today's lecture, and then I will carry that over into Wednesday's lecture. I do want to reserve enough time to give the lecture on Wednesday that I have a little bit of review, give you guys an opportunity to ask questions and you're not clear on, so we'll try to reserve 15 to 20 minutes to give the class for that. Just to get you up to speed on the exam, all of you will be wearing one of these class forms of class. The exam will be 50 questions that we'll use for today, alternatives, A, B, C, and D kinds of questions. I will post either tomorrow or no later or Wednesday morning something like a study guide. And basically what it will be is there will be like 20 or so questions. The kinds of questions that will be on the exam, so it gives you an idea, gives you some practice with that format. And I will also talked about, some of which I talked about in class. You use those to help you study, right? So those are terms that you need to be familiar with for exam. Now, that shouldn't be your only strategy. Just think of the example. It's just memorizing these key terms. You need to read the chapter. You certainly need to go to the lectures. But I find the key terms are helpful. Most of the questions will have one of the key terms as part of the question. So if you know what that term means, it will definitely be helpful. So we'll talk more about the example on Wednesday. No, it's not. It'll be all strictly learned students as well. I have one very part-time today that actually take the exam and put it into the machine grade. Other than that, if I did essay exams, you guys would probably get the first exam back sometime in June. All right, so let's get on to talking about attention. I'm a little behind, not too bad. I think we'll easily make it through all of these we need to get through by Wednesday. All right, so last time we finished up, we were talking about one of the key functions of attention, or kind of, as I said, 3 different aspects of attention. There's this notion of searching. One of the things attention does for us, it allows us to search the complicated environment that we're presented with. Like in the case of visual modalities, right? It's being able to look out onto a classroom like this and be able to search and find a particular individual. Right? Whereas Waldo is kind of a classic example of that. And researchers that have studied this particular aspect of the searching aspect have found that there really are 2 different basic kinds of searching processes that we engage in. The first one is what I call easy search or automatic search. This is the kind of search that when what you're looking for is so distinct from all of the other things in the environment around you that it almost pops out. The item that you're looking for pops out in its background. And this can happen very frequently in cases where there's some very distinguishing attribute of the item you're searching for. So it's very different in most ways from the things behind it or things around it. So that's an easy search. And one of the things we know about easy searches is that they happen pretty much automatically. And you don't really even have to think about searching for something. It's the item that you're looking for just kind of literally pops out. And another important attribute is that it kind of is about the same difficulty to do this. In other words, it takes you about the same amount of time to find an item like this. If there's only one other item with a distractor, over here, or if there's as many as 7, in fact, if you go out there to 20 or 30 or even 40 items, it could be surrounding the item you're looking for. And if the item you're looking for has this kind of easy automatic pop-out attribute, it really doesn't matter how many items are around it. You'll be just about as fast at finding it if there's 20 or 30, or if there's only 1 example other item. Right? So that's what we call easy or automatic searching. The more typical type of searching, of course, is the type of search where it's not quite as easy, where in fact you have to kind of attend to what you're looking at and come up with some kind of strategy, some kind of systematic strategy to search through the space that you're looking at. Visual searching. And what we know about that kind of search is that the amount of time it takes you to find an item is actually a linear function, increasing function response time. Right? So this is how much time before you actually find the item when the array is presented. The more distractors there are, right? So as you go up from 1 to 2 to 3 to 4, 7, and so on, more and more distractors, you take systematically longer. Each time we add another distractor to the set. So this is why searching in most environments is fairly difficult, right? Because when you're looking for your keys, for example, in your apartment, there are lots of other things that are key-like, right? They're shining, small, shining, bright objects. And you have to look through usually a fairly large search space, in your entire living room, your bedroom. And so you have to literally do this kind of controlled item by item search, right? Look on this surface, is it there? No. Look on this surface, is it there? No. And of course, the more surfaces you have to look on, the more other items in your keys in that search space, the longer it takes to do. So, this is, well, in pretty simplistic terms, the results of a very large body of research looking at the role of attention when researching. So let's move on now to the next topic. There are actually two topics that are somewhat related to each other here that I'm going to talk about. And the first of them is a phenomenon known as inattentional blindness. And probably some of you have seen this particular video before. And by the way, you can, if you want to look at this video again, this was the YouTube link here, so that'll be in the watch. So I'm not going to really play the audio, I will play on my computer here. It doesn't really matter. So just watch the video and follow the instructions that are written. So some of you saw the gorilla, particularly if you've seen this before, right? I mean, obviously, this only works once. So those of you who have not seen it before and didn't see the gorilla, you can now counting passes. So how many people didn't see the gorilla first? It's a normal thing to not see. Yeah, so what we're looking at here in this particular demo is something called inattentional blindness. And the way this works is that even though we have this impression when we're looking at something straight in front of us that we're taking in most of what's going on around us, generally speaking, that's true. But under conditions of high load, in other words, in this case, where you're literally having to count, a fairly complicated thing going on the screen, right? How many times the players wear t-shirts, pass a basketball back and forth. That occupies your attention. That takes up most of your, what we call attentional resources. We'll talk about some resources later. But what happens is that when you're doing that, you actually, even though you're looking right straight at the screen, you miss some of the features, some of the things that are going on. In this particular case, right, a really obvious stimulus that if you weren't having to do something that was very bad, you would easily see that go along across the screen. So yes, this is this idea that attention really is limited in what it's capable of doing, particularly when you put a high load on it. So yes, when you're involved in some kind of difficult task where your attention is being occupied by that task. So that's inattentional blindness. A somewhat related phenomenon is something called change blindness. Everyone sees this before? Most of you probably have not. So this is another video. Let's watch this. It's a crime scene.
I was posting grass in the master bedroom.
I was buffering before it goes the most stressful.
I was planting my potatoes in the rocking chair. Constable, somebody in this room called Smart, who, at precisely 3:34 this afternoon, was brutally bludgeoned to death at Blunt Institute. I want you to tell me your whereabouts and precisely the time that this dastardly deed took place.
I'm hunting drops last minute.
I was buffering towards its goals the most I was watching my juniors in the rocking chair. Constable, arrest. Lady Slide.
Obviously, we were not privy to what was going on in the larger camera angle. And that was, they did pan back to all of the various points there during the presentation, right? So things like coats being changed, flowers changing, roses changing, even the person on the floor was a different person. And then probably most people did not notice those kinds of changes going on. Why is that? Well, according to change blindness, we process much less of a scene than we actually think we do. Right? We have this impression when you see a scene like this one, you're taking most of it in. And you realize you're probably in the English countryside somewhere, right? In a well-person's house. Obviously, a crime is taking place. Lots of cues that are familiar to all of us. Avenue, for example, would be very familiar. But yeah, many things, somewhat subtle in some cases, but in other cases not subtle at all. Changes were taking place, and you were not registering those changes. Again, like the phenomena with intentional blindness, this usually happens under conditions of fairly high load. Right? You're trying to process, what this guy is telling you about a crime scene, and you're processing the characters who are playing the various parts. And that makes you susceptible then to these subtle little things that can change our characters when the scene is switching back and forth. Yes, and when there are no overt scene changes like this, it can happen obviously like this. So, what is this telling us? Well, one of the things it's telling us, one of the things that both of these kinds of intentional manipulations tell us, is that actually our attentional abilities, even though we generally put a lot of stock into them, right? You're fairly confident of what you see in many cases, that in fact, our ability to pay attention is actually limited in its capacity. In fact, if you remember way back in the lecture last Wednesday, one of the things I said is that this is in fact kind of what attention is for. The reason we talk about attention is because kind of implicit in the definition of this term is the idea that you literally cannot focus on all of the possible things that are happening around you at one time. And we've evolved an attentional ability basically to help us deal with the problem of too much information. So what our selective attention, what our attentional abilities are here, is an ability to focus our limited resources, our limited capacity to attend to a few items at a time. And the result of that, things that are going on outside of what you're focused on, in many cases, can go unnoticed, not attended. So yes, when tasks are simple, you do see most of what's happening. However, when the task is more difficult, when the stimuli become more complicated, then we start to get into trouble, right? Our attentional resources become spread thin, as I say. So when objects compete for attention, it's possible to miss some or all of the competing objects. And those of you who find this an interesting topic, there are tons of YouTubes out there on inattentional blindness, on change blindness, that are pretty fascinating. I mean, it really is amazing just how little our attentional ability actually tells us about what's going on in many cases. So we've covered the first two topics. We've talked about orienting, we've talked about searching. Now we're going to move on to the third dimensional topic, which is filtering, the third aspect of the attention. So what do I mean when I talk about filtering? Well, it plays into what we've just been talking about here. The idea that attention allows us to concentrate on one of many potential objects, one of many potential items that might be something that we could pay attention to. And of course, what that means is that when the things that we're not attending to, we're ignoring, or as the term here refers to, we filter those operates like a filter in that it allows certain stimuli to pass through, and allows us to think about those stimuli, to move them on to what I'll call higher level processing, right, where you become aware of the stimulus immediately and do some fairly complicated things with it. While other items that don't make it through this filter, obviously are ignored. And this is what we refer to as the selectivity of attention. In fact, I could call this third aspect of attention selective attention as opposed to filtering. Filtering is a better term here. So what is selective attention? Well, it's basically what it is it's the information that's used to pick certain inputs and to reject others. Right? Selective attention is the selective attention. So some things are passed through for higher level processing, and some things are not. And some of the things that are important in making this kind of selectivity are, for example, the location of things. In the visual world, right, the location of the particular objects you might be attending to. Color turns out to be another important attribute that helps us with selectivity. We saw that back with the filtering, with the searching function. Finding where's Waldo problem as we're looking for the red striped shirt. But also context plays a pretty big role. The context meaning that when you look at a scene, that you immediately make some assumptions, right? So when I look at a view, this is a classroom. I expect to see students sitting in seats. Right? We do this all the time. We use the current environment that we're in to activate previous memories, previous experiences with that environment, and we use that information to help us figure out what it is that we want to select, what it is that we want to attend to. And also the task. Task turns out to be an important variable as well. In other words, what are you trying to do? Right? If you're searching, for example, you're looking for a particular thing. So all of these are important attributes having to do with selective attention. I'm not going to talk very much about any of those right now. Instead, what we're going to do is we're going to turn to another question that people that study selective attention get interested in. And that is, so what happens to all that stuff that we don't select? All the stuff that we ignore. Now, there's this kind of popular notion in lay psychology that, in fact, we don't ever really ignore anything. Like, all the information you've ever processed, all the things you've ever seen, somehow registered in your brain, and there's some kind of record of this. Now, I'll tell you that there's not a lot of evidence for that particular point of view, but it's still a reasonable question to ask. What happens, right, when you don't attend to something? Does it completely, leave the system and never have any impact on what your brain is doing? Right? So did that gorilla, if you didn't see him pass in front of basketball players, does that mean that it had no impact on your mental faculties? Well, it's possible, right? Because afterwards you're probably surprised to see that there wasn't a gorilla. So maybe it doesn't have an impact. But maybe it does. Maybe there's an impact. And maybe what's going on is you just don't remember seeing the gorilla. Maybe your visual system actually did process that object moving through the people. But in fact, you don't have a memory of that. All right, so this question of what happens to information that gets filtered out, what happens to the ignored information, has been studied most often in the auditory modality. This is one of the reasons I'm going to take a few minutes to talk about this, because everything we've talked about so far has been about visual. And of course, implicit in everything I've been saying is that, of course, all of the modalities have the ability to select certain information and reject others. As you sit in your chairs right now, most of you are not aware, right, that the seat of your chair is pushing up on your bum. If I tell you to focus on that, you can probably feel the chair. But most of the time, you're ignoring it. Right? That information is getting filtered out. Unless, for example, you sit there for too long and your light falls asleep. Then you get very aware of it. But under most circumstances, we're really completely unaware of many of the things that are going on around us. And all of our modalities. And it's only when something really big kind of happens, something falling asleep, or maybe a really nasty odor all of a sudden in the room, that we become aware of those other modalities. In other words, we orient to that information. We kind of reflexively respond. So let's talk about auditory selective attention. And in particular, a phenomenon that some of you probably heard of before, something called the cocktail party effect. What is this? What is the cocktail party effect? Well, all of you have this experience. You've been at a party, there's lots of people in the room, there's lots of noise, you're talking to someone, and all of a sudden you become aware of something happening in a conversation, maybe 2 or 3 people away from you. And more often than not, one of the reasons you might become aware is if you hear your name mentioned. Has anyone ever had this happen before? All of a sudden, yes, the radar goes up and you start paying attention to some other conversation. In fact, you might, stop paying attention to the person that's talking to you when this happens. Or it might become much harder to pay attention to those person. This is the cocktail party effect, right? The fact that we all seem to have this kind of uncanny ability to all of a sudden orient to another conversation, even though we might be very engaged in this one. And of course, the question here is, how does that happen? If you're attending to someone standing in front of you, and you're filtering out all the other conversations, how is it that you become aware of your own name that you mentioned? Background noise you normally are ignoring. Right? What does it suggest? It suggests something, basically a point of view I poo-pooed a few minutes ago. Right? The idea that basically it seems as though maybe our auditory systems actually are paying attention to all of this information that's happening around us. Maybe under normal circumstances, we just forget all those other conversations because we're really focusing on this one. So that's one possibility, right? That our brains actually are processing all that other information, but that because of attention, we really only remember the one thing that seems to be happening right in front of us. Unless something like, for example, the name is mentioned, in which case you switch your attention to the other conversation. So that's the basic idea here, is that, is this evidence for our brain being able to process all of this additional information, even though later on we don't remember doing it. This has been studied in the laboratory using a particular experimental task called the shadowing task. And the way this works is people wear headphones like this. And the first experiments done with this were actually done way back in the 1950s. Stereo headphones like this. First presented, psychologists were like, hey, we can use those to try out some of these ideas that we've been talking about selectively. So what happens is that you listen to the headphones, and a message, usually a conversation, so you hear someone maybe reading a story, is played in one of the 2 ears, right? So you hear someone talking in your left ear. You hear a conversation going on in your left ear. And you're told to repeat back that conversation as fast as possible. So imagine you're listening to someone tell you a story in one ear, and your task is basically to say everything that they're saying with as short a latency as possible between when they say it and you say it. This is what we call shadowing. Right? You're shadowing their conversation with your own voice. And as you can imagine, that's pretty hard to do. Right? To basically repeat back everything that's going on and basically someone reading the story to you as fast as you can. And of course, the reason for this is, the reason we're doing this is that will occupy your attention. It's a really difficult, hard thing to do. And the closer you shadow, in other words, the closer your voice is to their voice, the harder it gets. So yes, you're hearing this person talk to you, and you're repeating, this is a sound wave here, and you're repeating back everything that they're saying. That's shadowing. Now, of course, the manipulation in this experiment is that in the other ear, what we call the unattended ear, in other words, the ear where you're not hearing the conversation. After you get going, after you get involved in the shadowing task, now in that unattended ear, we play certain information. So, certain stimuli are presented to the right ear while you're doing the shadowing task. in that. Or maybe we play someone else saying something, another person saying something good. Lots of different kinds of unattended ear stimuli can try. Of course, you can see where this is going, right? What we're going to do is we're going to do this experiment for a little while, and we're going to stop, and we're going to ask the person, what did you hear in that other ear? All right, while you were doing the shadowing task, did you notice that there was something else going on in the other ear? What do you think? Did you notice? It was a really hard task. Did you hear something coming in the other ear? Well, the answer is yes. You do hear something happen. People remember, you stop the experiment after a minute or two and you say, what did you hear in that thing? What shadow went on? What did you tell me what information came through? People could say that they heard something. Oh yeah, there was something. I heard something. We could vary what that something was. It could be music or someone else talking, or maybe something as simple as like a deep, something like that. So after the task, he asks the subject, what did they hear in that unattended ear? So here's what people recall. Sure. Usually they do recall. But they do recall. They can usually tell you that what they were hearing was, they can distinguish between speech, for example, and music. Oh yeah, it was someone talking. Or, oh, I think I heard maybe it was music, I'm not sure. But they can't tell you anything about it. If it was someone speaking, for example, or it's even a very familiar song. They can't tell you what song it was, they can't tell you anything that the person was saying to them. What the person was saying yet. They can't tell if it's the same or a different story. This is the really strange one. A number of experiments that have been done with manipulating the language in the unattended ear. Some people are unable to tell you whether or not the language they're hearing in the unattended ear, whether it was the same one that they were hearing in the shadow ear, or whether it was a different language. Now this turns out to really only be true if the language of the two languages are somewhat related languages. All right, like, so for example, something like Spanish and French. If someone is speaking to you in a tonal language, like Mandarin Chinese, which has a very, very different quality to the voice, people can hear that usually. But the point is that, right, even though something drastic will change in the language can't be detected, it's pretty surprising. So what can they do, right? I mean, these would be very interesting experiments, and you don't need people to detect, oh yeah, I can hear something going on over there. Can't tell you anything about it. So what can people do? So they can hear this change, right, from language to non-language. So people will say, yeah, I think someone was talking, but then I heard, these weird beeps and boops. So they do tend to remember that. They can also tell you about speech that was not really normal speech, so backwards speech. You've ever heard this before when you play a tape or a record backwards? A favorite thing to do, right, was to look at this information included with music to play backwards. People can pick this up. They can say, yeah, I can tell it was someone, it was the voice, but it was really, really weird. Right, and this kind of fits with what I said, that if you were shadowing in English and you heard Mandarin Chinese, which sounds very, very different, you might be able to detect that as well. Changes in gender. So if the speaker is female, so you're shadowing a male speaker and a female speaker says something. People will notice that, yeah, someone speaking, it probably was not the same gender. So what do all these things have in common? Well, these are kind of low-level physical differences in stimuli that people are detecting. Right? Gender change is really a pitch change in voice that people are picking up on. Non-language, right, is really physically different stimuli. Obviously, what they're not able to do is pick up on anything having to do with the content of what's being said. In other words, what we would call the meaning of, when we talk about language, we're in a term called semantics, which is the meaning of language. Nothing about the meaning seems to be coming through in that unattended channel. So how do you explain this? Well, one of the early cognitive theories that try to explain what happens in shadowing proposed something like this. This is a British psychologist by the name of Broadbent. Broadbent, proposed this idea. He said that what happens in selective attention is that very early during the processing of information, probably not long after the information gets to the auditory cortex, the first place in the brain where auditory information is processed, that there's actually a filter, a protected filter, if you will. And the idea of this filter is to get rid of all this extraneous stuff that you're not paying attention to, right? So it doesn't invade your take away from your precious attentional resources. So that filter basically throws away all of this unwanted information. And in the shadowing task, what the filter is doing is basically blocking really any kind of anything beyond the kind of initial physical energy, the physical stimulus itself, from making it further along the process. So this is how Broadbent envisions it here. So basically you've got auditory messages coming in here. There's some kind of very early sensory memory, which we'll talk about later. So this is really early in processing, right after the physical stimulus is being registered in the brain. And here's where Broadbent said the filter is. He said there's a filter that sits here. All of the information comes in to sensory memory, right? So you process in parallel all the information coming in from both ears. But that, because of selective attention, we're able to filter out all the stuff on the unattended ear. And only the information, right? Only the attended message, right? A single stream of information makes it on to higher level semantic processing. And then eventually makes its way into some type of more permanent memory. Right? So this is Broadbent's notion. But the filter, the selective attention filter that's blocking out all this extraneous information is happening relatively early in the processing of auditory information. And this theory has been called a bottleneck theory. You can see why here. I was superimposed right a bottle. And imagine a bottle, right, with a very thin neck on it. You go there to dump it out. You've got a bottle of wine. It takes some time for the wine to pour out of the bottle. It's not like you have a wide mouth glass. You can dump it all immediately. What does the bottleneck do? Well, it restricts the flow of the liquid. In this case, though, it restricts the flow of information. Right? So you've got all this information coming here from sensory memory, and some of it is going to be basically blocked by this bottleneck that occurs due to this attentional filter. This is why we call it bottleneck. Basically, it funnels down information, so really only one stream of information is allowed to escape from the bottleneck. Now, interestingly, one thing that I hadn't mentioned yet in the shadowing task is the original cocktail party observation, right? The fact that your own name seems to have special status. And in fact, many of the shadowing experiments demonstrated this as well. That while people can't tell you anything about what they're hearing, anything about the message in the unattended ear, what people are able to do is they are aware that their own name is mentioned. I like the cocktail party. So even though you're doing this heavy-duty shadowing task, if that other ear your name comes up in that ear, more often than not, people would remember that. You ask them after the experiment, stop, did you hear anything in the unattended ear? They say, yeah, maybe. Oh yeah, I heard my name. Someone mentioned my name. And this is the cocktail party. And of course, if you think about that for a second, what does that mean? Your name. What is it about your name? Well, everyone here's name is arbitrary, right? Your parents named you. You didn't pick your name, well, because somebody changed your name along the way. But in general, it's a very arbitrary thing. The sound pattern that makes up your name is not something that has meaning in the real world. Right? Bob, Mary, Pat, all of those names are arbitrary things that just happen to be assigned to you. Now, they're super well learned. Right? You've been hearing this name all your life. But nevertheless, it's arbitrary, and this is kind of the hallmark of semantic information that has meaning in language. It's arbitrary, right? It just so happens to align this particular set of sounds, with a particular thing, my identity. It could just as well have been rosemary or some other arbitrary sound pattern. The point is, this is basically, as I said, the hallmark of semantics. Arbitrary sound patterns get matched up with things that have particular meaning to us. This is what we'll learn later about languages. This is basically what language is about. And therefore, this is a pretty high-level process. Right? In order to match up those sound patterns that basically register your name to the information you have about, yes, that's my name, presumably happens at a fairly high level. Although, if that's true, how is that information making it through the bottom? Doesn't seem to be compatible with this theory. Lots of the other things I told you about, right? The shadowing test seems to be consistent with this notion. But that one piece of evidence, and it's a pretty significant one, because it's been replicated many times, the fact that you hear your own name does not seem to fit with that notion. So what could we say? Well, I'll tell you that around the same time that Broadbent was coming up with his theory, another British psychologist named Kay argued that no, Broadbent's all wrong. The filter isn't where he said it was. It's not between low-level sensory memory and semantic processing. In fact, he said that everything's processed in parallel all the way through meaning. And the filter, the attentional filter, really only happens here, right at the end, right before you kind of stuff the information coming into memory. And there's a bottleneck there because, you can only feed a certain amount of information into the memory system. You can only do it so quickly. So here you can imagine the K does have an explanation for basically the cocktail party phenomenon. Right? Your name does make it through. And it's only half memory where the bottleneck actually is. But I'll tell you that the K and Broadbent and many of the people that did experiments in this area argue back and forth for 10 years. And I'll tell you that this woman, Anne Streisman, who's actually a student of ours, came up with a theory that I think most people think is more than likely the correct one. She said that yes, there is a filter of sorts, but she didn't call it a filter, she called it an attenuator. It's an attenuator. Attenuator is something that basically just turns the volume down. Rather than turning the volume completely off, it just turns it down. So what she said happens is that information coming in an unattended channel is not turned completely off. It's just turned down to a lower level. And therefore only special kinds of information. Right, so something coming here, like your name, would actually make it into a higher level processing. She called these dictionary units. It's the same thing as with Broadbent semantics. So you can see how this kind of changes the situation here. It isn't about there being an absolute filter, but rather a selective filter, if you will, one that's more like a volume control on your MP3 player. You turn the volume down on the information coming in from the unattended ear, but nevertheless, there's still a little bit of volume. It's just very, very quiet. And only things that have really important personal relevance to actually make a big enough impact coming through to impact our behavior. All right. So let's move on now. We're leaving filtering, kind of entering the last phase of talking about attention. So we're out of kind of three components of attention. So we're done with talking about the searching function, the filtering function, and the orienting function. We're now going to just talk about some other general properties of attention. And one of them I always like to do right before the first exam is to talk about this topic. The relationship between attention and arousal. So what am I talking about? Well, this is the world we live in, right? We live in a world where there's tons of stuff going on around. You guys live in it more at your age than I did at your age. There's just so many things competing for your attention all the time. Your phones, your computers, other conversations going on around you, your television. Many things are happening that are competing for your attention. And of course, under normal circumstances, you guys are talking all pretty skills for what we call multitasking. Well, I'll tell you that's a misnomer. Multitasking is not what it's cracked up. If you were doing more than one thing at one time, more than one thing that requires selective attention, both things you're doing are suffering. Not doing them both optimally. And that's because more than likely what you're having to do is switch your attention back and forth between the two different tasks that you're doing. And that switching comes with a cost, right? There's time And here is a function that I think is a really important one to know. It's actually a function you find in many places in psychology, and it's referred to as the Yerkes-Dodson law. Right? So it's such an important phenomenon that we call it a law. Too many laws in psychology. This one seems to hold fairly well in relating arousal to attention. So this is a graph here. So arousal is along the x-axis here along the bottom. And notice that it goes from low arousal levels to high. And yes, it's indicated here, low arousal would be, for example, sleeping. Whereas high arousal might occur maybe during some really strenuous exercise or some really strenuous mental task. Right? Something that really is pushing you to the limit. So maybe a time calculus test, for example. That would be a high arousal condition for many people. Attention is on the y-axis here, going again from low to high. And you probably have some idea here now from what we've been talking about. If you're really attending very carefully to something, your attention is going to be high, and if you're not paying attention to it, your attention is going to be low. So what this graph shows is the relationship between these two things, right? This curve here that you're looking at is basically the function that relates these together. And what it's telling us is that, for example, when you're asleep or maybe you've just woken up, one of the things you might notice is your arousal is low, of course, but your ability to attend is also not going to be particularly good. You know this when you first wake up. It takes a while, right? You use a cup of coffee or something before you actually are able to really optimally attend to what's going on around you. And so, yes, after a cup of coffee or two, wait for an hour or so, you'll notice that your arousal level is pretty good, close to optimal, and your ability to pay attention is quite good as well. Of course, the whole reason for telling you about this is what happens on this side, right? As you pump arousal up, as you start to get worried about the test next Monday because you didn't study anymore, right? It's Sunday night, oh yeah, I had that test in cognitive on Monday. I didn't study for it. How many people here would that push your arousal level into the anxiety? So what happens to your attention under those circumstances? Well, as it turns out, as arousal levels go beyond the optimal arousal level, right, where you start to now become really nervous and anxious, your ability to attend actually falls off. This is a very well-researched phenomenon. But attention, your ability to focus and to selectively attend will be less than optimal as your arousal level gets too high, as you start to become anxious about what's likely to happen around you. And the kind of proof of this is, right, and how many of you can resonate with this, is that maybe normally when you study, maybe you listen to music, maybe there's even TV on in the background or something going on your computer, and that is just fine. You can handle that. But what happens when you start to get anxious? Your tolerance for other stimuli around you starts to drop off, at least for many people who work this. Right? Your ability to attend in noisy environments, you become less tolerant. Right? So that roommate that might have been talking in the background that wouldn't bother you normally, you're anxious about your exam you haven't studied enough for. That anxiety pushes your arousal levels up and makes it harder to pay attention under these now distracting conditions. So the point is that you want to try to stay out of this side of the curve, right? Just as much as possible. And how do you do that? Well, you try to always do your best studying, or most studying for something, at times when you're less likely to get anxious about it. So this is the plug for doing your studying ahead of time, before you get to the point where you become very anxious about the fact that you haven't been studying. And what happens is that the 2-hour study when you're super anxious aren't nearly as good as the 2-hour study when your arousal levels are optimal and your ability to attend and focus is optimal. So important public service announcement, right, for the rest of your college careers. There is science basically that backs up this notion, right, that waiting for the last minute, putting yourself into a highly anxious situation is not optimal for allocating your attention to important tasks. All right, we're going to move to the last topic now on attention. I'm not going to get very far into the memory lecture. All right, so Posner and Snyder are two guys, experimental psychologists, who made a really big impact on our understanding of the relationship between attention and other cognitive and perceptual processes. And what they actually propose is that there are actually two important processes, or sets of processes that are involved in thinking about cognition when you talk about attention. So their model or their theory is called the two-process theory of attention. According to them, all mental processing can be classified as falling into one of these two categories. And the two categories are automatic, we've already touched on this, and what they call strategic attention. Earlier when we were talking about searching, we called this controlled attention. Same idea. So what are we talking about here? Well, let's think about automatic processing first. Actually, a large amount of people talk about this when it falls into this category of automatic processing. These are mental processes that are mostly used for low-level mental tasks. For example, all of those processes when we talk about object recognition, all of those things that happen right up to the point when you actually recognize the object would fall into the category of automatic processing. But also, many of the skills that you learn, really well-learned skills, like tying a shoe, or writing with a pencil, or typing on a keyboard, these would all be considered automatic kinds of processes. In fact, much of learning, right, really as a young child, involves taking tasks that might have, much of learning involves taking tasks that were acquiring from effortful to automatic. This is what learning is about. Things that are really tough to begin with practice, right? Lots and lots of practice get easier. Playing a sport, for example, the first play of the sport required a lot of effort. Play tennis, for example, learning how to hit a tennis ball, learning how to be in the right place on the court when the ball comes across. All of these things are things that are really effortful in the beginning. And that's why we're not very good at them usually either, because we're having to use all of our attentional resources in order to do these things. But with practice, lots and lots of practice, what happens is the tasks that were formerly very effortful and require a lot of attention become more and more automatic. You don't have to concentrate on the basics, right? You don't have to think about, when I pitch the tennis ball, my arm has to start back here, and has to flow smoothly, and has to contact the ball in front of it. If the tennis player has to do that, they're probably going to miss the ball. It's going to take too much time, too much effort to do that. It's got to be automatic. So, one way to think about this is, what happens when you go from being, a novice at something to becoming an expert in that? This is the process of taking things that are effortful and require a lot of attention to making them automatic. Now, having said that, not everything can be made automatic. Right? No one probably can automatically make a calculus problem. Doing differential equations always is going to require attentional research. But lots and lots of things that we do, right? Lots and lots of the skills and everyday behaviors that we engage in can be done automatically. So what are some of these characteristics of things being automatic? Well, the first is, and the biggest advantage of automatic processing is that it's really fast and efficient. Right? So when you make something automatic, you really have increased your efficiency in doing whatever that thing is. And you can do it very quickly without much effort. Another example, another advantage of automatic processing is that it basically you can do many of these things simultaneously. So while you can only really focus your attention on one thing at a time, automatic processing many things can happen, right? So I'm up here pacing back and forth, and at the same time I'm talking. Right? So I'm doing both of those things simultaneously, and that's because most of the mechanics of talking are automatic, and the mechanics So the processes I'm using to speak are optimized for finding words and producing those words, uttering those words and hear them. But I couldn't use my speech processes, for example, to do a calculus problem. This is what we mean by that. It really means inflexible. That a mental process that is automatic and devoted to one particular thing that you hone and learn and overlearn and become an expert at. You can't use that process for doing something else. It really is a one-trick pony, if you will. It can only do one thing. So what about attentional processing? I mean, why don't we just make everything automatic, right? I mean, that would solve lots of problems. And the answer is that, yes, as I hinted earlier, you can't do everything automatically. Some things require an effortful, strategic attention to do this. And when you do that, of course, some of the attributes of using attention is that it tends to be slower and more effortful. In fact, these things are probably related to each other. Because things are harder, it takes more time to do them. There are also, and this is probably the hallmark of this, one of the hallmarks of this ability, is that it is limited in capacity, right? As we've said all lecture long, we can only attend to so much at one time. Right? Our capacity, our ability to attend to multiple things is very limited. So why bother with it? Well, because according to Posner and Snyder, attentional resources are extremely versatile and flexible. We can use those same attentional resources when we focus on something to do many, many different things. Right? So even if you don't know how to play basketball, right, you can use your attention. Right? If you watch someone else play basketball for a minute, you can use your attentional resources to say, oh yeah, I take this wrong thing, and I'm bouncing up and down with it, and I don't have to shove it with my arm. So you can do something brand new that you've never done before using attention, right? Thinking all along the way, all the little things you have to do to make it happen. So it's extremely flexible. It can be adapted to new tasks in a very short period of time. But it comes with these liabilities, right? If you're trying to play basketball for the first time, you should really be concentrating very, very hard on the mechanics of the game. And you're probably not going to be very fast or efficient. But you'll be highly flexible. Right? You can do new, different things with this aspect of cognition. And in fact, everything starts out this way. No one's born a world-class tennis player. It requires lots and lots of practice. It requires going from an intentional, strategically processed game to one that is largely automatic. Of course, tennis isn't automatic completely, right? The best tennis players will tell you there's lots of strategies, lots of intentional things going on. The difference is, where is a good tennis player paying attention? They're not paying attention to their stroke, or to where their feet are, or even where the ball is going to land. All that's automatic. In tennis, the strategy, of course, is knowing and seeing where your opponent is, and where am I going to place the ball so she can't get to it, right? So she's over here, I'm going to make sure I place it over here. That's where attention comes in. Any really high-level skill like tennis and basketball, or any skill, even writing. If you're writing, you're trying to write creatively, the mechanics of writing should all be automatic. But the actual creativity, right? Putting the right words together, all of that requires this flexible attention ability. So in a sense, that's what automatic processing is about. Automatic processing, according to Posner and Snyder, leaves open it frees up, if you will, the attentional resources to be used in a way that is most beneficial for these high-level tasks. All right, so that's the theory. How do you show this? How can you demonstrate that this is actually what happens? Sounds great, right? I tell a good story up here. But where's the evidence that something like this really is how it happens, other than just circumstantial evidence, as I've mentioned? Well, it comes about through this particular kind of experimental manipulation called priming. We'll encounter multiple times this semester. We'll talk about priming quite a lot. So what is priming? It refers, at least experimentally, to this idea that usually you have to present two stimuli, two items. And you present them very close together in time. Right? So, for example, it could be two words, it could be two pictures. For example, two letters, as we'll see Posner and Snyder's actual experiment. The first item you can present is called the prime. And the second is usually called the target. So for example, here's a really good classic experiment done with priming where you present participants with words like dog. So that's the prime word. And then shortly after that, I present you with a word like cat. And now I ask you to make some kind of decision about cat. For example, I ask you to say, is this string of letters refer to a cat? Now you can probably see where I'm going with this, right? If I show you dog first, and then I told you ahead of time, you're asking, you're going to see the word written under these letters to say whether it's a cat or not. So if I show you dog as a prime, and I show you cat as a target, you think you're going to be faster under these circumstances than if I showed you dog and I show you the word like here? Probably so, right? I mean, the way our brains work is that we tend to try to find relationships between things. Particularly if I give you a task to press a button, one button, these uppercase words are animal names, and one button are not. And so this is a really actually robust finding in much of psychology. People are way faster when they've been primed. If you present someone with a prime word that's related to the target word, people are faster at making decisions. Okay? So that's priming. Dog and cat. This kind of priming, by the way, is called semantic priming. You can probably see why. These are related in meaning. You can also see priming, though, in circumstances like this. Is what we call orthographic priming. That's really a fancy word for the fact that the priming that's going on here is the fact that these two words share many letters in common. People are actually a little bit faster at making decisions about words like dig if they're preceded by a word that has similar letters. So this paradigm has been used to study a lot of phenomena in cognitive science. And here's just a graph of what the reaction might look like. So if I had you making a response to the cat following dog, you'd be much faster, right? There's a reaction time of 575 milliseconds, so roughly a half a second. And when I show you dog and air, you respond to air, it might take you as long as 0.2 of a second longer, 200 milliseconds longer. 125 milliseconds longer to make this decision. So this is the kind of data that cognitive psychologists collect. How quickly can people make responses to these target items when they're preceded by another item that primes it or an item that doesn't prime it. Got this basic idea here? So this is priming. And here's a study that Posner and Snyder did. Basically trying to find evidence for their model of how items prime. So what they did is they just used single letters. They didn't use words. The target letters are presented here, the prime letters here. So you see a prime letter, and then maybe 1 second later, you see a target letter. And what they had people do was people had to make a decision about whether the target letter was a vowel or a consonant. And they had to press 1 or 2 buttons. Vowel button and consonant. So you can see you're going to need to press vowel here, you press consonant here, consonant here. What they manipulated was the relationship between the prime to the target. So in this case, the prime is a vowel, although people didn't have to do anything with the prime. They're just told, look at the prime, don't have to do anything. Only when you see these targets being pressed. So you see a prime, 1 second later you see a target, you say vowel. You see a prime, 1 second later you see the target, you press consonant. And of course, you can probably see where the priming is here, right? So if both of these are vowels, this is going to be a prime vowel. One of these is a vowel, the other is a consonant. This is going to be an unrelated vowel, right? The prime is not related to the vowel. And this is what they call the neutral trial. Neutral because, in fact, all they did was present an empty box followed by the target. So no priming at all here. Basically, you only see the target. They call these conditions valid. Right? So valid meant that the prime and the target were in the same category. So if you vowel Invalid trials were trials where the prime and target were in the opposite categories. And it's invalid because the prime really doesn't tell you what the target is going to be. When you see this, you might think it's going to be another value. In fact, it's a constant. In the neutral trial, you don't make any kind of prediction. You don't say anything about what the target is going to be, as you can see. So that's what they did in their experiment. They actually did 2 experiments. In the first experiment, what they did was 45% of all the trials, and literally in these experiments, subjects in their experiments, participants, actually experienced hundreds, maybe even thousands of trials. Pretty boring to sit for an hour, maybe 2 hours in a chair watching a computer screen pressing valid and constant. You got tons and tons of trials. Some of them were valid trials, some were invalid, some were neutral. And they scored people's reaction time. So they had really stable reaction time. They were really good at this test. So in experiment 1, 45% of the trials look like this, another 45% look like this, and 10% were neutral. Okay, so that's one experiment. In a second experiment, in a different group of participants, all they did was manipulate how many of the trials, how many of the thousands of trials that people looked at fell into the different categories. And in this experiment, 80% of the trials were valid. Right? So 80% of the time, you got a vowel here, you got a vowel here, you can get a vowel here. You got a consonant here, you can get a consonant here. Only on 10% of the trials would they be invalid. In other words, the prime would be in the opposite category from the target. And then still, we have 10% of the trials were neutral. So what's going on here? What's going to happen? Well, the way people respond turns out to really differ as a function of whether they were in experiment 2 or experiment 1. So let's look at, by the way, they called their first experiment the automatic experiment, and the second one they called the strategic experiment. So let's look at the data. Here are the reaction times from these experiments. And this experiment has been replicated many, many times. So here's what it looks like. So here's reaction time. Fast reaction times are down here. Right? So this is when people are really fast. Slow reaction times up here. And here are the 3 conditions: valid, neutral, and invalid. Now let's look at the automatic experiment. The solid line. When the trial was a valid trial, right? It was one of those 45% of the trials that were valid. People were significantly faster than they were on neutral trials. And neutral trials were, people were almost identically the same speed as they were on invalid trials. So this is the pattern here. So why is this? Well, according to Posner and Snyder, this is automatic priming. Right? Even though people are not really paying much attention to the prime, because the primes in this experiment aren't very reliable. They don't tell you anything about what the target is going to be. So people start ignoring them. Even though they're ignoring the prime, however, right? They're not putting any effort into processing those primes. They're still faster if the prime was in the same category as the target. And they reason that this is because this is kind of an automatic thing that's going on. As you've been in this experiment for a long time, you kind of just automatically have learned this relationship. And it speeds you up somewhat when these things happen. However, notice that there's no cost associated with things being unrelated. Subjects aren't any slower than they were in the neutral condition. So while you get some benefit from things being related, there's no cost for things being unrelated. Why is that? Well, if this is automatic processing, it's because, in fact, automatic processing is not limited. Right? You can do this kind of without any cost associated. So you're not going to get slower when things are unrelated than, for example, when they're neutral. But you will get faster when they're related. Now, this might make a little more sense if we consider the other experiment. So, in the experiment where people learn that the prime predicts the target, people actually start to pay attention to this relationship. They want to be faster. So, when they are related, they're somewhat faster than they were in the automatic conditions. Right? So, using their attention to predict the target, and on those trials when their prediction was correct, they're even a little bit faster than they were in the automatic condition. So this just shows showing you that in this kind of strategic case, right, when you process the prime and you use it to pick the target, you get the automatic processing benefit, but on top of that, you get the benefit of having made a conscious strategic prediction. So this demonstrates that attention adds over and above what happens in automatic processing. Of course, what happens when the prime and target are not related, right? Those 10% of trials when the prime and target weren't related, there's a huge hit in reaction time. Subjects get really slow, right? So you get a constant prime and a vowel target, and people just like the bottom of the they can't respond, right? Why? Well, because they got a vowel in prime position, they're expecting a vowel in target position. And when a consonant occurs, they basically have to inhibit their normal inclination to press the vowel key. Right? They have to inhibit the vowel key because they've invested all these attentional resources in pressing the vowel key. And now they have to inhibit that and then use the other hand to press the consonant. Right? And that cost, if you will, is one of the downsides of attention. Right? So you can't switch your attention from one thing to the other fast enough to compensate for these things being unrelated. And as a result, the reaction time is slow. So, not complete evidence, right, for all of the aspects of automatic strategic attention I'm talking about, but it's not. Like the idea that attention comes with some additional benefits, but also with some costs. Because automatic processing doesn't have that. All right, so this is probably a really good place to stop.